membangun teknologi industri laboratorium dan farmasi yang lebih baik.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Ladies and gentlemen, we cordially invite you to be seated because the opening ceremony is about to commence. Ladies and gentlemen, may I have your attention, please? The opening ceremony is about to commence. The Honorable the President of Indonesian Biotechnology Consortium, Mr. Insinyur Master MSc PhD. The Honorable Director of Conservation of Species Biodiversity from Indonesian Ministry of Environment and Forestry, DRH Indra Eksploitasia Semiawan MSI. Representative of Indonesia National Research and Innovation Agency Bali Region, Mr. Tua Malembangun from Eka Karya Botanic Gardens Bedugul, and Professor Dr. Eni Sudarmonowati. Representative of Universitas Nasional, Professor Dr. Ernawati Sinaga MS APT. The Dean of Faculty of Pharmacy Mahasaraswati University Denpasar, Bali.
Recording in progress.
The Head of Alumni Relation and Cooperation Office of Mahasaraswati University Denpasar, Bali. All respected keynote and invited speakers from the United States of America, Tajikistan, the Philippines, and Indonesia. Distinguished guests, honorable speakers, and participants, ladies and gentlemen. Om Swastiastu. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya, may all blessings be with us. Good morning and welcome to the International Conference and Workshop in conjunction with the 8th Indonesia Biotechnology Conference here in Prime Plaza Hotel Sanur, Denpasar, Bali, today on the 16th of May 2023. First of all, we would like to express our gratitude to the Almighty God for His blessings so this important event can be conducted. Ladies and gentlemen, it is our pleasure to gather here in this special occasion as we will be hearing many insightful research about biodiversity and biotechnology delivered by remarkable keynote and invited speakers that are profound in their own respective fields. We also appreciate all of the presenters that have submitted your research papers. We believe we all will share fruitful knowledge together in this conference. Ladies and gentlemen, to begin this opening ceremony, we invite distinguished guests, speakers, and participants to rise for the singing of National Anthem of Indonesia. Ladies and gentlemen, please remain standing as we move to the next agenda, the singing of the March of Indonesia Biotechnology Consortium.
Kami datang dari Biotek Indonesia, siap berkarya pada nusa bangsa, membawa perubahan untuk kebaikan dengan keikhlasan hati yang utama, makmurkan bangsa dengan sumber daya alam, dengan kemandirian nusa bangsa. Mari bergandeng tangan untuk memajukan negara dengan Biotek Indonesia. Dengan semangat bekerja, dengan penuh keikhlasan, tak pernah kenal lelah dan maju terus. Utamakan pelayanan, efektif ruang yang ada, kami pasti akan bertumbuh subur. Rintangan tak menghalangi, semua pasti dilalui. Maju terus Biotek. Dengan semangat bekerja, terus. Utamakan pelayanan, efektif ruang yang ada, kami pasti akan bertumbuh subur. Dengan tak menghalangi, semua pasti dilalui. Maju terus, Biotek.
Ladies and gentlemen, please remain standing as we move to the next agenda, praying. Let us pray according to our belief. Praying begins. Praying finished. Thank you very much, ladies and gentlemen. Please kindly be seated. Ladies and gentlemen, Bali is very popular with culture and tradition. One of them is our famous Balinese traditional dances. Well, ladies and gentlemen, please allow us to extend our warm welcome with a beautiful Balinese dance about peace in the world symbolized by fragrant flowers. This is also a popular welcoming dance in Bali, and today we'll be performing the dance are the students from Mahasaraswati University, Denpasar, Bali. Ladies and gentlemen, please enjoy Sekar Jagat Dance. Distinguished guests, ladies and gentlemen, proceed to the next agenda is the opening remark of International Conference and Workshop in conjunction with the 8th Indonesia Biotechnology Conference 2023, delivered by the President of Indonesia Biotechnology Consortium. Unfortunately, the President is not able to be with us directly today due to other urgent agenda, but no need to worry because the President has prepared us a video of his remark. Therefore, ladies and gentlemen, let us listen and watch a video of opening remark prepared by Mr. Insinyur Master MSc PhD.
His Excellency, Chairman of Biological Safety Commission and also Chairman of Advisory Board of IBC, Professor Dr. Bambang Prasetya, the Honorable Director of Species and Genetic Resources Biodiversity Conservation, Ministry of Environmental and Forestry, Republic of Indonesia, Veteran Dr. Indra Semiawan, Professor Dr. Eni Sudarmonowati, and Dr. Masteria Putra BIN Indonesia, Professor Dr. Enamati Sinaga from the University of National Indonesia, Professor James Simon, PhD, Professor Ilya Raskin, PhD, and Professor Erin Vogel, PhD, and Rong Pais, PhD from Rutgers University, USA, Professor Philip Bradley, PhD, and Paula Bradley, PhD, from Louisiana State University, Professor Hedler Zwittke, PhD, from National University of Natural Medicine, USA, who three of them unfortunately could not join us here in the last minutes due to sickness and family pathway, but they will deliver the presentation via Zoom or recorded video. Professor Safik Daskendov, PhD, from City University of New York, USA, Professor Hiroshi Kamikahara, PhD, from Japan, Professor Igede Putu Wirawan, PhD, the University of Udayana, Dr. Sultia from Tajik National Medical University, Tajikistan, Dr. Inas Ludin from IBC and also IRI, Alan Lam, Become Culture Life Sciences, PT Martek Indonesia, Conference Partners, UNAS, BRIN, UNMAS, APN, NIH, Artek, Elokarsa Utama, Crop Life, Singenta, Corteva, Agri-Science, Pandu Bioscience, PTIS Science Indonesia, CG Bio, Indotech, Indolab Utama, and Map. Distinguished guests and participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu. Good morning, ladies and gentlemen. Allow me in this opportunity to welcome all of participants by first praying our grateful and praise to the Almighty God for His blessing, grace, and mercies that have made us possible to gather here in this room in excellent condition and health. It my great pleasure to welcome you to the International Conference and Workshop in conjunction the 8th Indonesian Biotechnology Conference, 16th and 80 May 2023 at Prime Plaza Hotel, Sanur, and Eka Karya Botanical Garden Bedugul, Bali, Indonesia, around the theme Crossroads of Biodiversity, Biotechnology, and Health for Enhancing Sustainable Development. Distinguished ladies and gentlemen, it is my pleasure as President of Indonesia Biotechnology Consortium (IBC) to introduce this international conference in collaboration with the University of National Indonesia, the National Research and Innovation Agency BRIN, Rutgers University USA, and its funding support from APN and NHA. I'm sure this conference and workshop will have an important outcome and bring out the best from all participants and lead them to engage in the future collaboration. Moreover, may I mention briefly that Indonesian Biotechnology Consortium, as member of University Research Institute, government institution, industry, and business sector in biotechnology. IBC is purpose to promote and develop biotechnology in Indonesia through research, education, policy, industry, and business. Regarding this international conference on biodiversity, biotechnology, and health, be partly supporting national development and sustainable development goals, SDGs. Allow me first to welcome all the participants of this international conference have assembled here to deliberate on many problems and scientific solutions concerning biodiversity, biotechnology, and health. It is an honor and a great pleasure for me to meet you here. May I also thank all of you, honorable ladies and gentlemen, for your participation, which is evidently motivated by sincere interest in the scientific as well as industry problem related to biodiversity, biotechnology, and health. To our sponsor who support this moment, my sincere appreciation. I would like to thank all the members of the steering committee for their idea that have culminated in the structure of this activity. To the chairperson of organizing committee and all members of committee who have worked so hard, I would like to extend my congratulations on success to make this seminar and exhibition happen in properly organized way. Distinguished ladies and gentlemen, in closing, I would like to wish you very fruitful conferences and workshop filled with the spirit of sharing, listening, and constructively responding. With the remarks and handful of hope that international conferences and workshops will give all of us fruitful results to support sustainable use of biodiversity, biotechnology, and health. May I now declare the international conferences and workshops officially open. May God bless us all with good health to make this event successfully and enjoyable. President of IBC, Master. Thank you very much.
Mr. Master from Indonesia Biotechnology Consortium or the representative, Professor Dr. Eni Sudarmonawati from Indonesia National Research and Innovation Agency, Professor Dr. Ernawati Sinaga from Universitas Nasional, Mr. Tua from Eka Karya Botanic Gardens Bedugul, Dr. Ayomi from Mahasaraswati University, and Professor Ilya Raskin from Rutgers University to come forward to strike the gong 3 times.
So this is just the ceremonial things. This is Indonesian culture. Yeah, so it's not just Balinese, but also in other part of Indonesia, just to wish that the event will be going smoothly. Okay, so here, so everybody may be part half in here and half in here. So here, you may be next to me or something, yeah. Berapa kali nih?
3 times.
Yeah, we're going to strike the gong, we call it the gong, yeah. So in the past when there's like the election, 3 or 2 can be related to the political parties, but this one not the election yet. So I will strike 3 times, okay, with the wish. Bismillahirrahmanirrahim. Okay.
Ladies and gentlemen, hereby with the striking of the gong from Indonesia Biotechnology Consortium, Indonesia National Research and Innovation Agency, Universitas Nasional, Mahasaraswati University, and Rutgers University, the International Conference and Workshop in conjunction with the 8th Indonesia Biotechnology Conference 2023 is officially commenced. To all the honorable guests on the stage, please kindly return to your seat. Ladies and gentlemen, we believe that this is the moment we all are eager to have in this conference, which is the keynote speaker sessions. In this agenda, we will hear 3 presentations from 3 remarkable keynote speakers, which we will introduce one by one in their own session. Let us begin this session with our first keynote speaker coming from Rutgers University, the United States of America. He will present us his research entitled Indigenous Plants for Health, Nutrition, and Income Generation, Case Studies from the Bush for Biodiversity Preservation African Case. Before I welcome him, I would like to introduce you a bit about our speaker today. Our speaker today serves as Director of the New Use Agriculture and Natural Plant Products Program and as Faculty Director for the Rutgers University Core Analytical Facility for Natural Products and Bioanalysis. He also serves as the Director of the Center for Agricultural Food Ecosystems, whose mission is to bring a uniquely diverse and holistic approach to researching food systems and designing innovative and culturally appropriate methods to sustainably nourish our growing human population in balance within our global ecosystems. Therefore, ladies and gentlemen, please give a big applause to Professor James Simon.
Quite a pleasure for me to be here this morning. I wanted to thank the organizers to invite me. This is my first time in Indonesia. It's my 138th trip to another country to do field work, even though I'm not doing field work here. So I'll give you a different perspective of what happens in sub-Saharan Africa. It's impressive to see the science and the technologies that you already have in Indonesia. It's very impressive. Where I work in sub-Saharan Africa, we're not quite at that level in terms of science and bringing things together. So I want to give you some examples from the bush. But what directs us and what moves us and motivates us is to ensure, as you're doing here, is that the environment is safe, that people have food, people can live with dignity, and with safety, and that we could utilize plants and the environment in a manner that fosters good relations with all. So that we hope that there'll be lessons from sub-Saharan Africa that might be of interest for you here in Indonesia. So my topic will be indigenous plants for health, nutrition, and income generation, case studies from the bush. The picture on the map is the remaining 60% of the Guinea rainforest. The rest has been chopped down, has been replaced by oil palm, has been replaced by other anthropogenic activities, and that forest is now lost for good. It's lost to flora, it's lost to animals, it's lost to those people that are living in this area. So the question that always challenges us is, how do we as scientists not only work on basic and applied research, but to make the public aware of what is going on in the bush or the forest? How do we utilize science to help drive public policy? And it's a very complex problem. If I recall how to move this forward. So, how many of you have been to sub-Saharan Africa? Raise your hand. Okay, so we have one in the audience, okay? So even though many Americans think of Africa as a country, it's actually a continent, and there's over 50 countries and 800 languages, okay? To you in Asia, that's not a surprise, but when we look at the ecological ranges, we go from many different savannas to deserts to temperate rainforests. We have some of the most rainy areas in the world to the driest areas of the world, and because of that, it is a vertical treasure of plants, aromatic, medicinal, and food plants that can offer promise to people for health and medicine in the future. And because it's sub-Saharan Africa, and it's not on the big tourist area as you see here, and all over Asia, the plants here are probably least studied relative to continental work around the world. So the challenge is, how do we utilize these indigenous plants, and why do we bother? So our hypothesis is that there's a strong role for traditional and indigenous foods. Now, whether they're truly indigenous or they're really naturalized, and they've been here for hundreds of years, just as your capsicum peppers are not from here, but they're from the Americas, but moved here. So many of your populations see them as indigenous plants. We use this term loosely. So what is the role for traditional indigenous foods and botanicals to alleviate and to mitigate aspects of hunger, income generation, food insecurity? And we believe, and we have evidence to show that they can increase income significantly. They could diversify diets to provide opportunities for improved vitamins and minerals and bioactive phytochemicals. They could adapt diets to environmental stresses because the world is seeing significant changes with increasing rapidity with climate change. And many of these indigenous plants are by themselves resistant, or some degree resistant, to abiotic and biotic stresses that accompany such environmental changes. And we could utilize those if we do that purposefully to enhance conservation, environmental protection, and provide economic arguments for public policy. What do I mean by that? It's very easy for you as the scientific community and the scientific leaders in Indonesia, and for those of us at the university level in America, to talk about the importance of biodiversity. But when you go to these rural communities and people are just trying to eat and not starve, and people are trying to develop, it becomes an easier argument if you could show that there's something in those forests that have economic application in order to help drive public policy, rather than just say, we want to be able to have this forest preserved, okay? So this is our hypothesis. We could argue, we could debate, but our hypothesis is that by providing economic, whether it's ecotourism, whether it's plants, whatever the view is, that provides an economic reality to it, it gives better opportunities for public policy to change. And many of these indigenous plants we consider to be minor or orphan crops. So I'm a plant breeder and geneticist. You won't hear a lot about this today because we'll be talking about case studies from the bush. But these orphan and non-domesticated plants, through the understanding of genetic diversity and selection, not even breeding, can contribute and move into commercial opportunities for disease resistance, as you see a few examples in the future. So Africa and Asia do share the burden for forms of malnutrition, okay? In Asia, 56%, Africa, 36%. More than half of stunted children under 5 live in Asia, and more than a third live in Africa. In 2015, almost half of the overweight children under 5 lived in Asia, and a quarter lived in Africa. And in 2015, more than 2/3 of wasted children, that is relative to stunting, came living in Asia, and 1/4 lives in Africa. So clearly, if we want to utilize biodiversity and biotechnology and the importance of agriculture and plant science, we have a lot to do. It's funny, we have 2 slides moving on both slides, but not the slide in the middle, but eventually it comes to us. We have a hypothesis, because when you get grants from federal agencies, you always have to come up with an acronym. So the acronym we came up is the 4 A's. Okay, in America, if your car breaks down, you call it the AAA. Okay? Here in Asia, you can't do that, so we call it the 4 A's. It's access, affordability, availability, and adoption. Adoption to us are those people that are living in the bush or the forest, or growers or collectors that go and collect. The consumer adopts by changing their purchasing power, bartering power in order to consume more. But using these 4 A's, then we address issues of sustainability. And we define sustainability with 3 pillars. An economic sustainability, but then the environmentalist and the ecologist would be an outrage, because where is the environmental aspect of it? So we include environmental sustainability, and then we also include cultural sustainability, because environmental sustainability without economics has not been that successful. Economic sustainability without a consideration to culture and environment has led to a disaster. Cultural sustainability without the other 2 seem to be very limiting. So sustainability, which is a word so often used but not defined to us, our models embrace from the beginning an environmental, an economic, and a cultural sustainability in order to move things forward. And we do this using a feeds model. Now often, you as scientists, myself, we come up with models that look very linear and very static and well planned. But we know when we do research, it's the observations along the way. It's the observations we see in those tributaries along that main river that we bring together, so that after many years, we have a workable model that's flexible, opportunist, and based on real hardcore data. And we've published this feeds model because we use this environmental, economically sustainable food systems. And what I'm going to show you now is work that over a course of a decade and a half have generated over $40 million into the pockets of rural African communities where people had no jobs, had no access, but now have the ability to choose what they want to do with those economic opportunities that came from the forest. Our approach, even though I'm speaking here, this approach is multidimensional and very holistic. It involves many different people. It's a very humbling experience to realize that we act in a certain way, and together synergistically, just as this conference is trying to do, bring people together. That is, often we are hopefully more than the sum of our parts, but we bring together the private sector with people that champion from the community and from the private sector and from the government and the international government together, including the universities, to work on problems, these complex problems, to create solutions. Our research, rather than just only being very basic, which we do, we try to tie it into market development for this economic sustainability. So in concert with our communities, they define to us what they need, rather than the national government or the United States Department of Agriculture defining what's needed. We work with the communities for them to determine what they need at that local level. And so an example, we'll go through a couple different examples. We work on plants that could contribute to the nutritional benefit if consumed on a regular basis. We know that in the African indigenous vegetables, or African indigenous botanicals, there are many limiting minerals and vitamins besides phytochemicals. Relative to their daily simulated allowance, we have a whole range of different plants that are nutrient dense. We survey these communities and we ask the people, what do they want to eat? Now in Indonesia, you could drive by and you see a plethora of a variety of different horticulture and different plants. Where we work in Africa, you don't see this diversity. You see 1 or 2 crops that if you take a picture of the plate, it's white or it's brown, okay? If you take a picture of a plate here when you eat food, it's colorful. Why? because you have different plants that contribute to each person's meal, even though you are obsessed here with rice too, by the way, okay? They're obsessed with rice, they're obsessed with cassava, they're obsessed with plantain, but when you look at it, relative to the minerals and vitamins, it's still limiting. So we know that when we think about what are the key food environmental, what are the key elements, it's affordability, it's accessibility, it's having these sustainable properties, it's quality. It's not just what we as scientists can determine to be rich in their nutrient density, but they have to taste good, and they have to be accepted, culturally accepted, otherwise it doesn't matter. And they have to be convenient. So we utilize these foundations that other people around the world have looked at relative to food choices and understand what these external and personal food environments are. And then when these contexts of looking at these different domains, then we focus on the actual work that we want to get done. So using an example, just in African indigenous vegetables, okay? First we'd go out and we always do a SWOT analysis with communities, with our cohorts, our brothers and sisters in those countries, and we ask communities in urban, peri-urban, and rural communities, what do they want to eat? What would be their food preferences, particularly prior to colonial times? What would be their preferences if available? Okay? And then we ask them, what do they spend their money on? So we do a market analysis, a multi-generational analysis in the native languages, of course. In Africa, just in Zambia where I work, there's 42 languages. So we go and work with people that speak those languages, that have the trusting relationships. We do the economic surveys of what's there. We understand what the gaps are. And then we custom tailor our designs to overcome those limitations, those gaps. And then we come back and we verify it. So I'll do the first story as case study in Senegal. Now I know you appreciate roselle here. I was given roselle tea upon arrival. I've worked with hibiscus, or what we call bissap in French, or roselle, for over 20 years. And in Senegal, that is one of the local teas, okay? We did a SWOT analysis, and there the women were only allowed to grow a little bit of roselle outside the garden for the men, okay? So working with the Women's Foundation, the President's Foundation, this was funded by the President of Senegal and their Ministry of Health and Nutrition. We analyzed what the hibiscus environment would be, what the potential was, and we recognized what the health benefits were. You are recognizing in terms of their phytochemicals for cardiovascular, traditional use in hypertension, inflammation. We know the calyxes have a very strong health potential besides just tasting really good. They have antiviral. Studies at Rutgers, we show that it's really good against hepatitis as well. And there's been little focus on the leaves, despite its popularity as a fresh leafy green. What do I mean by that? Communities will grow the hibiscus, and they won't eat the leaves in this area. They only eat the calyxes. So working with leading champions, women's communities, bringing them together, this is some of them, talking to them what they wanted. Before our interventions, they had no selection, no uniform genetic material, and after low-cost practical interventions, here in this biotech, you're going to hear through many of the other speakers, not necessarily through me, cutting-edge science. And cutting-edge science is exciting, but when I go to these rural villages, they don't need cutting-edge science, they need applied science. They need improved genetic material. They need improved water management techniques. They need improved soil management so they could utilize the indigenous plants they're doing, while you at the university can improve and release improved materials. We did that through genetic selections and breeding, and after our interventions, we had much higher yields. We adapted for the very dry desert conditions. We introduced simple labor-saving devices. Now again, here I don't know if it's women or men's role, because I've only been in Indonesia for 48 hours, by the way, okay? But in Africa, it takes a long time to harvest your hibiscus for the calyx. So what we did is we took pipes, food-grade pipes, that had the diameter slightly larger than the seeds, and we put in a rubber bulb, and we were able to pop the seeds out, so it saved them 6 hours a day. Now to a woman who's cooking very early in the morning at 5 o'clock, who has to go back into their village to cook lunch and dinner, and yet the farms are not outside the house, this 6 hours is a lot of work. That's the difference between profitability and not profitability. We're not talking about high-tech science, are we? But we're talking about applied science that people can use and giving us good quality just for a labor-saving device, okay? In very short time, impacting over 4,000 women and was declared the Senegalese national crop, we began to develop organic certification where this material went to Europe, which is not easy, to Japan, which is even harder, and to the United States as organic certified material. And why do we do organic certified material? We do organic certified material not only because the communities wanted it to be organic, not only because there was market demand for organic, but the organic material that requires more time, more effort, and costs more also generates a higher price, as you can see in this histogram. So this is a histogram showing that the greater attention you do to quality, the higher prices at the farm gate the person has, as well as showing a picture for the first time of EcoSort and NOD for USDA certification to go into Europe or America. So we're very proud of this. And right now they are able to have systems where they have very specific varieties that have the colors that the industry wants. Now again, here comes the science of what is the color. We know that the hibiscus is due to 2 anthocyanins. Only one is really commercially available, so in our lab we make our own standards. And we could track the hibiscus in the different varieties, or we could develop mutant or anthocyanin-free hibiscus that they call green or white, where you want the taste and the flavor, but you don't want the red color. So this is available now over there. So what the communities tell us is this type of work brings us together, okay? In a short period of time, they were able to achieve social empowerment where the women earned more money, they maximized profits, and during a very dry year, and during a desert country, where the peanuts and the cotton and the corn yields went down, the hibiscus suffered, but they still had a profitable crop. Now, once we look at the calyx in other countries we work, the communities eat the leaves, okay, as a leafy green. And when we look at the leaves and we look at the phytochemistry in the leaves, we find, not surprisingly, but very delighted, that the leaves are full, chock full of a whole range of different phytochemicals, not unlike what the leaves are, but even more, very rich in a whole range of different polyphenols. And we've characterized those, as you can see here, or you won't be able to see it that closely, but in order to quickly pull out the genetic diversity, again, the conference is on genetic diversity, on biodiversity, we look at the biodiversity of each of the species, we grow it in a particular area to make sure it's climate adaptable, and then without a lot of fancy breeding, we do a lot of breeding, by the way, but why spend 10 years doing breeding where we could just pick and choose what something shows good and has good field performance and then can be packaged and lead into an economic trade. And then we do the more fancy SWOT analysis, and this is where you could see where the products end up in another country. That is going from the grower level all the way to local trade, to regional trade, international trade. This is very exciting on a crop that you are very familiar with, where the natural products are used for cheeses and for meats, flavorings and teas, and also of and by themselves nutraceuticals, and some, as Illy is always interested in, these new cosmetic areas with shampoos. Another quick example would be this is taking a field trip in Namibia. Now, Namibia is in a, you can see in the map on the bottom right, it's toward the southern part of Africa, above South Africa. And this is a project in which Professor Raskin got me involved in. I always appreciate traveling together and getting involved in the GBEX and the Ramses test that you do here. And it's such a delightful project and such an informative project because in a short period of time, it helps build trusting relationships between these rural communities, scientists, and ourselves from another country. So this is just in one, we did a lot of different plant expeditions. This is just showing and going into an area that's a little different than the rainforests or the mountains that I've seen in Indonesia, where we collected over 110 different species of plants over a course of a year. We characterize them, we botanically identify them, and we went after them with using a selection criteria because we were tasked by the US government and the government of Namibia to collect all the plants in the country. Now in Indonesia, that would be very daunting. In a desert country, it's a little easier because the diversity is not that high and it's more concentrated, but we have to go to these areas when it's flowering to get a good taxonomical species. And we used a selection criteria because we knew that there were plants, particularly medicinal plants, that were exported from Namibia to Germany for liver and other areas that they didn't need our help. We wanted to go into lesser explored ethnobotanical crops. So using a selection criteria, making sure we did everything properly with research permits, okay? We introduced into this country, as Professor Raskin and the colleagues here have done here in Indonesia, that Ramses, or what we used to call the Screens in Nature Technology, and we had 12 undergraduate and graduate students do their thesis on it in a short period of time. And this just gives you some update that is looking at it. What's beautiful about this, it doesn't confirm, but it helps validate why people use these plants in a very qualitative approach on certain assays, okay? It gives us an indication of what we want to be able to test. A different vignette, okay, would be going back up into the desert, into Sahel, and we always wanted to look at the medicinal and aromatic plants that we use, but only used locally. And here we worked on Combretum microcanthum, and this is something that's very exciting, because this is the bush tea of Senegal, okay? Most people outside Senegal and in the tiger bush area where this is grown would not know about this plant. Doing the typical classical ethnobotanical work, we were developing it into teas, and we know that you have a certain preference for the teas you like, for the taste. Different cultures have a different preference. When we go out and collect these, the different populations, this is all from the wild, have a different taste and characteristics. Some are very bitter, some have a different aroma, and so we put these in clinical, in a century, the equivalent of a century trial, to be able to understand what people wanted. And from these different populations, we collected the Kinkeliba. But importantly, this is just a wild shrub, and this is an area that's now being threatened by anthropogenic activities. Boab, tamarin, Kinkeliba. But now showing that the plant is rich in anti-inflammatory activity gets exciting, but a lot of plants are very exciting in anti-inflammatory activity. When you extract these plants using a potential like ethyl acetate extract, we find many of the same condensed tannins that you'd find in teas. That is a Chinese tea. That's exciting. But when we extract it and we find that it's rich in antioxidants, but we find that it's rich in using other solvents like a butanol extract, we find that it's rich in new compounds that were never explored before, okay? And through using mass spec and NMR, we identified that there were whole new skeleton compounds, these piperidine flavone alkaloids, a whole ring structure and individual components within a plant that we got to name to give honor to the plant. We call it kinkeloid A, B, C, and D. And these compounds are very biologically active. Collaborative work with the Raskin's lab show that they have very strong active for glucose lowering activity, okay? And in concert with others, we found that we have patents in many different papers on the Kinkeliba extract just for anti-diabetic agent, as an agent. Now this and of it by itself may or may not increase market potential because people are drinking it as a tea. But what's interesting about it is doing more studies and working with a Chinese company. It's got another patent where we're looking at the cosmetic applications of this. And this is a picture of this product, okay, that you can see in the upper left. This made the cover of Vogue magazine. Now in Indonesia, you may not know what Vogue is, but in America, it's a beauty magazine, an upscale magazine that women would use for cosmetics. And it has an unusual polysaccharide as well as these unusual flavone alkaloids that together look very exciting. And when we work with these companies, they now buy the product from the communities that harvest it from the wild. So this is exciting, right? It's using science into action, bringing it back down for development. Another example is we move in to West Africa, again in that Guinea rainforest area. It's Combo, Picanthes anglinensis. This is called African nutmeg. Now in Indonesia, you're famous for nutmeg. You're famous for clove. You're famous for spices. So for me, it's been a dream to come to Indonesia for these years because for the last 30 years, I work on spices and medicinal plants, okay? But here, it's called African nutmeg, but it has nothing to do with nutmeg. The chemistry is not nutmeg. And when you first boil the plants, it smells like chocolate. You all enjoy chocolate. But when the volatiles of chocolate dissipate, you now start to smell fatty acids that smell like fish. And so the whole area is surrounded by cats coming in because they want to eat it, okay? But what's interesting about it, it's traditionally it's used as for body aches. So we knew there was something there. It's a wild tree. This is a tree that people don't often use. But America was interested in it because it has two compounds, two short-chain carbons called myrstillic acid and myrstilate. These were used as rubefacents, okay, for anti-inflammatory activity. But it's not that exciting. It's only a few $1 million a year of an industry. But the waste product that's thrown away, that's used in candles, is this dark, smelly stuff. And you take a chemist like myself and you put it next to something black or pigmented, that's when the excitement happens. Because when we looked at this stuff, oh, by the way, these are 2 different cooperatives we started with NGOs in the area because they were selling the CMO now, which is now exported to the United States and to Europe. But rather than throwing away this other, what was called a waste product, in concert again with a local pharmacist, because we always do everything in concert with our brethren at the universities. This is University of Ghana at the pharmacy school. We isolated this other material, and we find that it's very rich in these vitamin E-like suguanic acids. Now this is exciting, because these suguanic acids are used in a variety of different health applications, just like chondroitin and glucosamine are used, okay? But they're much more potent and much more biologically active. They're a little bit unstable, so we made some synthetic derivatives from it as well. But in the end, we were able to do animal studies and find that it's not just good as an anti-inflammatory, but it's very excellent as for a neurodegenerative, a whole platform of compounds that could be used for different, particularly for strokes and other aspects of, just from the extract itself, of derivatives of these very novel products from what was a waste product. Again, if we look at the overview, whether it's Liberia or Ghana, we all are challenged by the same as you're challenged here. You have a product that's of interest, and how do you really get it to the marketplace? And there's a lot of actors and people along the way that are involved in it. And so I'll give you one example, another example of, again, plants that could help provide a compelling argument to preserve an environment. This is Griffonia. This is now over a $10 million industry each year. It's a wild plant both in Ghana, Nigeria, and in Liberia, but 95% of the world export now comes from Ghana. It's a source of 5-hydroxytryptophan, otherwise known as 5-HTP. It's used to treat serotonin deficiency. It's used to treat headaches and weight control, with not a lot of good evidence, by the way, that it does either any of those, but it hasn't stopped the market for people wanting it. And it's a weight loss and antidepressant and anxiety, okay? So this is a chemical compound that's down below. It's very easy, but the materials were harvested very crudely. People didn't know what to harvest, when to harvest, how to save it, because you get it from the falling floor. So using laminated areas, again, Indonesia is very different, but where we work, people are not literate. So giving them scientific guides don't help. So we use color guides that are laminated. This might be the only picture they have in their hut where they live, with crosses and checks as to what they can do to move toward good agricultural practices. The lessons we learn is that it's easy for people to follow pictures. It's easy for people to learn how to dry and to utilize the material. I'm going the wrong direction. And in the end, what do we have? We have a picture. This is a make-believe picture. This is the only PhD forest in all of Liberia in the blue shirt, Dr. Sam Kofer. And he's making believe he's buying a bag. And in there is a bag of Griffonia seeds. Now that bag of Griffonia seeds can feed a family for a month. That's not a lot of seeds, is it? My point is, here's a plant that people in a village would not know has value, has a tremendous amount of value, and with small interventions and the right connections, they could have products. Now this is a product coming out of Liberia. You could see in the packaged, in the truck, and you could see, I'm not trying to advertise this company, by the way, but last night was the only picture I pulled out. There's over 22 companies that have different products of Griffonia in the United States, and all of it comes from these forests. And these forests now, with the buyers, work to make sure the material is harvested in a sustainable manner, or else they won't buy it. So there's a connection between the buyers, the sellers, and the communities. We do the same thing for Vokanga. Many of you in the pharmaceutical industry know about the different alkaloids that are used to try to get you eliminate some of the opioid addiction from Volcomine. And these plants come from Vokanga africana, and this is now over a $10 million industry, okay? And by introducing low-cost, easy scientific interventions, but coupled by very specific national grades and standards, and now the nation of Ghana submitted these ISO standards to the International Board for ISO, so the country can be able to define what is a good quality product. And you can see the interventions we show here are not excitingly terrible. They're easy, okay? And again, the same checks and balances for Vokanga, okay? Wild plant, not cultivated, but collected. And in the end, this gives you some of the detail of the baseline interventions, going from transaction time being shorted, export price at $6 a kilogram, now it increases to $9, going from less than $1 million to now more than $10 million in industry. So this is tremendous value in a short period of time utilizing. So our hypothesis is that this can be done sustainable to many plants that come from the bush. It can be done sustainable and with mitigation plans to ensure that the material is not harvested in a destructive manner. Okay? It could address potential environmental effects. It helps provide cultural sustainability and cultural pride to people that understood the plants were valuable, but they didn't understand anybody else was interested in it. And it provides a compelling argument for public policy to protect these environments. This is I was very honored to be a co-author of the original WHO guidelines. It's now used in 42 countries on good agriculture and collecting practices of medicinal plants around the world. But now using this base, now we ask ourselves, how do we do this to improve health and nutrition? Again, using these 4 A's. And in this case, we surveyed people in different countries again for the plants that they wanted to eat. We found out how often they were able to eat it. And we found that the people in these countries, this was part of a flagship project from USAID to link horticulture and health and nutrition with African indigenous plants, okay? We realized that they couldn't afford it, they didn't have it available, and they were totally unaware of their nutritional benefit. So what do? We introduced them to what the nutritional benefits were. We introduced them to the different opportunities of different varieties. And we began to do what we call co-breeding. You do that for those that are working in plants. You grow a variety, a small subset of genetic diversity within a field, and you see which ones do well. We might be interested in the science, but the growers want to know which ones yield good or don't have insects and diseases. If you're a chemist, you might not care about the insects and diseases, right? You come in and you take your samples, and a year later you find that it has some exciting new compound in it. But that farmer there, they care about whether it has insects and disease. They care whether it grows. They care when it floods, the plant's still there at the end of the season. So we have to combine the field performance with the internal chemistry of it. As a plant breeder, we do this. This is our trade. Okay, so, and then we develop the best management practices in order to produce it in the cultural acceptable ways, which we do here. Okay? And then what we do is we go in and we look to see these plants. We know that soil, there's epigenetic and genetic differences. So it's not, we don't have the kind of resources to look at GWAS studies, genetic associated studies with a lot of it, but we do have enough ability to be able to look at the genetic diversity across different environments and ask ourselves, do the plants still accumulate the still high level of the nutrient-dense crops we want? And how do we define that? We use a European Codex Elementaris, okay, which is the high source threshold. This is what the handbook is that many countries use to be able to accept whether or not the national research institutes can work on an unusual plant, right? So we want to be able to work on all those underrepresented plants and get them listed in a Codex Elementaris, because then you as researchers would have greater flexibility and greater guidance and support to work on those lesser known plants here. And all they have to do is a high source of any one individual material. The areas highlighted in yellow indicate those factors that are of concern to women and to children, which is, of course, our major focus. Iron and availability of iron and the other compounds are listed here. So by understanding these and looking at this genetic diversity and using 4 examples that you see here of materials that consumers want. This is in the top 5 plants that they want to eat. And we're talking about millions of people, by the way. So when you think about the unmet market demand, rather than just collecting them from the wild, but introducing them into cultivation, you're talking significant dollars, okay? Is spider plant, nightshade, amaranth, Ethiopian mustard, okay? And when we look at that, we could grow them in different areas, which we have. And then we could look at the different accessions. We could categorize them as to where they're from. We could look at the chemistry. In this case, we're looking at the chemistry. The other graph with a similar picture was on the field performance of the growers. Then we could ask, in different areas, are they high source of iron or calcium, magnesium across different soil types? And so this is where we come in and do selection and breeding, where we have a type of amaranth that no matter what soil you grow it in, it'll still accumulate high amounts of calcium, magnesium, and iron. Others won't. Other varieties won't. And then we go back into the community, we say, okay, we have these plants, they're nutritious, they do good field performance. Would you like them? Because they're the ones who help identify them. And they tell us that they want to know the nutrition, they want to know that they have good field performance, but they also have to want to make sure that they taste good, okay? The way that they made and prepared food 20 years ago is not the way their lifestyles allow them to prepare food today. So we have cooking contests, okay? I know this is different. We're like hardcore. Now that might be a more interesting talk. I don't know, but I'll talk loud if you want, okay? I feel like I'm in one of those old-fashioned party systems, you know, where you're sharing a phone number and you're not sure who's talking to who. Okay, but be it as it may, we try to get together the national chefs that are really well known, and we get them to come out to the communities, and we get radio hosts. That is, we use social media in whatever cultural ways work, okay? It could be outside a church, outside a mosque, outside a temple, okay? And when people come out, then we have the food gathering and a food contest. We share the recipes, and we get them to embrace it, because those women in general, at least in sub-Saharan Africa, are going to be the ones making the final decision of what gets purchased to eat. In sub-Saharan Africa, the men might control the money, okay, and we won't talk about how they spend the money, but the women, if we want something to happen, it really goes to the women and the Women's Association in terms of spending correctly, making sure food get on the table. And we have a Let's Eat Leafy Greens contest. And this has worked good, because when we combine that to the cultural and the nutritional value and the right material with recipes, we get better behavioral change acceptance. Do you know what I mean? You don't just buy it once, but you buy it over a period of time. And we put this in Swahili, we put this in local languages of the plants that they're interested in. And you can see that upper plate, okay? The upper plate, I'm convinced that if you do just a color photograph, if you have more than one color, you're eating a little bit of diversity. In Africa, there's almost no diversity. I'm exaggerating to make a point and to make sure you're all awake, okay? But up here, we could see that the leafy greens and the other types of indigenous foods provide a natural bio-fortified food, even if you put in an ugala, even if you put in a cassava, okay? And we have so many different people in mining and so many different people in other types of industrial areas that have so little food that they eat instant wheat or instant cassava or instant single grain, maize. And so if we could just put in, whether it be a moringa or a nutrient-dense leafy green, it goes a long way, and this is what we're doing right now. Now, how do we do it when we get back to lab to be a little bit more sophisticated, because it is a biotech, okay, and we have to make sure you know that I'm from a university. Then we bring it into our core facility, and we have a whole range of different, more sophisticated analytical equipment that we could do the chemical identification of the material, or we could do the chemical identification of when it goes through an animal or a person, using the different types of analytical instrumentation here for bioanalysis or for metabolomics or proteomics. And so we have this. Dr. Rask and I, we work together on a lot of this analytical instrumentation. And so when we have materials that come in from another country, we have the ability, as you do here in Indonesia, of course you have tremendous, but in Africa they don't always have that level of sophistication which we need to confirm and validate. And then we have, of course, the GC mass specs and robotic GC mass specs, where we do headspace analysis in order to be able to identify what it is. And then we come back and confirm it. Is what is these materials like relative to the minerals or the phytochemicals that we're interested in, or the vitamins. We have systems, we measure vitamin A. Of course, we don't measure vitamin A. We measure the provitamin A through beta carotene. Okay? We measure the 4 different tocopherols of vitamin E, as well as vitamin C. We have to use it, of course, fresh. And this is just doing amaranth. And we could see that each of these plants we work with, whether it's amaranth, the spider plant, okay, or nightshade, okay, have tremendous nutritional benefits. That is, we have the backup chemistry, which is what we want. And of course, Indonesia, you have a large history of work on moringa. We've been obsessed with moringa from different aspects, but we are as well. Okay, it's not from sub-Saharan Africa. There are naturalized species in Africa, but we've worked with moringa in 4 different countries, and we could track it 4 times a year over 3 or 4 years, and we find that relative to iron, magnesium, calcium, and zinc, those same minerals, okay, that are deficient in the population are very rich in the leaves of moringa, okay? And so for us, this is what we're after. And then we have to ask, when we work with strange plants, is there something in it that we don't know? You know, you often talk about the good stuff, but we don't talk about what we call the anti-nutrition of stuff. So with solanum and edible nightshades, we did extensive studies because we were interested in whether it was going to have issues of glycoalkaloids in the leaves. We can't recommend plants that, so this is rich in some of the quercetin camphor. These are ubiquitous in most green leaf plants anyway, rich in phenolic acids, rich in saponins, but it has glycoalkaloids in it. So if we look at the specific...
Excuse me, Professor. Excuse me, I'm really sorry. I'm really sorry to interrupt your presentation, but we only have 5 minutes left.
Perfect. Okay, I'm from the Bronx, so I could cover another 200 slides in 5 minutes. Okay. So if we do a heat map, we could identify what the individual compounds are in these different accessions, okay? To be able to identify, is there anything we have to worry about relative to the glycoalkaloids? And the big message is, there isn't. We find some really great amounts of saponins, but the glycoalkaloids are absent in the leaves. Now in Africa, they eat the leaves. In South America, they eat the berries. The berries are very rich. There's ways to process it to get it low, but here, as an edible leafy green, it's fantastic. So we use this process. Okay, I'm just gonna go skip a couple because I'm not that fast. Okay. So my point is this approach with non-timber forest products are drivers of change for income generation, can be used in a very good way for environmental, cultural, and sustainability. You could hear it in the voice of the women of Sangwe. There's documentations that were put on for this, in this where they talk about the importance of preservation of the environment and of using plants for their health and nutrition. We could introduce in the international realm the science that we as scientists do to form the basis. But what I do want to do in my last 4 minutes, or 3.5 minutes now, is to show what we have found is if we do the science, but if we don't tell anybody about the science, we're not doing our justice. So in an international program on crossroads of biodiversity, we now embed in our science the science research telling stories. And we did this with a researcher at Rutgers who's trained as an anthropologist. And we do this together where we embed people that do films in our work along the way, because then we use this for public policy formation, public awareness. And this person has done it before. Now imagine if your work ends up in Smithsonian, ends up in National Geographic, just like where Professor Raskin's pictures will be, right? When you think about it, it reaches a broad-based audience. And our task is not only do good science, but make sure the public understands it and the public trusts us. And at least in America, the public is not trusting science so well because they don't think we're relatable. Doing these films allow us to make these stories. And this is just one picture of doing genetic diversity. We have 550 types of basil, including basils from Asia, by the way, but it's not part of the African bush. But it was devastated by a disease. And we found that there was a plant from Zanzibar in Africa, so I was justified bringing it in here, that we used to ret introgress in, and from a plant that suffered in the upper picture, you see the diseased plants. We now have plants that are resistant to this disease because we're able to take advantage of understanding the genome, understanding genetic diversity from an unusual plant that was collected in Zanzibar that was then used in breeding in order to get the types of products. And that was filmed in Fields of Devotion. So now, relative to climate smart and the importance of biodiversity, it allows us to move forward with a good story using film. So I think this is something we should all consider doing in terms of the work you're doing here. Sustainable collection, I won't go through the takeaways because you've heard me say it already, but I will say that I hope the journey that I've taken you through to Africa has been an interesting one. The success has been due to many different people, of which I'm just the one person who tells a story along the way. Okay, so as we bring it together, I just want to thank you for inviting me over here, and I'm looking forward to meeting you and listening to other talks. Thank you so much.
Thank you very much, Professor James Simon, for your presentation. I believe your presentation and topic and research will be very fruitful for us for our academic journey in the future. Unfortunately, because the time is limited, to participants that have questions, probably we can recommend you that you approach personally Prof. James Simon if you have further questions about his topic. Thank you very much, Professor. Please stay with us until the end of the keynote session because there will be a token of appreciation given to you by the committee. Well, ladies and gentlemen, move on to the next keynote session. For our second keynote speaker, we will hear a research entitled The Role of Biotechnology to Utilize Biodiversity to Support Food Security and Health, Current Trends and Challenge from the Indonesian Biotechnology Consortium. A little bit information, a brief information about our speaker today. Our speaker finished his PhD degree from Georg August University, Göttingen, Germany. He is a chairman of Indonesia Biosafety Committee of Genetically Engineered Products from 2018 until now. He has received National Development Award, Karya Pembangunan from President of Republic of Indonesia in 2003 and 2007. He had published numerous international publications and received patents of his inventions. Well, ladies and gentlemen, please welcome Professor Bambang Prasetya.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Distinguished guests, Bu Eni as the chair of the pengarah ya, the organizing committee, and Pak Mastur and colleagues from several institutions, and especially for distinguished guests from abroad that maybe for first time to come to Bali. Welcome to Bali and enjoy Bali. And I heard some of you will travel to Papua. It's very nice place. Ladies and gentlemen, I would like to share with you the role of the biotechnology to utilize biodiversity to support food security and health. I talk about the opportunity, current trend, and challenge. I come from the Research Center for Testing Technology and Standard, and I also responsible for Indonesian Biosafety Committee for genetic engineered product from the KKH. Allow me first, I would like to share with you about the situation, the global challenge and risk of mitigation. And I'm interested the report from the World Economic Forum, the top global risk for next 10 years. According to this report, it is noticed that the 10 global risks in the next 10 years, among other related with climate change, extreme weather and biodiversity loss, and also environmentally damage, infectious disease, natural resource crisis, and all crisis related poverty. And I take one example. What is actually the influence of the climate change? I take, for example, the food system. It is very challenging issue that when we're talking about the climate change, the impact on agriculture can increase the disease severity and also lower yield and also lower the nutritive quality. Of course, this is actually the PPT is the old one. Sorry, but no problem. According to the report from the global climate change, it is reported that the increasing of the temperature by year to year with the scenario, for example, here reduce the plan how to manage the temperature increase to reduce in the certain policy in Indonesia and also the global. We also have the challenge how to capture carbon dioxide in Indonesia and the plan of the climate change in Indonesia. As impact of the climate, we also facing the world hunger. As you saw in the picture, the color, the brown color is very critical point, and then also in the orange, it is around 50% of the hunger in the world situation is also very challenging to facing how to fit in how to provide the food, sufficient food with a good quality. For example, when we talk about the vitamin A deficiency, there is also we still facing the problem with also the malnutrition and also stunting, also the vitamin A deficiency. Growing population and limited resource and pressure on ecosystem is a very clear challenging issue in the near future because the population is increased higher and higher, and at the same time the availability of land for the agriculture is decreased because the environmental damage and also because of the climate change, also the effect, and also the man-made effect of the environmental and agriculture. Next. We also in this related the topic, the biodiversity loss also is the very challenging issue because there are many drive to make the biodiversity in several parts of the world lost. For example, here the number of the decreasing of the quality of the biodiversity is shown in this slide. And now I would like to talk about the biodiversity and the role of the biotechnology. First of all, the biodiversity, why is it so important? Because supply of food, energy for the pollination and agriculture, and also the production of food, several benefit for the people. It is for the cultural, for the recreation, for the medical product. As Mr. James from the Africa saw us about the report from the Africa, is very nice report because many biodiversity can use as a medical plant. When we're talking about the biodiversity loss, there are some research, maybe Bu Indra will be talk a little bit detail about the situation in Indonesia, but I would also just share that Indonesia is belong to the mega diversity country, maybe number 2 after Brazil, but it is not include the sea biodiversity. I think I'm sure that when we calculate the sea biodiversity, we will have maybe more than terrestrial biodiversity. And when we be talking about the potential of the sea, we are sure that there are medical potential for the benefit for the people in Indonesia. For example, the research can focus on the utilization of the resource of the sea, marine, I mean. And just to show you about the distribution of the biodiversity in the world, and several color, Indonesia belong to the country with very high density in the biodiversity, like in this picture show that in the color, red color and also the dark color is the density of the biodiversity as well. And in the table, we have some species, how is the potential of biodiversity in Indonesia. And about the valuation of Indonesian biodiversity, we can calculate the direct value for the raw material, as I mentioned before, the agriculture, for food, biological control, pest and disease, the fiber material for housing, for the paper and for textile, for industrial chemical, for housing, and in the same time, we have also the direct value, the ecosystem, climate improvement, and also carbon economy, environmental services, clean and the table we show you about the value of the Indonesian biodiversity. Now we're talking about the biotechnology for sustainable biodiversity management. In fact, that this is example how the biotechnology can support the better management in the biodiversity management. For example, through tissue culture, microorganism cloning, and artificial insemination, also bioremediation, genome editing can reduce the impact of the biodiversity loss. It is at the level of the genetic, also in the level of species, and also in the ecosystem as well. Just give the example, the role the new plant breeding technology for food security and the sustainable agriculture development, as saw in this slide, that is clear that the productivity of the several countries, Asia, Africa, and also another world, that increasing the productivity through by using the biotechnology. In the other side, the decreasing of the malnutrition is very clear here, that the role of the new breeding and biotechnology contribute to this to address this problem. From another report, it is quite good because it is quantitative. From the ISA, the contribution on biotech crop to food security, sustainability, and climate change and mitigation, that they saw the benefit of biotechnology over 20 years. It is in terms of the increased crop productivity is very clear. Conserve the biodiversity, also the reduce the CO2 emission, and also provide a better environmental, and also help to alleviate the poverty in the world, also hunger. The number quantitative make us convinced that through the biotechnology we can use for several problem which rise in the lately years. Biotechnology platform open the opportunity facing the global challenges issues. When we talking about biotechnology, there are addresses to the animal biotech, plant biotechnology, and also environmental biotechnology, health and medicine, microbe. There are several items we can as tools how to use this tool to utilize of the biodiversity. There are many platforms that is already done and provide the real fact that it is really utilize of the biotechnology can contribute for the economic and also environmental and also the food security and health. As a new tools, and the three pillars saw in this slide, we can also provide several platforms and use for the utilization of the biodiversity, from genome sequencing, genome silencing, and also genome editing, and protein engineering, and also we can engineer the metabolite. We can develop the cell tissue specific expression and phenotyping, and imaging, and also monitoring field and experiment. We can produce a certain product which subjected to the environmental and also the benefit of the economic or social aspect. Just to mention about the lately, maybe in the last decade about the genome editing, there are the data, how is actually the country which use of a certain trait. For example, this tabel, this is the commodity. For example, especially for the rice, it's mostly for the rice, and also the tomato, soybean, and also for the corn or maize. And the picture on the left is for the trait that very useful by using of the GA in the research in the country. This is the method used by research in gene editing and also the country conducted gene editing research. For example, here in China, dealing with GS very lot research here, and in the other hand, there are methods. We know that the CRISPR is very dominant in the use of the genome editing. It is the picture. I show you that the distribution, the regulation in the Asia and Australia country, and my PPT actually the new PPT I talking about the world, but this okay. This the green color is already adopted that the genome editing treat not like the GMO regulated, not like a GMO, and the green yellow color is discussing. Just for Indonesian position, genome editing, we already discussed that we accepted that the SDN1 and SDN2, we make exemption to the GMO regulation. This is a discussion about the SDN2, SDN2, because some country in Asia and Australia very often to discuss about the SDN2 definition. I talking about the business process, how to apply the technology resulted from genome editing. We have a regulation we call a biosafety regulation in Indonesia. The applicant should propose to the Minister of Agriculture and Ministry of the Forestry and Environmentally, and also the National Agency for Food Drug Control. And then this proposal will send to the biosafety committee, and then from the biosafety committee send to the technical committee, and then the assessment there. And then after that, we can finalize that the report in the meeting of the KKH. We can release the recommendation of whether certain product by shipped or not. And then we send back to the ministry, and then from the ministry can release the recommendation or certification to release this kind of product. Now, talking about the mechanism of the assessment of genome editing, as I mentioned before, that we at least SDN1 and SDN2 we not regulated as GMO. So that maybe from Asian country or from many country like Indonesia talking about this regulation. I'm talking now about the biotechnology for food. For example, here this is very clear that the role of the new plant breeding technology for food security and sustainable agriculture development. There is a data that the GM crop not only increase the yield but also the reduce the pesticide quality, and also at the end can increase the farmer income. And next slide also, this is also an example about BT cotton. The benefit, for example, from the economic aspect, from the environmental aspect, also from the value of the nutritive and health effect of the planting of the BT cotton. And here reporting by ISO also here that the distribution of how many GM crop plant from the position right now, it is around 2.7 billion already GM crop plant in around the world. Of course, the USA, Brasilia, and Argentina is the leading country with which planting a lot of GM product. And in year 2019, around 17 million people already benefit by planting of 17 million of small holder farmer beneficial from the GM crop. This is the position of the country which planting the GM crop. 5 countries leading is America and Brazil, Argentina, and also in Canada and India, as I mentioned before. And then we have now a report about the commercialized GM crop. For example, here what kind of trait used and what kind of gene used in this GM crop. And then when we talking about the GM trait position, there are around 46 trait already researched, and for the commercialized GM trait, around 8 trait already commercialized in the product in the market. And then we're talking about the genetic transformation strategies and genes used for the improvement for biotic and abiotic stress. For example, for the crop. For example, how to manage the heavy metal tolerance, salinity tolerance, resistant against oryzia, excavate and insect resistant, soybean virus resistant, and also disease resistant, higher rate on inhibition and of root knot nematode, and so on. This trait is very important to develop a new variety which very tolerant with abiotic stress and also the biotic stress. Another example, for example, here, how is genome editing implemented in the farm animal to increase the productivity? For example, the enhance the muscle of growth for a cow, for sheep, and for goat, and also some certain trait also developed in certain purpose. And now I would like to just a review about the representative biotechnology for health and environmental recovery. For example, here, the representative transgenic plant-based vaccine, for example. The subject of vaccine developed by plant, for example, for the hepatitis B, hepatitis E, influenza, infant polio, cholera, and also rabies, swine transmissible, and also gastroenteritis virus. This very vaccine already developed in a certain research and already implemented in some country by using of the plant used for the vaccine. Why it is a vaccine is this is my old paper not translate in English. Why we do the vaccine in the plant? Because there are several benefits. For example, as alternative to develop vaccine and also as alternative to added value of the agricultural product. For example, here we use potato and also banana, rice, and also maize, and tobacco, also the other fruits. And when we're talking about the biofortification, which is very important to facing of the stunting matter in like in Indonesia, there are also crop which can engineered by certain, for example, here is trait developed for anti-tumor, antimicrobial activity, for the antibacterial, antifungal, and so on. And about how to improve the environmental problem, for example, through the P2 remediation, we can also develop a breeding by mercury tolerance, increase the accumulation of cadmium, copper, and mangan, increase the accumulation of mangan and also cadmium, increase the CO tolerance, and also here enhance tolerance to zinc, enhance tolerance to cadmium, increase the tolerance, and also tolerance to the zinc, and so on. The challenging issue related with regulation and harmonization toward, I would like to show that for develop trust, there are three pillars important to make regulation smooth, acceptable in the stakeholder. For example, here the first one is our bioethics, to ensure smooth adoption based on ethical, moral consideration, and also biosafety risk assessment to ensure the scientific based risk analysis and certification, and also we also need the conformity assessment. We do, for example, standard and accreditation, calibration to ensure traceability, so that between laboratorium, between risk assessment, they have new data with a single platform or acceptable platform. I also including the bioethic to make a biotech product more acceptable to the people. There are also some religious aspects, for example, about the halal standard. Indonesian, for example, like in the Islamic country, also have a standard about the halal. This is also including to the GM product. We already accepted that GM product is halal food when the component also come from the halal material. For example, this is the fatwa MUI number 35 2013 about the product. And now I would like to talk about the regulation on biosafety and genetically engineered product. We based on the law number 21 2004. It is the ratification of biosafety Cartagena protocol, and we have also the government regulation number 21 2005 about the biosafety, and then we have also the president regulation, president decree, and all decree of Ministry of Agriculture, Ministry of Regulation, and also we have also regulation from the National Agency for Drug and Food Control. And the committee, we divide biosafety committee and technical team committee. Biosafety committee consists chair for food safety, chair for feed safety, and chair for the environmental safety. And for the biosafety technical team, we have division of the food safety also, division feed safety, and also division of the environmental, including plant, animal, fish, and also microorganisms. This is the business process, the procedure how to apply the GM or genome editing product in obtain the food and feed and environmental biosafety recommendation from the KKH. This is example the plenary meeting of biosafety during the pandemic. We every month at least one or two meeting to decide recommendation of the biosafety of the proposed proposal from the proponent. This is some example the existing food safety and feed safety and also environmental product. For the corn, it is related with insect resistant, corn herbicide tolerant, and drought tolerant for corn, and also the soybean for insect tolerant, and so on. We have also a product original in Indonesia is about the sugarcane drought resistant. There are some product that we already received the recommendation. And this is also the product researched by Indonesian researcher. For example, here I have mentioned before that the sugarcane and also the potato and also the corn, we already released in the plantation or in the agriculture field. Now I'm talking, the last one is about the harmonization of the procedure and regulation. It is very important to make method and also regulation based on the general standard which they can use compatible between the method X country, X laboratorium, inter-laboratorium. For example, harmonization through ISO Technical Committee 331 about the biodiversity. The picture shows us that the map of the member of the ISO which dealing with biodiversity harmonization and standard development. For example, here P member including Indonesia is a participating member. They had also voting to decide whether standard which can apply in each country. And the observing member is in the green yellow color. It is without voting, but they can also contribute the matter about the development of standard. For example, this year they decide the roadmap how to make a harmonized standard between country, especially the member of the ISOTC. For example, we discuss about the terminology about the biodiversity, about the biotechnology, and also we talking about how to meet the method of conservation, ecology, restoration, and integration biodiversity issue in organization, and integration of biodiversity issue in certain territorial. There are many information about the each country have a certain regulation, and meet together in this forum. We can harmonize the methodology or the regulation related with biotechnology issues. And sometimes we still have happen the simplistic dichotomies. For example, GMO versus organic in Indonesia still happen. Synthetic versus natural, alternative versus the real, dream versus now, plant versus animal, and good versus evil, hoax, and versus traceability. There are many problems facing to how to engage the people, accept the biotechnology for the many innovation for the production and new product, especially for the food security and for the health. Thank you very much for your kind attention. I'm sorry, it is my old paper, but the new one I already have sent to the organizing committee. Maybe this one is much better because update many information there. Thank you very much and enjoy in this seminar. We wish you a productive discussion. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Bambang Prasetya, for your amazing presentation. Ladies and gentlemen, similar to Professor Bambang Prasetya, please kindly return to your seat. Thank you very much. To ladies and gentlemen who have questions about Professor Bambang Prasetya's topic or research, we recommend you to approach professionally Professor Bambang if you have any further questions. To Professor Bambang, please stay with us until the end of the keynote session because we will have a hand-in token of appreciation from the committee. And ladies and gentlemen, we arrive at our final keynote session. For the third and final keynote speaker, we will listen to a presentation with a topic, Genetic Resource Diversity, Biotechnology Towards Conservation and Sustainable Utilization of Biological Resources, presented by the Ministry of Environment and Forestry of Indonesia representative. Brief information about our speaker today. She is a veterinarian that has more than 30 years of experience in the area of forestry and wildlife conservation, including wildlife crime. She is the Director of Biodiversity Conservation, Directorate General of Natural Resources and Ecosystem of Ministry of Environment and Forestry of Indonesia. She has extensive professional experience. One of them is the National Focal Points for Convention on International Trade in Endangered Species of Wild Fauna and Flora. She is also active in conducting research proven by many international publications. Therefore, ladies and gentlemen, without further ado, let's welcome the RH Indra Eksploitasia Semiawan MSI. To Dr. Indra, time is yours.
Very much. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning, Bapak Ibu, ladies and gentlemen. First of all, I would like to thank you for the organizer for the inviting us from the Ministry of Environment and Forestry to attend this important and special event on the International Conference for the Biotechnology. Thank you especially for Ibu Eni and Pak Bambang for this nice place, and hope that we also have a nice enjoy in Bali. So without no further speak in the introduction, I would like here to present the theme of my presentation is about the biotechnology towards conservation and sustainable utilization of the biological resources. There are two keywords of this presentation. First is the biotechnology, and the second is biodiversity. So if we talk about the biodiversity, so we all aware that the biodiversity is crucial for our life and economic development as the source for food, housing, clothing, medicine, industrial raw material that have already mentioned by Professor James, and many other things needed for human well-being. So we all aware that there is an urgent need to conserve biodiversity, and it is found biodiversity degradation caused by habitat fragmentation, invasive species, overexploitation, pollution, and climate change. So we are here to discuss how biodiversity loss has become our concern, particularly international fora such as Convention on Biological Diversity, focusing on why we also should protect, conserve the biodiversity through the biotechnology. So we were urged to enhance our work to halt the biodiversity loss by some intervention on biotechnology, and therefore in Indonesia, biotechnology can have two functions here. Biotechnology to support the conservation, and the second one is the biotechnology to explore the potential value of the genetic resource, and for example is for the bioprospecting. So the outline of this our presentation is about we have biodiversity conservation. We will talk about the policy and how the conservation in regulation in Indonesia, and also we talk about how genetic resources as a national asset and how bioprospecting is a potential economic for the economic development. So the next presentation, I would like to share about our policy in the biodiversity conservation. There are three levels of the biodiversity. We are aware the level of the biodiversity is the ecosystem, the species, and then any genetic resources. So the conservation principle in Indonesia different in the Convention on Biological Diversity. The principle of the conservation in biodiversity in Indonesia, there are protection, preservation, and sustainable utilization. And for the Convention on Biological Diversity, we have three principles: conservation, and then sustainable utilization, and access and benefit sharing. There are three principles in Convention on Biological Diversity, different with Indonesia, because Indonesia focusing on sustainable utilization on the three levels: ecosystem, species, and genetic resource. So in the Convention on Biological Diversity, focusing also in the conservation, sustainable utilization, but in the genetic resources, they are focusing on the access to the benefit sharing, access to the genetic resource, and also access to the technology. So developed country can access to genetic resource from the developing countries, but also the developing countries could access the technology from the developed countries. To conserve means to protect, to study, and to utilize. That's the also the principle in our act and in regulation in Indonesia. So in the next slide, I would like to emphasize of the ecosystem diversity. So many literature and studies shows that our current ecosystem, we have natural ecosystem and also artificial ecosystem. So we know that the artificial ecosystem, such as rice field, and then mixed estate like agroforestry, agro-silviculture, and garden, and etc. And while natural ecosystem are marine ecosystem, freshwater, semi-terrestrial ecosystem, and riparian terrestrial, such as lowland forests and mountain forests. So the ecosystem diversity in Indonesia is divided into 19 natural ecosystem, consisting 74 of the vegetarian types, which spread across nearly all the bioregion. We have 7 bioregion in Indonesia: Sumatra, Kalimantan, Jawa, Bali, and then Sulawesi, Maluku, and Papua. So the such variation shows that ecosystem is also rich in flora and fauna. And we are talk about the species diversity. The species diversity in Indonesia that already mentioned also from the presentation from Professor Bambang. Indonesia is the large mega biodiversity country in Indonesia. Around have 30,000 to 40,000 terrestrial species, which are more than 1,700 mammal species, around 1,700 birds of species, and more than 200 fungi species. So if we see the map of the Indonesia, the spread of the species diversity is all over into the 7th region in Indonesia. So after this level of the species diversity, my presentation focusing on the genetic resource. In the genetic resource, talking about the genetic diversity in Indonesia, we are aware there are so many diversity of animals and plants in our natural forest, and we can see in the picture we have two species of the rhino, Sumatran rhino and Javan rhino, and then there are different genetic, there are different species. So we can see also that we have 3 species of the orang-utan Sumatra, orang-utan Tapanuli, and also orang-utan Borneo, orang-utan Kalimantan. And we can also see that the orang-utan Borneo or orang-utan Kalimantan have 3 subspecies: Pongo pygmeus pygmeus, Pongo pygmeus wumbi, and Pongo pygmeus mario. So if we see that the genetic diversity in our species is very various and diverse, and also for the genetic we can see that our most species in Indonesia is very specific appearance, behavior, and physical condition adjusted to their natural habitat. Next slide, I would like to mention about the policy and regulation in Indonesia. It's our task in our ministry to give the policy and regulation on how to make a conservation and how to utilization a sustainable utilization on the biodiversity in Indonesia. Indonesian policies on biodiversity conservation is referring to both global and regional policies, as we already ratified some convention, such as Convention Biological Diversity, Cartagena, and Nagoya Protocol. These policies referred specially in accordance with our country's situation and need, that for examples regarding for the issues of biodiversity, biosafety that I've already mentioned from Professor Bambang. We especially put our attention on the precautionary principle approach to use the biotechnology, so as implied in the Cartagena Protocol. So also we highlight that the fair and balanced access and benefit sharing in genetic resource utilization, which in line with the Nagoya Protocol. And last but not least, we refer to the CITES, Convention of International Endangered Species, for our work related to wildlife trade. So this is in line with the policies on the sustainable utilization. So more importantly, we understand that all these policies conserve the biodiversity, sustain the biodiversity use, and achieve the fair and equitable sharing of the benefit which areas from the utilization of the genetic diversity. In national regulation in Indonesia, several laws and regulation were issued to support the conservation of biodiversity, such as the Law Number 5, 1990, on the conservation biodiversity of resources, ecosystem, and many others. In the ministerial decree on number 2, 1918, so this is basically, especially for the mandatory for the government to conserve the biodiversity, because the biodiversity is considered as a nation wealth. So therefore, it is management become the responsibility of the government for the benefit of the entire community, with encouraging community and non-government organizations to support and participate in the conservation activities. However, to ensure the genetic resources are used sustainable, a ministerial decree on access genetic resource on wild species and benefit sharing from their utilization was issued on 2018. Especially, this regulation provides a detailed arrangement for the access to genetic and or traditional knowledge, material transfer, institutional arrangement and mechanism of the supervision, monitoring, and sanction provision. So the genetic resource here is interpreted as the genetic material and or genetic information of the plant, animals, microorganisms, and other origins, including the derivative, which contain functional unit of the heredity, which has real and or potential value. Ladies and gentlemen, Bapak Ibu, on the genetic resource and bioprospecting, genetic resource utilization can be divided into non-commercial and commercial issues. So the non-commercial use of the genetic resource covers the taxonomic research, species identification, and distribution, study of the wildlife conservation, and study of the crime evidence, or we call forensic. So the commercial use of the genetic resource, as we call bioprospecting, is for vaccine that already also mentioned by Professor Bambang, and in isolate of the growth promoting bacteria, anti-aging, skincare, already also mentioned detailed by Professor James, that there's two things that become one of the fast-grown business. Then anti-cancer drugs, food source by technology development and other commercial purpose is very important, that the intervention of the biotechnology to biodiversity is right now is needed and is very necessary for us, and how the technology, biotechnology could develop the economic growth of the Indonesian. And also, beside the economic growth, we also should also concern of the ecological development. So the bioprospecting is one of our focus discussion today, and as I mentioned earlier, that has supporting the business growth and in almost all countries in the world, because it's a potential for almost aspect of human being. However, we are all aware that the genetic resource utilization for the bioprospecting needs in deep research to reveal the potency. So nowadays we observe that many corporates and private sector has also engaged in the research and development of potential for the bioprospecting. For the biotechnology for the conservation, We have already conducted the conservation program in Indonesia to the how we in biotechnology in Indonesia could support the conservation in the condition in Indonesia that we have two species, endangered species here in rhino and Javan rhino, Sumatran rhino. Both are categorized as a critically endangered, and its population is natural habitat is very low. So there are challenges for increasing the population of the rhino we have conducted by the assisted reproduction technology, or we call bayi tabung in Indonesia, assisted reproduction and fertilization. So the ART is applied in the Sumatran Rhino Sanctuary. In Sumatran Rhino Sanctuary is in Lampung, in the Waikambas National Park. So the technology was developed to help the Sumatran rhino breeds naturally with the support, the technology, and with the collaboration, of course, with the expert. This the ART assisted reproduction technology approach target is first to generate of the new baby rhino and embryo. So the choice of the assisted reproduction technology for the Sumatran rhino are to manage breeding based on the genetic diversity and to promote high heterozygosity, and to by the artificial insemination, intracystoplasmic sperm injection, and somatic cell nuclear transfer, and also by the our biobanking is also developed in Indonesia. So this is the example how biotechnology could support the conservation for the fauna in Indonesia, for the wildlife in Indonesia. So as the talk about the biotechnology for biodiversity as bioprocessing, we have already know that biodiversity in Indonesia as a national asset. So considering the biodiversity as a national asset is a basic capital for the supporting for the sustainable development. So therefore, Indonesia acknowledges that our biodiversity, including the material genetic, is an asset, and at the same time is the property right of Indonesia, which is in line with the Nagoya Protocol. So recognize the value of the genetic utilization for the commercial issues atau bioprospecting. It is a source of the sustainable funding for the conservation. So this is our paradigm right now. The paradigm we have already known that conservation is the cost center, but the new paradigm right now is how we can use that the biodiversity use for the economic growth. So we can change the paradigm to the cost center to the profit center. So the conservation as the investment, as how to biodiversity as the funding source for the economic growth. However, we also seriously taking into consideration that any activity to take sample of genetic material from animal, we should consider the principle of animal welfare and also animal health code. So also for the outsider access and utilize genetic resource should be followed, but not only benefit sharing, but also to the access to technology, or we call technology transfer. Indonesia also promotes to produce final product from genetic resource and minimize the producing intermediate products. The next slide, the potential of biodiversity. So we can see that the abundance of the genetic resource for the bioprocessing. So the versus of the promising world market. So Indonesia has 11% of world flowering plant species, 12% of world mammal species, all are in Indonesia. So while at the global, there's around 40 to 50% market for medicine coming from the natural resources. So one of the top 25 sales of the pharmaceutical products came from the natural ingredients, and there is still need for drugs and vaccine. So the new drugs also, we have already developed many herbs that also could as a medicine for the COVID-19. So the stages of the development of wild species bioprospecting. So we can see in the slide. This is the stages of the bioprospecting from the wild species, from the survey, exploration, to research, and the test, and from the raw material preparation until production, and the last thing is the marketing and promotion. So we divided these stages into two, upstream and downstream industries. So these stages is also already developed in Indonesia. And we can see the next slide that the bioprospecting of the evolution, as I mentioned, that the bioprospecting has evolved from the industrial era to the biodiversity era. So then the sustainable development, and in the future as the source of the sustainable financing. In the two next slide, I will share about the bioprospecting potential in Indonesia. While Professor James mentioned about the genetic diversity in Africa. So we have already also identified and discovered about many various of the genetic diversity that could as a potential as a for the bioprospecting, spread from the west to east, from the Sumatra island to Papua. Quite many species have the potential for food source, medicine, housing, and also as a pest control. So like in Sumatra, we found that the Taxa sumatranus as a anti-cancer, and also as we found this plant in national park in Bukit Barisan Kerinci Seblat National Park, and many other things that we found in national park also as a potential for the bioprospecting. Next slide. Also, we shared about many species and genetic resource from the plant and animal that already discovered too many function for the medicine and also for the food health. So this is the last slide for my presentation. As my conclusion here, we are aware that the biodiversity is an important asset for the sustain the human well-being, and the benefit of biotechnology is a tool to support conservation and sustainable use of biodiversity, and will continue grow in line with the human needs and future condition. So we have to balance the economic development with the ecological development. So bioprospecting can be used as a potential sustainable funding mechanism for conserving biodiversity and sustaining its component by three levels of biodiversity: ecosystem, species, and genetic diversity. So the last but not least, please, precautionary principle should be considered and implemented. Thank you very much. My quote today as next slide. Thank you. So the quote today for, so we have to live in harmony with nature, growth and prosperity. Biodiversity is an asset. Sustainability, the asset is balancing ecology and economy. Thank you. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Indra, for your excellent presentation. For Dr. Indra, please remain on your position because we will have a hand-in token of. Once again, we would like to invite Professor James Simon, Professor Bambang Prasetyo, and also Dr. Indra to come forward to receive token of appreciation provided by the committee. Once again, thank you very much to our remarkable keynote speakers. We believe their presentation will be very beneficial for all of us. And now we would like to invite Professor Ernawati from Universitas Nasional, Professor Annie from Indonesian National Research and Innovation Agency, and also the representative from IBC to come forward to hand the certificate of appreciation for our speakers. Yes, we have udang and also traditional fan. Udang is for our male speakers. It is a traditional Balinese headwear used by males in traditional ceremonies, just like what I'm wearing today. So it's not for traditional, but also a symbol of Balinese tradition for our male speakers. We also have like 3 different types of udang. So it's not the first one is not 100% finished. So you still have like fabric and you create it by yourself. And then the second one is almost finished, like this one, half finished. And also the last one is completely finished one. So Professor James Hyman already wear the udang. It looks very great on you. Thank you very much to Professor Bambang for your presentation. And last but not least, to Dr. Indra, we have a traditional fan. It symbolizes the Balinese culture, usually used by females in traditional Balinese dance. Thank you very much once again to all of our keynote speakers for amazing presentations. Now we will have a photo session with all the guests and also keynote speakers. The photographers will direct you. For the formal ones, 1, 2, 3. Okay, now say Bali in 3. 1, 2, 3, Bali. Okay. Thank you very much once again to our keynote speakers. Right now, we also would like to have a photo session together with all invited speakers. To all of our invited speakers, would like to please come forward. To our keynote speakers, please remain on your position. And now we will have our all invited speakers to come forward to have a photo session together. To all of our invited speakers, please come forward to join the photo session. All right. So for the formal ones, 1, 2, 3. Okay, now say Bali in 3, 1, 2, 3, Bali. And probably we can say biotechnology as well because it rhymes with I. 1, 2, 3, biotechnology. Okay. Thank you very much to our keynote speakers, invited speakers, and distinguished guests. Please kindly return to your seat. Photo session with all of the participants will be conducted later on during our lunch break outside at the yard. So please stay with us. And ladies and gentlemen, as previously we already heard remarkable presentations from our notable keynote speakers, now we will be getting more knowledge from our invited speakers, especially in the area of biodiversity and its relation to traditional knowledge and socio-cultural aspects from our invited speakers. The abstract and the program of this conference is available on the link tree provided by the committee. So if you want to see the abstract and the program, please visit the link tree provided. Ladies and gentlemen, without further ado, we will begin our plenary session 1. And this session will be led by our moderator. Our moderator today is a lecturer from the Faculty of Foreign Languages in Mahasaraswati University. Ladies and gentlemen, we would like to invite to join us to the stage Dr. Igung Agung Sri Rua Jayantini SSM Hum. Please give a round of applause to our moderator. To Dr. Sri, time is yours.
Thank you, MC. Well, good morning, everyone. Distinguished participants, honorable invited speakers, keynote speakers, ladies and gentlemen. It is a great pleasure for me to welcome you again to the International Conference and Workshop in conjunction with the 8th Indonesia Biotechnology Conference. I'm Sri Jayantini, who's going to chair the session, and this is the plenary session 1 for the conference. And here, we are going to listen and learn from our invited speakers for about 2 hours, with each 20 to 20 minutes presentations from the invited speakers, and then will be continued by question and answer session. And ladies and gentlemen, as you can see from our agenda, the topic for this session is biodiversity and traditional knowledge and socio-cultural aspect. I think this is very interesting issues for our discussion because the existence of biodiversity, of course, cannot be separated from the existence of human being and their social relationship with others. Well, ladies and gentlemen, let me firstly announce the arrangement for the invited speaker session or our plenary session. We are going to listen first, all the invited speakers share their insightful knowledge, and then after that, we are going to have a question and answer session at the end of the session. For those who are here joining the conference on site, please prepare your questions. I'm going to open the opportunity for you to ask questions directly, or if you join via Zoom, then you can write down your question on the chat box, or if it is possible, then you can ask directly the questions. Well, ladies and gentlemen, without further ado, let me firstly introduce you to our first invited speaker in this session. Well, allow me to welcome Professor, sorry if I mispronounce your name, Professor Vyacheslav Dusenkov, PhD. He is a professor of Houston Community College City University of New York. Would you like to join with me here? Okay. So ladies and gentlemen, this is Professor Dushenkov. Professor Dushenkov is a highly accomplished scientist and business executive with a wealth of experience in the fields of botanical therapeutics, environmental protections, phytoremediation, and STEM education. Professor Dushenkov now currently holds the position of the professor at the Department of Natural Sciences at CUNY Hostos Community College. Okay, and Professor Dushenkov diverse professional experience encompasses various areas such as educational course development, research and development program management, intellectual property protection, strategic planning, and contract or joint venture negotiations. Please give a big applause to Professor Dushenkov. And I would like also to invite our second invited speakers. There are two speakers. Okay, Professor Saidbek Satorov. So please join us here with Dr. Sulfa Maslona Zarova. Okay, Professor Satorov is the head of Department of Microbiology, Virology and Immunology, Medical Social Institute of Tajikistan. Is it your first time here, Professor Satorov, from Tajikistan? First time? Okay, so enjoy Bali. And his main fields of research are to study characteristic of pathogens microorganisms circulating in Tajikistan and their spectrum of antibiotic resistance. Professor Satorov pays special attention to the antiviral, antibacterial, and antifungal activity of endemic plants growing on the territory of our country. Our country is Tajikistan, right? Okay, yeah. Besides that, do you know his hobby? So what is your hobby, Professor? Yeah, play chess and travel to different countries and get to know Balinese culture today, yeah? Okay. Next, I would like also to introduce you to Dr. Sofia Maslonazarova. Okay, she is a lecturer from Fishena Tajik State Medical University, right? And Dr. Mavlonazarova research interest is devoted to the study of pharmacognostic properties of plants of the genus Ferula in particular, endemic forms growing in Tajikistan. And the third invited speaker in this session, please join me to welcome Professor Hiroshi Kamitakahara. Would you like to join us here, Professor Hiroshi? Professor Hiroshi, research interest is biomass refinery, forest products for pharmaceutical, wood chemistry, cellulose science, organic chemistry. And Professor Kamitakahara is working for Graduate School of Agriculture, Kyoto University, Japan. So, ladies and gentlemen, those are all our keynote speakers. Can you give a big applause before their presentation? Okay, for about 25 minutes each, we are going to listen for the presentations of our invited speakers. Now, without further ado, I would like to ask and invite Professor Dushenkov? Oh, okay. To have your presentation. Time is yours. Okay. Would you like to stand there? Yeah, okay.
morning. And first of all, of course, I could like to say my sincere thank you to the organizers and sponsors of this conference. It's such a pleasure to come for this very important discussion in the center of biodiversity. And I am not sure about statistics and this rival between Brasilia and Indonesia. For me, Indonesia is the hottest spot of biodiversity in the world. And I easily combine both terrestrial and marine biodiversity to do it very well. Thank you so much for organizing this conference and bringing us in this wonderful island of Bali. Also, I am in a very difficult position. After a presentation of Professor James and Bambang and Dr. Indra, I have basically nothing to say. Everything knows what the biodiversity is and how to protect biodiversity, where to move. And I hope that after this very short presentation, you will find nothing new, but you will be convinced that biodiversity is there, we should use it, and we should go ahead and protect it. Biodiversity is there for us, and we are there for biodiversity. And as we heard in presentations today, we should always watch about biodiversity and how to use it, and above all, how to protect it. Without biodiversity, without this wonderful nature that we have, we will not be surviving on this planet. And everyone knows about the Earth Day, right? More or less. But I want to remind you that there is a Biodiversity Day established by United Nations Organization also. And that will be on the 22nd of May, very soon. And I believe we will be still in Indonesia, in the hottest biodiversity spot in the world. What's biodiversity? Actually, if we go a little bit into the history, even at the beginning of the 20th century, the concept of diversity of life was there. But the word biodiversity, the short word, was not existing yet. And in the 70s, when the Roman Club was, when there's hot topics about the protection of nature and what we've done with that nature at that time, become very hot topic in the negotiation and discussions, the concept was there, but the word, again, was not there. According to the notes of Edward Wilson, when the conference on this National Forum on Biodiversity was created in 1986, at that time, Walter Rosen proposed this short name of biodiversity instead of diversity of life or diversity of biology. And since that, especially after publication in a couple of years materials of this National Forum on Biodiversity, the word became very popular, and that's what we're using today. Of course, the formal definition of biodiversity still stands, and that was established by Convention on Biological Diversity in Rio in 1992. And I want to bring your attention to this. Everyone knows this definition, so I'm not going to read it. But biodiversity at this point does include not only the diversity of animals or plants or organisms. Even today, we saw that when we talk about diversity, a lot of cases we are bringing these wonderful merismatic animals like rhinos or orangutans, but it's just not even the top of iceberg of biodiversity. It's what we humans do. We're relating to something beautiful and close to us. But biodiversity is much more than that. And the formal biodiversity definition includes biodiversity of within the species, between species, and above all, biodiversity of ecosystems. All of these aspects are evolving at this time and getting more and more recognition. For example, when we're talking about the biodiversity of species, in between the species and in the species, we are not only talking about the biodiversity of organisms or genetics, we're also talking about the diversity of the metabolism and metabolites and the chemistry around those organisms. And diversity matters. Diversity matters because, well, if you go to the system theory, the system is established and stable only if it has variety of elements. Only in this case the system could be stable. And biodiversity produced the stability of natural ecosystem. And more diverse systems are more stable and they are more resistant to the climate change that we are experiencing right now. When we talk about biodiversity and when we're bringing this topic to the policymakers, to the government, for a long time we're trying to establish how we measure what the nature gives us. And the two are a little bit competing, but also complementary concepts: nature contribution to people and ecosystem services. And we talk about the natural contribution to people. It's a whole range of materials that we are getting from nature. We tend to think that it's all free, but there's no such thing as free lunch in this world. And we have to pay for everything one way or another. When we are talking about the ecosystem services, we are talking about the benefits that wildlife and ecosystems provide to people. And sometimes we're forgetting about those, but it's very broad spectrum of services. Some of them are listed here, like decomposition and water purification, nutrient cycling, and pollination, for example. But the list could be extended more and more. It's, for many years and in many aspects, people try to put the monetary sign on biodiversity and the ecosystem services and the values that nature provides to people. And it was partially successful. Partially in terms that, yes, in some reasons we can bring the government saying that's a complementary, if we will destroy this, that we have to compensate with the technological solutions, and that will cost us more and more. However, it's creating more and more clear these days there is no such thing as the cost of biodiversity. You cannot put the monetary sign on biodiversity. Because when we do, it's happened that the alternative of biodiversity is nothing. It's either we survive on this planet or not. And there's no price that we can pay for that. However, to bring it to the policymakers, we're trying to do our job. And one of the things which was done quite recently, it's the summary and the big actually document which was provided to help policymakers and government to make decisions about the biodiversity and about technological development. I'm not going to go in details, but basically it's an assessment of finding of patterns and how to do that. It's guidance, what we need to do to make sure that biodiversity is protected, because without biodiversity, we will not be surviving on this planet. And this guide for many, many aspects of our life and policy making and decision making. Of course, you know, just working for a long time with the plants and human health, I cannot miss to put this slide in. Indeed, biodiversity sustains human health in a variety of ways. And what Professor James was talking today about, and we will hear more talks about it, it's just the iceberg. Iceberg on what we can get from plants and to sustain our health and produce new drugs, new medicine from plants. And not only new medicine from plants, but also implementing the plant remedies to fight the disease, new diseases. It's a huge, huge experience with using botanical therapeutics in individual plants and mixtures to treat the COVID condition, for example, and as antiviral in particular. To protect biodiversity, we actually need to know what it is. What do we have on this planet? And the answer is very simple. Nobody knows. We do know that probably we have two super kingdoms, eukaryotes and prokaryotes, divided by kingdoms itself. But how many species we have on this planet, we don't know. And of course, one thing that nobody knows about how diverse they are and what they are and whether they are alive or not are viruses. That's the estimate. 10 million peoples, 10 million species on this planet. The majority of them are eukaryotic species, and that may be underestimate, and that might be not a very particular accurate figure also, because we tend to know more about something that we are studying more. And we do study more the eukaryotic species. You can look at biodiversity in a variety of ways. You can look at it from the position of species diversity. You can look at it as position of phylogenetic diversity. And finally, you can look about the position of biomass. And if you look at the position of biomass, you'll find out that humans, along with the animals that they support, constitute the majority of biomass on this planet. Virus, just to give a small stroke about it, we just started to understand what they are and how they work a little bit. For the last 20 years, we went from a couple of dozens to 100, and we do not call them species, we call them the taxa, and about 100,000 taxa. How many there are viruses on this planet? Again, nobody knows, but the estimates tell us that there are at least tens of millions of viruses estimate in bacteria viruses, and about the same number of eukaryotic viruses. So that's the field we will be moving next in biodiversity. There is no date. There is no date on when it started, but there is a consensus that we are living in a new epoch. We are living in a new epoch which is called Anthropocene. Anthropocene is the epoch, the geological epoch, the complete change of geological status of our planet, which is caused by humans. And along with what we do as species, one way or another, we are affecting biodiversity. And the factors we're affecting biodiversity are different, of course, on different parts of the planet. Terrestrial and marine ecosystems are affected in different ways. But overall trend is that the majority of the land systems are affected by changes of the land use. The marine systems are affected more by the direct exploitation. But all of this, including pollution and invasive species, have effect on biodiversity. And we should carefully watch and regulate it. Global biodiversity loss. We are on the edge of the abyss. We are in the situation when we are losing many species, and actually right now the estimate is that about 10% of species that we do or do not know may be close to extinction or moving to the extinction. What extinction risk? And as you know, there is a red book, red list, and this million species is under threat of the extinction. This graph represents what do we think we know about the threats of the extinction. Again, it shows that in some taxa that we know a little more about, we know that more species are critically endangered and potentially under threat. It's absolutely biased list. It's absolutely biased list because if you look at the amount of species and you look how many of them were evaluated, you will see almost a reverse trend. The majority of the animal species in this world are insects, and they're the least evaluated for the threats. We cannot put biodiversity protection onto to protection of species. We actually have to learn how to live on this planet safe and in just an equitable manner. Humans are the only species which can do on this planet so much harm and so much good. And we need to think about the transformative change to meet the goals of protecting biodiversity. We need to address different drivers of the biodiversity loss. We need to focus on outcomes and basically the several concepts which are trying right now present in biodiversity protection. Those are bending the curve, popular especially after the bending the curves with viruses, COVID virus in particular diseases, and the nature positive features. That's one of the examples of bending the curves. Modeling different scenarios under which we'll continue businesses now and we will completely destroy this planet. We will do something and we can hold it on a little bit, or we can do the right thing, not only protecting the species that they way they are, but also changing our practices of utilizing the species. And we are doing the real steps toward that. For example, Indonesia is pioneering the development of this magic center, which is basically protects genetic and chemical diversity of plants by creating databases and storages using the RAMS technology, and you'll hear more about this, but it's a field deployable technology with little resources can do a lot of wonderful things. As soon as we're talking about indigenous species and the local populations, if you look all around the world, you will find that about 80% of the planet remaining intact biodiversity is linked to the indigenous species, indigenous people, and local communities. And that creates opportunities and also risks. Because these local communities, as we heard from Professor James, should not only think about protecting biodiversity, but they should think about how to survive and how to prosper and how to develop their communities. So poverty alleviation, improvement of healthcare, gender equality, resilience to nature disaster, potential of human rights abuse in the name of conservation, it's all there. It's all at the local level, and we have to be aware of that. The situation when you have to move indigenous people from the areas which would like to conserve are still happening. They were much more common 50 years ago, but it's still happening in this day. One of the hot topics recently was this different attitude toward protecting of biodiversity between global north versus global south. And global north and conservationists, they want to protect biodiversity basically at any cost. They want to halt biodiversity globally, and sometimes basically there's no accountability for historical and current trends, and no really full commitment to pay for that. With the global south actors and the social actors, it's a little bit different story. They are ready to protect, but what's happening? They're concerned about their rights to development. The basic story is, okay, you global north basically developed yourself, used us, exploit us in your capitalistic way, and now you want us to hold and do whatever you do and don't want to pay for that. And it was globally discussed last December in Canada, one of the way. Also, there is a lack of money to do whatever we need to do. And commitment from the last meeting on biodiversity in Canada, actually a very important step into resolving this issue. Kunming-Montreal Global Biodiversity Framework, which was adopted in December, it's a very, very important step to fulfilling this idea of living in harmony with nature to year 2050. It established 4 different goals with 23 targets. The next 8 years will be a very important, crucial time and crucial activities for that goals and targets. And it's a major step in the right direction. It's probably benefited because of the 2-year delay due to COVID. It has a double name because initially it was planned for Kunming in China, but had to be moved to Canada 2 years after dealing with the COVID situation. The goals are in 4 different areas. A is ecosystem, species, and population, basically conservation and trying to avoid degradation. Very important goal under B is biotechnology and biodiversity is sustainable use of management or changing the way we are using the nature. C, genetic resources, and we heard today about the genetic resources. I'm sure you will hear about it more. And finally, goal D actually provides money to do it. Actually try to at least bring the situation to when the financial resources are available for conservation. 700 billion a year is a substantial amount of money, and the majority of that are supposed to be raised just by reducing the amount of subsidies which are harmful for biodiversity. And 20 billions supposed to be raised by the countries and actors to help with that. Out of this, directly subsidiary to the developing country will constitute about $20 billion a year, and I hope $30 billion will be reached by 2030.
Excuse me, Professor Vyacheslav?
Yes.
Your time is still 5 minutes.
That's my last slide.
Okay, thank you.
That's my last slide.
Thank you.
So perfect time, very good, thank you. And the last slide just basically summarized what I want to say. The convention actually moved those targets along 4 wonderful sustainable development goals, meaning altogether, comprehensively, and it takes into account both nature, economic, society, and provide means to do that. So we shall do our part. I hope the Kunming-Montreal Global Biodiversity Framework will be fulfilled, and we need to save biodiversity of this planet, for the sake of this planet, for the sake of the humanity, for the sake of our families, and for the sake of everyone in this room. So you should do your job also, as an individual, as a policymaker, and as an administrator. Thank you for your attention.
Thank you. Very insightful presentation. We learn about the perils of doing nothing, about biodiversity within the species, between species and our ecosystem. Thank you for your presentations, Professor Vyacheslav. And now we are moving on to the second invited speakers. We would like to hear from Professor Satorov and Dr. Wait, it's quite hard to remember your name. Ladies and gentlemen, please welcome Professor Satorov and Dr. Masluna Zarova. Time is yours.
Assalamu alaikum wa rahmatullahi wa barakatuh. Dear colleagues from India, Indonesian, and good morning and good afternoon, dear colleague from USA. Already you know that my name is Saidbek Satorov. I am head of the Department of Microbiology, Immunology, and Virology in the Medical Social Institute of Tajikistan, Dushanbe city. It is a great honor for me to take part in this conference. I hope that next conference will be held in Tajikistan, which will allow you to get acquainted with our specialists in our country. We have a joint presentation with my graduate student, Sulhia Maulana Nazarova, and she will report for you our presentation. Thank you very much.
Good afternoon and assalamu alaikum, dear colleagues and participants of in this conference. I am Sulhia Maulana Nazarova, was born in a beautiful mountainous regions of Tajikistan in Pamir. In 2021, I graduated from the Avicenna Tajik Medical University. After that, I worked for some time in various fields of pharmacy. And after that, I worked for some time in various fields of pharmacy and as a teacher at the Khorog Medical College. That is in the Pamir. In 2021 year, I entered graduate school and currently a graduate student of the second year of Avicenna Tajik Medical University. I want to say a big thank you to the Professor Ilya Rashkin and Vyacheslav Dushenkov, who included in this project, which helps in carrying out scientification and resource work of my dissertation. And also all to say a big thank you to all of you and your beautiful country. The topic of my presentation is today biodiversity of Tajikistan, medicinal endemic plants, species of genus Ferula. Tajikistan is an intercontinental country located at the boundary of the subtropical and temperate climatic zones. It occupies the southeastern part of Central Asia. It borders with Afghanistan, Uzbekistan, Kyrgyzstan, and China, and is close to India, Pakistan, Turkmenistan, and Iran. Tajikistan is a continental country. About 93% of territory occupied by mountains, while about half of the territory lies in altitude of our 3,000 to 7,495 meters above sea level. This geographical position causes a wide variety of natural and climatic conditions here. The highest place of residence of the village of Murgab, 3,600 meters above sea level. The capital of the country, Dushanbe, is located at the altitude of 750 meters and Selementes with low altitudes from sea level located on the territory of the Bokhtar Khujand regions. Biological diversity in this main source of all life, varieties, formation, and development, keeping a stable ecological balance, and providing mankind with material welfare. In modern Tajikistan, a considerable world-specific diversity of flora and fauna. 75% of population living in Tajikistan engaged in cultivation and use of biological resources, and their living standards depend on the state of this resource. 30,000 species of fauna includes biodiversity of the Republic of Tajikistan. The animal world of our country is characterized by a great variety. More than 13,000 species of animals live on the territory of modern Tajikistan. Of this number, 800 species are endemic. Rare and endangered endemic animal species that are listed in the Red Book of our great scientific and socio-economical interests. These include snow leopard live at an altitude of 5,200 meters above sea level. Marco Polo ship live up to 3,500 meters above sea level. The flora of our country varies in large ranges. According to experts, there are more than 9,000 species of spore and flowering plants growing here. Of these, 1,132 species are considered endemic. As can be seen from this slide, the composition of the flora of Tajikistan is represented by 7 types of plants: Angiospermae magnoliophyta, fungi, algae, lichenes, bryophyta, gymnospermae, and prediophyta. Traditional medicine in Tajikistan has deep centuries, old roots dating back to the time of Avicenna. His book, The Canon of Medical Science, describes 900 plants and how to use them back in those days. Folk healers of Tajikistan widely use medicinal plants for the treatment of various diseases in order to treat diabetes, hypertension, liver, kidney diseases, anemies, helminthiasis, parliamentary tuberculosis, gastritis, bronchial asthma, malignant tumor, female infertility. Medical medicine have become guidelines for the selection of promising areas for the study of medicinal plants, and they used in traditional medicine. Currently, more than 23 enterprises engage in the processing and production of medicines which produce more than 540 different types of medicinal plants. The most widely used herbal remedies in Tajikistan include glycerized radicus syrup, extractum altae, an effective secretic as a choleric, diuretic, antiseptic, and angiogelmentic. Artemisia absinthium for atania and antistania. In is used carium crow or extravagant gemolytic anti-inflammatory agents used Hypericum perforatium as a tonic medication, radiola, extract fluid, extract from various types of ferula as biological active and additives. There are 39 species of ferula growing in the territory of the Republic of Tajikistan, of which 6 endemic, which grow at altitudes from 300 to 3,600 meters above sea level. Endemic species include Violatiae caratiginica, Karoteginica, Kosopolyansky, Linchevsky, Decurrens, Bochensi. Of the 39 species of the genus Ferula growing on the territory of our country, the most studied is Stagicorium. The remaining species, including all 5 endemic, remain unexplored. That is, there is no information about their pharmacognostic, physicochemical, and other properties. In the regard, my research work is devoted to the study of the properties of endemic Ferula species. Ferula ovalis, Ferula coistanica, and Ferula gigantea native to our country. Materials and method of research. For the study route, juice or game were used and 70% alcoholic extract and the seeds of the study Ferula species were abated. The content of total polyphenols was determined according to the methodology developed by Raskin Laboratory, Rates University, using a stationary spectrophotometry. The specific virus inhibiting activity of the extract was determined in accordance with the guidelines for conducting preclinical studies of medicines. To determine the antiviral activity of the studied extract, influenza virus strains with different engenic Foila were taken of Vladivostok and Almaty. The antiviral activity of the study extract was combined with the antiviral activity of the commercial anti-influenza drug Tamiflu. At the first stage of the work, we determined the content of phenolic compounds, various parts of the studied plants. It was found that the total content of polyphenols depends on the plant's organ and the method of absorption the extract. First, the roots of all the species of these plants characterized by a slightly high content of phenolic compounds, these their species. Secondly, the concentration of total polyphenols correlates with the method of binding the extract. As can be seen from this slide, the juice from the root of simple squeeze of the seeds contain a higher concentration of this compound, this alcohol extract, which experimentally is related to the ability of the extragent to extract biological active component of the studied part of the plant. At this stage, we studied the antiviral activity only one type of ferula, Ferula coistanica. As shown in the slide, the extract abide from the root of Ferula coistanica inhibit high antiviral activity against influenza viruses than the comparison drug that is Tamiflu. It was found that the separation of replication of both influenza virus strains used in the experiment occurs at significantly low inhibitory concentration of the study extract than the Camprisi drug. For example, the studied extract inhibiting replication of both varieties of the virus a concentration of concentration. This indicates for the drug comparison that Tamiflu was. Conclusion. Analysis of the content of phenolic and of the criteria for influences the inflective of potential source of antiviral drugs is the indicators of effective concentration and selective index. Results of our study showed that the Ferula coistanica extract has a pronounced effective concentration and selective chemotherapeutic index. Thus, the study extract has a high chemotherapeutic effect, 23 times more against the strain competent to Tamiflu. Another strain used in the work showed approximately the same sensitivity to both the extract and the drug Tamiflu. Widely used in influenza, that the index of the selective index of the extract and the Camprisi drug was in the range from 25 to 32.7. Analysis of the content of phenolic component in 4 endemic species of the genus Ferula Tajikistan allude as to identify them species with a high accumulation of phenol. The content of phenols varies against of these plant species varies in large ranges. There is high content of the compound in the roots than in their speed. The maximum total content of phenol was noted in the roots of endemic species Violacea in Ferula coistanica. The study of the antiviral properties of Ferula coistanica extract in relation to strains of Hymen influenza virus showed that the studied extract has a selective effect against influenza virus of viruses injection structures. Thank you very much.
Thank you for the presentation. It's really great for us to know about the flora, fauna, and especially about the plants medicine in Tajikistan presented by Professor Satorov and Dr. Mafluna Zarova. Ladies and gentlemen, before moving on to the invited speakers number 3 in this session, I would like to remind you the coffee and also some refreshments are provided already outside the room, so please enjoy if you haven't got it. Okay, you can enjoy while joining this session. Okay, we would like to continue to welcome Professor Hiroshi Kamitakahara. Time is yours for your presentation.
Good morning, everyone. I'm Hiroshi Kamitaka of Kyoto University. First of all, I'd like to thank organized committee, especially I'd like to thank the Professor Dr. Eni for inviting me for such a fascinating workshop held in Bali, Indonesia. For me, it's the first time to visit Bali. So today, I'm going to talk about our result for the cooperative research project between the former LIPI and now the BRIN and the Kyoto University. So the title of my talk is Chemical Analysis of Some Health-Related Plant Species based on the ethnobotanical knowledge in the case of the Central Kalimantan. Next slide. Okay, yes. Let me talk about my research field, so the cellulose chemistry and polymeric drug for the antivirus, means anti-influenza. And the third one is the utilization of the forest product. And the fourth, now I'm very interested in some product from the biomass towards the circular bioeconomy. Yes. So let's move on to the circular bioeconomy and the health. The bioplastics and the polymer, we can say that is commodity chemicals. And as you can see in the some picture, the forest can absorb the carbon dioxide and produce oxygen, and the circle like the bleeding the forest and taking some product from the forest must be very important. And in this sense, the pharmaceuticals as special chemicals, we have to protect the rare plant species in the forest area. Yes, my research field is Central Kalimantan. And there is one company name is Salibumikusma SBK, and there is a logging concession in the Central Kalimantan area, as in the black circle. And there's a plantation area, but the logging concession, in the concession area, local Dayak people live in, and they are living in the protected area, but outside the protected area, the company is taking the logs, and also they are making some logging trees from the seed. Okay, next. Okay, when about in 2011 and 12, we have visited the concession area with our colleague from the LIPI, Indonesia. And yes, now we these are the picture for collecting the samples in the forest or in some villages inside the concession area. And as you can see, we had interviewed the person in the village, how they use a plant in the village and for curing some diseases or something. This is some example about ethnobotanical knowledges. As the left side, you can see this Indonesian language and the local name, and also the third name, there is a scientific name. So we knew this very interesting knowledge from the healer. I can say the local doctor in the village. And I think after getting the knowledge in our lab, we have to investigate what kind of research had been already carried out. And then after that, we will decide what kind of plant we will study in detail. So this one is found. It is so-called giant fern, Angiopteris ebecta. Might be interesting for our project, and we collected the rhizome of this Angiopteris species. And the right side, it is the name of the compound is angiopleroside, and we had extract such a compound. And yes, I have to say, he is Pak Garuda. He knows many medicinal plants in the village of the inside concession area. So I'd like to thank Pak Garuda. And after the this slide shows result of neutral sugar analysis and lignin analysis of the rhizome. So there are some sugars inside in the rhizome, probably by cellulose, hemicellulose, and a small amount of the lignin. Yes, rhizome of the Angiopteris ebecta is extracted by the acetone with some amount of the water, and concentration of acetone in water affects the yield of the active compounds. And after getting the data from the extraction yield, we had purified the compound by preparative TLC to obtain the pure compound. Here you can see right side there is chromatograms of LCMS. After purification, compound is pure, and we can say from the next slide. Yes, this slide shows two-dimensional NMR experiment data, HSQC, and the left side, as you can see, the full rhizome analysis data. So there is some black-colored spot sugars, or probably the polysaccharides, and the red one, angiopteroside. And after complete purification, we can see only such an angiopteroside. This slide shows proton and carbon NMR spectra. So this spectrum shows the compound is completely pure. We could assign all the carbon and proton signals. Yes, up to now we could not do the biological activity using live animals. We have checked the antimicrobial activities, but unfortunately this compound has no effect, has not antiviral activities. As this Angiopteris ebecta is said to have some effect against cancer or lung diseases, but we have no chance to test such clinical effects for this compound. So I'd like to talk about the similarity of the compound between angiopteroxide and osmundarine. The right side, osmundarine would be obtained from the zenmai, the local name of Japan. This is the food, a local food in Japan, and you can say agricon of the is an epimer of at the C5 position. So this is, similarity of the compound is very interesting for us. So that we'd like to continue further investigation for such compounds. Yes. Now let us move on to the next topics about natural product from the central Kalimantan. We say the ginseng hutan, the forest ginseng. So we had collected 3 kinds of ginseng. Scientific name is Lenneria elliptica, Lenneria speciosa, and so on. So traditionally, these ginseng are used as a tonic in Kalimantan, but surprisingly, it is also used as tonic in Thailand and Myanmar too. Yes. So the lubiadin, its color is like the yellow or orange, and the color of the root of bark is like the very specific, and we can see the red colored root in the forest that must be comes from the color of the anthraquinone, having the long conjugated system of the compound. And we had evaluated aphrodisiac activity of yellow ginseng using the mice. So the maybe detailed study is very complicated using the mice. So the background I'd like to talk about is sexual dysfunction could result in the medical and the social and the reproductive problem, and we should find some drugs, natural product for such diseases. So the Nemrenia elliptica, the local name is written here. So distribution, I can say the southern Myanmar, West Malaysia, Sumatra, Borneo, and traditionally they are using used as treated deer here, fever, cancer, and so on. The aim of this study is to determine such activity of yellow ginseng extract on the sexual behavior. So the root of extractive from the yellow ginseng is collected and extracted by using the ethanol 96%, and we use mice, and we have checked the ethical approval was obtained by the Indonesian rule. So we have managed the experiment under this chart of the experiment. Negative control, positive control, the sildenafil citrate. It is the common name for the commercial name is Viagra. That is used as a positive control, but different activity. So the 5 animal behavior were tested and compared, and yes, you can see the from this graph the amount of latency of the mice. That, as you can see, the left side the pink star, and please compare the pink star, 2 pink stars. The left side is the negative control, and the right side is extract dose of the 250 milligram per kilogram of the mice. So we can say the number of is decreasing using the 250 milligram dose of the yellow ginseng extract. So that means improve the sexual response. And next, this one, the month frequency. So some positive control compared with the positive control. After 10 days dose of the extractive of the 250 milligram per kilogram, so we can say that after 3 days dose, that have increase the number of frequency. That means increase libido or sexual motivation. So this one shows, as you can see from the left side, negative control for the 3 days to the 10 days. Number of the latency decreased. So that means stress in feels the mice feel the stress under the this experiment, but after 10 days dose, we can say that compared with the negative control, the dose of the 250 milligram of the ginseng extract would increase the latency. So that means that have the some effect for the mice behavior. So that we'd like to conclude from my point of view, the animal behavior study is very difficult to conclude using some chemicals, but such a yellow ginseng extract have some medicinal effect regarding the such an as used for the tonic. Yes, maybe I have to talk about access and the benefit sharing process. For doing the research in Indonesia, we have to consider the access and benefit sharing (ABS). We have to make the letter of agreement, materials transfer agreement, and of course, we have to consider importance of the ethnobotanical knowledge. So I'd like to talk about what kind of collaboration system developed during these 15 years. So this is the Japan ASEAN Science, Technology and Innovation Platform, JASTIP. Is very effective for the collaboration between Indonesian researcher and Japanese researcher. For me, like me, the promotion of the sustainable development research. So actually, you can see that this is a homepage of JASTIP. Our package is work package 3, and bioresources and the biodiversity research groups. And Indonesian collaborator, Professor Dr. Andrea Augusta has some webinars in the framework of the JASTIP. We have getting some knowledge, and we knew the new result and enlarge the communities in the field of the biodiversity and the bioresources. Okay, so I'd like to acknowledge the Professor Kanzaki for his continuous support. He is a former leader of this large project, and we had a chance to visit Central Kalimantan about 10 years ago. And now we had a next project between Indonesia and Japan. And for example, Bioprospection Group would have the collaboration with Dr. Rudiana Santi for identification of the plant collection and identification. And we'd like to thank the former LIPI and now the BRIN for the very close collaboration for a very long time. So thank you so much for kind attention.
Thank you, Professor Hiroshi Kamitakahara. Ladies and gentlemen, we have heard about chemical analysis of some health-related plants, species collected in central Kalimantan, and Professor Hiroshi also take us to the ginseng forest in central Kalimantan. Ladies and gentlemen, now we're moving on to the question and answer session. We have about 30 minutes. Okay, I would like to give the first chance to the audience here who would like to ask questions to our four invited speakers. Please take this opportunity. If you don't mind, please stand up in front so you can be seen on Zoom and ask directly to our invited speakers. Perhaps from that side, here and there. Questions or things you would like to share? Oh yes, Professor Annie. Okay, time is yours. Would you like to stand there in front? Yes.
here. Well, just my idea, because you can see here in front of us, it's 3 countries, 4 with the moderator. So Indonesia, US, Tajikistan, and Japan. We have term in common, biodiversity, yeah, so, and health. So we all heard already, so maybe then we can connect after this, not just between, like Professor Kamita Kahara mentioned, with LIPI, Brin now, myself, and then I've been also involved a lot in JUSTIP project from the beginning. And from Tajikistan, this is new for me. I mean, I haven't heard about your research on this specific area, and Slavic I know from just from last year with Professor Ilya. So I think from this international conference or this event and workshop on the 3rd day, maybe we can develop something that we can write a joint proposal, for example, and not just listening to the invited speakers or the keynote speakers, but also during the break or lunch break, we can gather, whatever you think that interested to you, so that you can develop something. So I just want to ask to the Tajikistan colleague that it's very interesting, but because your area is more temperate region, right? Yeah, and we are in Indonesia more tropical regions. Yes, so then we can exchange that one. So we can explore how to collaborate through Rutgers and also with the Kyoto University. I'm sure that we can develop something. This is not a question, but this is like an appeal that maybe we can work together after that, after this event. Thank you.
Thank you, Professor Annie. The 3 invited speakers would like to give comments on the statement of Professor Annie, please. Professor Satolo?
I think it's an excellent idea of connecting different regions, and as we heard today from Professor Hiroshi, that collaboration between different countries is very fruitful. Also, it's great that for the regions there is a wonderful mechanism of sharing benefits. I'm not sure that this mechanism is completely developed for Tajikistan or other parts of the world. Just going back to the history, in 1992, the Convention of Biodiversity had very wonderful idea that by proclaiming that biodiversity belongs to the country and there is a need for a mechanism of benefit sharing, it will be pushing for collaboration. That's exactly what we're talking about. Unfortunately, for the last 30 years, we've seen that the first part worked excellent. Indeed, countries took possession of their genetic resources and biodiversity very well, efficiently, including Brazil, India, and I'm sure Indonesia also. Unfortunately, the second part of benefit sharing and promoting collaboration was a little bit abstracted. And that's actually resulted in slowing down of exploration of bioprospecting and biodiversity on international level. So I hope that it could be overcome, and this international collaboration, comparing different regions with the different climatic zones, may be very, very productive. Thank you.
Thank you. Professor Satoro, would you like to say something? No? Professor Hiroshi? Yeah.
So I can say that Indonesia is a country of biodiversity, And I think from my point of view, I, as Professor Eni told us, that I'm working for making some collaboration between the Japanese scientists and scientists in the Asian, ASEAN countries. So the problem is we don't know each other for what they're doing. So like Professor Eni, she knows very well many researchers have doing the many used for making the James Simon talked about research in the South Africa, but I myself don't know at all about African research. So I'm very interested in the such a field, but I know that some research in the Asian country like Myanmar, Thailand, and of course Indonesia. So I think collaboration in other countries will produce much more interesting scientific results. Thank you.
Well, thank you. Another questions, Professor Ilya Raskin. Okay.
First of all, thank you for
Yes. Yeah.
Yes. Am I seen now online? Yes. So I think, first of all, thank you, the morning speaker. It's been wonderful, and I'm delighted to be here. It was very informative. I think what might be very important, again, related to the subject of this morning's discussion, if as the result of this conference, we start forming at least some idea, what is the most important incentive for Indonesia with its wonderful biodiversity to protect it? I think some kind of recommendation, some kind of ideas would be very, very important, because obviously, yes, we started with this notion that bioprospecting is going to extract some value from biodiversity, therefore we have to protect it. And I have been there, and some of my colleagues have been there, and preaching this for a long, long time, for many years. Unfortunately, and we have to be honest, unfortunately, not much came out of it. I mean, yes, benefit sharing, yes, Rio Convention, yes, Nagoya Protocol, but if you really calculate how much money came back to the community from new drugs developed from biodiversity, I frankly think it's a very small amount. So what's next? Is it ecotourism? A lot of countries now investing in truly creating, and I see in Africa, farms are being transformed into the lodges, and quite a bit of money actually come back to the communities because they employ local communities. Is that a model which, for example, Indonesia can adapt? Is it an educational potential? Is it just purely promotion of the aesthetic values of biodiversity? So I think it will be very important if we can formulate at least something, because frankly, with all due respect, and I'm the one who has been preaching this dogma of bioprospecting. We don't call it bioprospecting, we actually call it bioexploration, because bioprospecting sort of has a bad connotation, so we changed the word for bioexploration, but still, I'm not aware of any new drugs which came out of the novel discoveries in the last 20, 30 years. So what do you think? I would love to hear your ideas. What is the model for Indonesia? Which model will work? Because Dr. Rushenkov said, well, biodiversity is priceless, so we can't really put economic value. But in essence, we have to, because it's a capitalistic world and everything is driven by economic incentive. And if there's no incentive, of course, oil palms will replace forests in Borneo, as we see. So what do you think, my dear colleagues, is an incentive for Indonesia to protect biodiversity? What should we tell the world and the funding organizations? What's the next model?
Okay, thank you for the questions and also ideas, Professor Ilya Raskin. Okay, next questions before all the invited speakers answer the question.
Thank you. I was very appreciative with all presenter in this morning, but my suggestion in my experience, I came from academic sector, and I also practitioner in the field, this kind of things. What is lacking in Indonesia or need to be strengthened is involvement or participation from private sector. So, but of course, we have to select private sector who own their heart, you know. It's not just private sector, you know, but specific private sector. That's where we are lacking. So if we can develop this kind of integration between those sectors, would be wonderful. This is my comment.
Ya, dengan Bapak siapa, Pak?
Saya Bapak Eni Sudarmono.
Oh, ya baik. Terima kasih. Okay, now would you like to respond the two questions from perhaps Professor Satoru? Would you like to start? Yes?
Thank you. I will be speaking Russian, and my friend Slavik Dushenko will translate. Okay? Во-первых, еще раз большое спасибо организаторам конференции, что нас пригласили, и вы впервые приехали к вам, и заодно приняли участие в организованном вами совместно с Радгерским университетом конференции.
First of all, I would like to thank you one more time for inviting us to visit this wonderful country and participate in this conference.
Так, кроме того, я хотел бы отметить, что наша страна, наша республика Таджикистан является богатой страной по различным биологическим ресурсам, в частности, лекарственным растениям.
I would like to mention that Tajikistan is one of the very rich countries in the resources, especially in the medicinal plant resources.
И, к сожалению, несмотря на вот большое разнообразие различных эндемических видов лекарственных растений, 99,9% этих растений до сих пор остаются неизученными.
And it's a pity that even with this wonderful biodiversity of medicinal plants, and mostly of them are endemic for Tajikistan, 99% of are not researched yet.
Это связано с тем, что, опять же, к сожалению, у нас в республике нет лабораторной возможности, чтобы проводить такого рода исследования, изучить фармакогностические, биологические или другие признаки этих растений.
And to a big regret, the country still doesn't have the modern technology for investigation of the medicinal plants.
И наше сотрудничество с Радгерским университетом и с другими научными центрами, которые работают для Радгерского университета, с которыми нас они свели, дают свои результаты и плоды.
That's why our collaboration with Rutgers University and with other universities in the United States is very fruitful.
И я думаю, что дальнейшее сотрудничество с вами и с теми специалистами, которые здесь присутствуют, особенно индонезийскими коллегами, которые имеют достаточно опыта, нам дают возможности расширить свои возможности, изучить неизученные виды наших лекарственных растений. Спасибо.
And I hope our trip to this conference will result in extending collaboration with Indonesian colleagues so we can study the medicinal plants on different level.
Спасибо большое.
Thank you very much for your attention.
Would you like to respond directly? No? Okay. Well, thank you, Professor Satorov and Vladislav. And now I would like to invite the questions from the participants via Zoom. Okay, let me check whether there are participants who would like to ask questions. For those joining via Zoom meeting, if you have questions, you may raise hands using the features of raise hand, or just give your questions on the chat box as I have invited you to ask questions. I think there's no questions yet. So how about moving on to lunchtime to close the session? Okay. Well, before we enjoy our lunch, perhaps Balinese cuisines are provided to you in the lunch meeting. I would like to invite the three speakers to give concluding remark regarding the presentations and also our topic today. So, time is yours.
I just want to say that the conference is going very well so far, and this particular session is of great value, in my opinion. The indigenous populations, the local populations, and preservation and utilization of natural resources is a very hot topic and very difficult one. And I hope that this goal of living together and in harmony with nature by 2050 is achievable, and we all should do our job in making it happening. Thank you.
Thank you. Professor Satarov and Dr. Surya, would you like to say something? No?
Еще раз здравствуйте, дорогие участники конференции. Я хочу сказать всем вам спасибо за организацию такой чудесной конференции. Это для меня...
Well, I want to say one more time, thank you so much for organization of this brilliant conference.
Это для меня, я как являюсь аспирантом, это для меня большая возможность, чтобы участвовать здесь. Это для меня большое достижение, и я очень рада этим.
For me, as a junior researcher just starting my career, it's a very important step, and I'm very happy that I was able to join you.
И я всех вас приглашаю к нашим, к нашей страну, в нашу страну.
And I invite you all to visit Tajikistan, and I hope we can have the conference there.
Thank you very much.
Thank you, Professor Vyacheslav. Unfortunately, I don't speak Russian, so I can't replace your role as an interpreter. Next time I'm going to learn Russian. Okay, Professor Hiroshi?
Yes. So actually, I enjoyed the morning session very much, and I knew much interesting topics in the field of biodiversity, and of course, this is a very good chance to know each other in this room, and hopefully we could talk in persons, and probably that is the most important for the face-to-face in-person meeting. So hopefully our talk will be a kind of the catalyst between the persons. Thank you.
So thank you. I think we can end the session with a very fruitful and insightful knowledge in this session about biodiversity, traditional medicines, and also the involvement of community and the potentials of making collaborations. Thank you again for your participations, ladies and gentlemen. I would like to end the session by inviting you to give a big round of applause to our invited speakers. Thank you, Professor Vacheslav, Professor Satorov, Dr. Surya, and Professor Hiroshi. And I'm going to give the time back to the MC. Thank you.
Thank you very much, Dr. Sri. But unfortunately, I think we have one question in Zoom for Professor Hiroshi. Is that okay to state the question or? Okay, so this is a question for Professor Hiroshi. It's about medicinal plants in Kalimantan. When you first got into the bioexploration research in medicinal plants in Kalimantan, what was the first stage you did? Was it mainly based on the local wisdom, where you talked to the local shaman about the medicinal plants, or you already got a specific database of the local medicinal plants? And if someone wants to start an exploration in this field, in different islands in Indonesia, what is your suggestion?
Thank you for the question. The first stage of our exploration, we had collected many plants, not only the medicinal but also other plants. And after identification of the plant name by the Bogor Botanical Garden, we could understand what is important species. And then after the discussion with other members of our collaborators, we decide which plant species will be focused. So after focusing, we will start actual experiment like the extraction and identification, purification. So then maybe for there are many plant species in many islands in Indonesia. So actually, I think for me, I have only explored the plant species in Kalimantan, but I think many researchers from the Blink or the LIPI had visited many islands to collect the plant species. So I think, of course, I had informed that, for example, Papua is the eastern part, must be very interesting, but the important thing for the foreign researchers is to get research, how can I say? Permit, research permit, and we have to collaborate with Indonesian researchers and get good knowledge about how we manage like the administrative thing is very important. And after that, maybe for the foreign researchers, they can visit many islands to get a new knowledge about the plant species or bioprospecting. They can do the bioprospecting research there. Thank you.
Thank you very much, Professor Hiroshi, for your answer. So that is the suggestion from Professor Hiroshi. If the researchers here want to explore the same field in different islands in Indonesia. Well, ladies and gentlemen, we have seen our invited speakers presenting their amazing papers. I would like to say thank you to our moderator for leading the session, and also to our invited speakers for remarkable topics of research and presentation. Therefore, we would like to present certificate of appreciation to our invited speakers. We invite representative of Hartek Indo, PT Hartek Indo, PT Elokarsa Utama, and PT Syngenta to hand the token of appreciation to our invited speakers. To our invited speakers, please kindly stand up and come to in front of the stage to receive the token of appreciation. We would like to invite the representative of PT Hartek Indonesia to present the token of appreciation to our invited speakers, and then we invite PT Elokarsa Utama, and then PT Syngenta. Thank you very much to our representative of our sponsorships and also the invited speakers. For the representative and invited speakers and also moderator, please remain on your position to do a photo session. We would like to invite back our representative from PT Hartek Indo, Elokarsa Utama, and also PT Syngenta to have a photo session together with the invited speakers. So the committee have prepared udang and traditional fan that symbolizes the Balinese culture. Now we will have a photo session. 1, 2, 3. Okay. One more with Bali in 3. 1, 2, 3. Bali. Okay. Well, thank you very much. We hope you enjoy your stay in Bali, and thank you once again for amazing topics and presentation. We invite all of our invited speakers for in plenary session 1 to kindly return to your seat, because after this we will have a lunch break. But before that, we realize that this international conference can be conducted with the support from many parties. Therefore, we would like to express our gratitude to our sponsors through sponsor presentation video. And also, we would like to invite all of distinguished guests to have a photo session together. So please come forward. We will take a picture with our invited speakers before we have our lunch break. We also would like to invite all of the participants before we have lunch break to come forward to have a photo session together. Once again, we would like to invite all of the participants. Kami silakan pada para peserta, presenter paralel sesi untuk maju ke depan dan foto bersama dengan para invited speakers. Baik, kami untuk agar semuanya bisa di dalam frame, kami atur posisinya. We would like to arrange the position. For the ones who are taller, please stand on the stage. So on the stage behind, where the speakers give their presentation, and then some of you can also bend on your knee. So please make sure that everyone can be on frame. We also have a banner. We hope that everyone can hold it. There are places still over there. So for participants, there are places over there as well, so you can be on frame. Okay. 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 Photographer, ready? Everyone already on frame? Ibu Gung, silakan ikut bersama berfoto. Oh, tidak perlu, Ibu. Mejanya kena nggak? Kena. Mau digeser aja. Boleh. Minta tolong Pak, digeser. Jadi ada kata Bali, ada biodiversity, ada bioteknologi. Baik. Okay. Okay, so formal ones, 1, 2, 1, 2, 3. Once again, 1, 2, 3. Now say Bali in 3, 1, 2, 3, Bali. Yeah. Once more. Okay, biodiversity. Okay, is that too long? Okay, okay, Bali. 1, 2, 3, Bali. Okay, thank you very much to all of the participants, presenters, speakers, distinguished guests. Thank you very much. Now we will have our lunch break. Because time has shown lunchtime, we will have 60 minutes break. Please enjoy the lunch provided by the committee and the hotel at Sanur Harum Restaurant on the first floor of this hotel. I'm really sorry, but we will have only 30 minutes break because at 1 p.m. everyone should be here because we will continue the plenary session too. So please enjoy the lunch provided by the committee and the hotel on the first floor, Sanur Harum Restaurant. Sekali lagi kami ingatkan kepada peserta untuk kembali ke hall ini, ke venue pada pukul 1 karena kami akan melanjutkan acara dengan plenary session kedua.
Kami percaya bahwa setiap hasil yang dikeluarkan haruslah sebuah aksi terbaik dari tindakan melayani sepenuh hati.
We would like to remind you that at 1 p.m.
Memberikan
everyone
pelayanan
should
terbaik
gather
dari
here
hati,
again
menjawab
because we will continue
tantangan
the
dan
agenda
kebutuhan
with plenary
agar
session
Anda
2.
dapat menemukan pilihan
Sekali lagi
sesuai
kami ingatkan
kebutuhan
kepada
Anda.
peserta bahwa
Kami merupakan
waktu
distributor
makan siang
alat
hanya 30
laboratorium
menit
dan farmasi
dan kami
yang dapat terus diandalkan
himbau kepada
sejak
seluruh
tahun
peserta untuk kembali ke ruangan
1978.
ini pada pukul 1 siang
Fokus
untuk
kami
melanjutkan
adalah
agenda
untuk membangun
sesi
Indonesia
pleno nomor
lebih
2.
maju lagi dengan memanfaatkan teknologi berkualitas tinggi di bidang laboratorium dan farmasi.
We also would like to
PT
remind you for
Hartek
your
Indonesia
valuable
selalu berdedikasi membukakan jalan
Make
bagi
sure that
teknologi terbaik dunia
your
agar
valuable
dapat diterapkan dengan
belongings like
maksimal
laptop,
sesuai
your
kebutuhan Anda.
smartphone, or any other
Dengan pengalaman
belongings
dan
to
ilmu yang
be
kompeten,
taken while you
PT
are out
Hartek
of this
Indonesia
venue.
telah berkontribusi memajukan industri hingga universitas-universitas di Indonesia. Bersama dengan brand terkemuka di dunia, kami berkolaborasi mempersembahkan yang terbaik
Our
untuk
participants and
Anda,
speakers can have
memberikan
lunch
pengetahuan baru,
on the first floor,
saling hadir
Sanur
untuk
Harum
membantu,
Restaurant.
tanpa mengorbankan mutu, menjadi solusi bagi Anda dalam menyediakan produk-produk Anda. Dengan sumber daya manusia yang ahli pada bidang-bidangnya, kami saling berkolaborasi membangun tim yang berintegritas, yang selalu dan akan terus melayani Anda dengan sepenuh hati. Dalam hal penjualan produk, PT Hartek Indonesia memiliki tim layanan purna jual yang handal, memberikan layanan terbaik, dan membantu menemukan solusi terbaik untuk Anda. Kami adalah partner yang dapat diandalkan untuk membangun masa depan. Kami terus menatap ke depan untuk terus berinovasi, mengembangkan visi besar, membangun teknologi industri laboratorium dan farmasi yang lebih baik. Kami percaya bahwa setiap hasil yang dikeluarkan haruslah
penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Food doesn't come from the grocery store. It comes from the land. which means if we want this, we need to protect this and them. How can we produce enough food while also nurturing the land and the farmers who grow on it? 2 words: sustainable agriculture. That means the soil stays healthy, the environment stays healthy, and the farms, you get it. So what does that look like? At Corteva Agriscience, it looks like seeds that are adapted to different environments. So farmers not only conserve much-needed water, they increase the likelihood that seed will make it to harvest. Our biopesticides help farmers control pests and protect crops, naturally. And it's not just our products. We're advising farmers on sustainable farming practices, maximizing yields, and enhancing food quality. Corteva knows that the future of food requires reimagining how we grow it, protecting where we grow it, and ensuring we can keep growing it.
Sustainability. It's a word you hear a lot. But what does it really mean? At Corteva Agriscience, sustainability is more than a word. It's who we are and what we do. We're in it for the good of farmers. We're working hard to increase the livelihoods and sustainable farming practices of 500 million smallholder farmers. We're in it for the good of the land. We're improving nutrient and water stewardship, productivity, and soil health on 30 million hectares of global agricultural land. We're in it for the good of our communities. We're empowering women and youth across the globe to help them become successful business owners and providers for their families. And we're in it for the good of the planet. We're reducing greenhouse gas emissions, conserving water, and enhancing biodiversity in our operations and supply chain. Sustainability. It means more than ever, for us and for the world. It's the foundation of our future, and we're building it right now. For farmers, for consumers, for our planet. Together, we're in it for good.
ITS Science Indonesia is one of business division under ITS Group with headquarters located in Singapore. ITS Science Indonesia offers various analytical instruments for laboratory, medical instruments, and laboratory furniture. For more than 40 years, ITS is moving forward with a vision to make an impact in the industry they serve and a mission to serve better. ITS has core values to put professionalism, partnerships, and performance forward. ITS Group is providing its services around Southeast Asia countries such as Malaysia, Singapore, Thailand, Vietnam, Indonesia, Philippines, and Cambodia. In Indonesia, ITS Science has 2 offices, located in Jakarta and Surabaya. Inside ITS Science Indonesia office, there are facilities to support the work such as training room, and demo lab. ITS Science Indonesia also actively create or participate in various events such as webinars, seminars, product training, and exhibitions. Here are some brands that ITS Science Indonesia distributing. Products that distributed are from top and reputable brands worldwide. We are also proud to serve the needs of our customers, from governments, hospital, industrial, to universities. If you need help for your laboratory needs, contact us. ITS Science Indonesia, bridging science, inspiring possibilities.
CJ Indonesia adalah bagian dari sebuah bisnis global CJ yang berpusat di Korea Selatan. Memulai ekspansi bisnis pertamanya di luar negeri yang berlokasi di Indonesia pada tahun 1988. Kini perusahaan telah tumbuh menjadi korporasi besar, memiliki beragam unit bisnis di antaranya bioteknologi, feed and livestock, food, logistik, bioskop, dan bakery. Seiring perkembangan pasar dan perubahan tren dunia menuju peningkatan kehidupan yang lebih baik, sehat, dan ramah lingkungan, CJ terus melakukan berbagai terobosan-terobosan baru dan inovasi dalam bidang bioteknologi untuk menciptakan produk bio yang ramah lingkungan. Hanya ada sedikit perusahaan yang mempunyai teknologi biodegradable atau penguraian dengan organisme hidup. CJ Bio secara khusus memiliki pengetahuan dan pengalaman pada bidang ini, karena CJ Bio telah mampu mengembangkan dan meningkatkan proses bioteknologi yang kompleks, melalui diproduksinya berbagai produk asam amino dengan teknologi fermentasi. Dalam pengembangan produk White Bio, CJ Bio kini telah berhasil memproduksi produk bioplastik polihidroksialkanoat atau PHA. PHA adalah salah satu bahan untuk membuat plastik ramah lingkungan. PHA adalah polyester cadangan yang diakumulasi sebagai butiran intraseluler oleh berbagai macam bakteri. Polihidroksialkanoat atau PHA dihasilkan melalui beberapa tahapan diawali dengan pemrosesan bahan baku raw sugar menjadi glukosa. Proses sakarifikasi pencampuran glukosa dengan pH strain dilanjutkan dengan proses fermentasi yang menghasilkan produksi intraseluler. Setelah itu dilakukan proses pemurnian atau purifikasi, yaitu pelepasan unsur PHA di dalam sel untuk menghasilkan polimer PHA. PHA adalah satu-satunya plastik yang dapat terurai secara hayati 90% dan dapat terurai setelah 120 hari di lautan. Sejalan dengan filosofi CJ dalam menjalankan bisnisnya untuk berkontribusi pada perekonomian nasional, CJ akan terus menjadi perusahaan nomor 1 yang bergerak dalam bidang bisnis bioteknologi, yang terus peduli pada kelestarian lingkungan dan maju berkembang bersama Indonesia.
ITS Science Indonesia is one of business division under ITS Group with headquarters located in Singapore. ITS Science Indonesia offers various analytical instruments for laboratory, medical instruments, and laboratory furniture. For more than 40 years, ITS is moving forward with a vision to make an impact in the industry they serve and a mission to serve better. ITS has core values to put professionalism, partnerships, and performance forward. ITS Group is providing its services around Southeast Asian countries such as Malaysia, Singapore, Thailand, Vietnam, Indonesia, Philippines, and Cambodia. In Indonesia, ITS Science has 2 offices located in Jakarta and Surabaya. Inside ITS Science Indonesia office, there are facilities to support the work such as training room, and demo lab. ITS Science Indonesia also actively create or participate in various events such as webinars, seminars, product training, and exhibitions. Here are some brands that ITS Science Indonesia is distributing. Products that distributed are from top and reputable brands worldwide. We are also proud to serve the needs of our customers, from governments, hospital, industrial, to universities. If you need help for your laboratory needs, contact us. ITS Science Indonesia, bridging science, inspiring possibilities.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions, science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Kami percaya bahwa setiap hasil yang dikeluarkan haruslah sebuah aksi terbaik dari tindakan melayani sepenuh hati. Memberikan pelayanan terbaik dari hati, menjawab tantangan dan kebutuhan, agar Anda dapat menemukan pilihan sesuai kebutuhan Anda. Kami merupakan distributor alat laboratorium dan farmasi yang dapat terus diandalkan sejak tahun 1978. Fokus kami adalah untuk membangun Indonesia lebih maju lagi dengan memanfaatkan teknologi berkualitas tinggi di bidang laboratorium dan farmasi. PT Hartech Indonesia selalu berdedikasi membukakan jalan bagi teknologi terbaik dunia agar dapat diterapkan dengan maksimal sesuai kebutuhan Anda. Dengan pengalaman dan ilmu yang kompeten, PT Hartech Indonesia telah berkontribusi memajukan industri hingga universitas-universitas di Indonesia. Bersama dengan brand terkemuka di dunia, kami berkolaborasi mempersembahkan yang terbaik untuk Anda, memberikan pengetahuan baru, saling hadir untuk membantu, tanpa mengorbankan mutu, menjadi solusi bagi Anda dalam menyediakan produk-produk Anda. Dengan sumber daya manusia yang ahli pada bidang-bidangnya, kami saling berkolaborasi, membangun tim yang berintegritas, yang selalu dan akan terus melayani Anda dengan sepenuh hati. Dalam hal penjualan produk, PT Hartech Indonesia memiliki tim layanan purna jual yang handal, memberikan layanan terbaik dan membantu menemukan solusi terbaik untuk Anda. Adalah partner yang dapat diandalkan untuk membangun masa depan. Kami terus menatap ke depan untuk terus berinovasi, mengembangkan visi besar, membangun teknologi industri laboratorium dan farmasi yang lebih baik.
adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Food doesn't come from the grocery store. It comes from the land, which means if we want this, we need to protect this and them. How can we produce enough food while also nurturing the land and the farmers who grow on it? 2 words: sustainable agriculture. That means the soil stays healthy, the environment stays healthy, and the farms, you get it. So what does that look like? At Corteva AgriScience, it looks like seeds that are adapted to different environments. So farmers not only conserve much-needed water, they increase the likelihood that seed will make it to harvest. Our biopesticides help farmers control pests and protect crops naturally. And it's not just our products. We're advising farmers on sustainable farming practices, maximizing yields, and enhancing food quality. Corteva knows that the future of food requires reimagining how we grow it, protecting where we grow it, and ensuring we can keep growing it.
Sustainability. It's a word you hear a lot. But what does it really mean? At Corteva Agriscience, sustainability is more than a word. It's who we are and what we do. We're in it for the good of farmers. We're working hard to increase the livelihoods and sustainable farming practices of 500 million smallholder farmers. We're in it for the good of the land. We're improving nutrient and water stewardship, productivity, and soil health on 30 million hectares of global agricultural land. We're in it for the good of our communities. We're empowering women and youth across the globe to help them become successful business owners and providers for their families. And we're in it for the good of the planet. We're reducing greenhouse gas emissions, conserving water, and enhancing biodiversity in our operations and supply chain. Sustainability. It means more than ever, for us and for the world. It's the foundation of our future, and we're building it right now. For farmers, for consumers, for our planet. Together, we're in it for good.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions, science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
CJ Indonesia adalah bagian dari sebuah bisnis global CJ yang berpusat di Korea Selatan. Memulai ekspansi bisnis pertamanya di luar negeri yang berlokasi di Indonesia pada tahun 1988. Kini perusahaan telah tumbuh menjadi korporasi besar, memiliki beragam unit bisnis di antaranya bioteknologi, feed and livestock, food, logistik, bioskop, dan bakery. Seiring perkembangan pasar dan perubahan tren dunia menuju peningkatan kehidupan yang lebih baik, sehat, dan ramah lingkungan, CJ terus melakukan berbagai terobosan-terobosan baru dan inovasi dalam bidang bioteknologi untuk menciptakan produk bio yang ramah lingkungan. Hanya ada sedikit perusahaan yang mempunyai teknologi biodegradable atau penguraian dengan organisme hidup. CJ Bio secara khusus memiliki pengetahuan dan pengalaman pada bidang ini, karena CJ Bio telah mampu mengembangkan dan meningkatkan proses bioteknologi yang kompleks, melalui diproduksinya berbagai produk asam amino dengan teknologi fermentasi. Dalam pengembangan produk White Bio, CJ Bio kini telah berhasil memproduksi produk bioplastik polihidroksialkanoat atau PHA. PHA adalah salah satu bahan untuk membuat plastik ramah lingkungan. PHA adalah polyester cadangan yang diakumulasi sebagai butiran intraseluler oleh berbagai macam bakteri. Polyhidroksialkanoat atau PHA dihasilkan melalui beberapa tahapan diawali dengan pemrosesan bahan baku raw sugar menjadi glukosa. Proses sakarifikasi pencampuran glukosa dengan PHA strain dilanjutkan dengan proses fermentasi yang menghasilkan produksi intraseluler. Setelah itu dilakukan proses pemurnian atau purifikasi, yaitu pelepasan unsur PHA di dalam sel untuk menghasilkan polimer PHA. PHA adalah satu-satunya plastik yang dapat terurai secara hayati 90% dan dapat terurai setelah 120 hari di lautan. Sejalan dengan filosofi CJ dalam menjalankan bisnisnya untuk berkontribusi pada perekonomian nasional, CJ akan terus menjadi perusahaan nomor 1 yang bergerak dalam bidang bisnis bioteknologi, yang terus peduli pada kelestarian lingkungan dan maju berkembang bersama Indonesia.
welcome back to the hall. Ladies and gentlemen, we hope you enjoy your lunch, and I hope that your energy has been recharged, because after this we will have our second plenary session with a theme of biodiversity for agriculture and health. Therefore, to participants who have friends that are still outside of this hall, we hope that you can contact your friends, your colleagues.
So everyone can come here and we can start.
For more than 40 years, ITS is moving forward with a vision to make an impact in the industry they serve and a mission to serve better.
Selamat siang.
ITS has core values to put professionalism, partnerships,
Selamat siang kami ucapkan kepada para peserta konferensi.
Its services around Southeast Asia countries such as Malaysia,
Selamat datang kembali di ruangan ini, dan kita akan melanjutkan
Vietnam,
agenda kita selanjutnya, yaitu
Indonesia,
sesi
Philippines,
plenary yang kedua. Kami mohonkan kepada para peserta yang
Indonesia,
memiliki kolega,
ITS
teman,
has two
kerabat
offices
yang masih
located
berada
in
di luar
Jakarta
ruangan ini,
and Surabaya.
silakan untuk dihubungi
Inside ITS Science
sehingga kita
Indonesia
bisa
office,
segera mulai.
there are facilities to support the work such as training room, and demo lab. ITS Science Indonesia also actively create or participate in various events such as webinars, seminars, product training, and exhibitions. Here are some brands that ITS Science Indonesia distributing. Products that distributed are from top and reputable brands worldwide. We are also proud to serve the needs of our customers, from governments, hospital, industrial, to universities. If you need help for your laboratory needs, contact us. ITS Science Indonesia, bridging science, inspiring possibilities.
Yes, there's someone already. Want to check? Okay.
When the genetic code of our DNA was discovered, our world changed. We had identified the biological blueprint that defines everything about who we are. From there, the technology to explore our genome evolved rapidly. Programmable DNA editors like zinc finger nucleases and TALENs could change specific genes, and RNA interference could prevent gene expression. These tools transformed us from genome explorers to genome engineers. We could not only observe, but also change genes and investigate their impacts to understand how our biological blueprint is read. The advent of CRISPR-Cas9 technology brought versatility, expanding the engineer's toolbox to include virtually any type of change, from gene silencing to custom sequence insertion to transient activation or inhibition. Anything is possible. The efficiency of these methods allows us to explore and engineer on previously unimaginable scales, studying entire genomes and gene expression in single cells. Genome engineering has even entered the clinic, initiating an exciting new era of personalized medicine. CRISPR can generate chimeric antigen receptor T cells that target cancers like leukemias and lymphomas, and gene therapies to repair damaged genes are being tested to treat diseases. Our team is here to make all your genome engineering ideas possible with expertly optimized tools to create any type of modification at any scale. We provide more than just reagents. Our scientists also partner with you, sharing their knowledge through technical support to help you take those ideas from possibility to reality. The future of genome engineering is filled with the potential to transform medicine, and we look forward to building the foundations of better health with you.
Halo Sobat Merk, apa kabar? Aku Intan dan aku akan membawakan kabar terbaru dari Merk. Merk sebagai prinsipal kini sudah terdaftar dan hadir loh sebagai penyedia dalam e-katalog LKPP. Nah, Sobat Merk bisa menemukan produk kami di etalase nasional untuk katalog peralatan laboratorium dan etalase lokal untuk katalog bahan kimia. Wah, sebuah kabar yang menggembirakan.
Ladies and gentlemen,
benar kan Sobat Merk?
we are about
Yuk,
to
kunjungi
start
website e-katalog
our
LKPP
next agenda,
dan
which is
mulai
second
berbelanja
plenary session.
sekarang.
Therefore, to all
Gimana ya
participants and
caranya untuk
speakers,
temukan
please
produk apa
kindly
saja
be
sih yang ada di e-katalog LKPP? Ingin tahu kan gimana caranya? Stay tune ya. Bye!
Once again, ladies and gentlemen, may I have your attention, please? We would like to start the second plenary session, which is the next agenda for this conference. For participants who are present here and still have friends or colleagues outside of this hall, we recommend you to contact your friends and ask them to be here as we will start our second plenary session. Ladies and gentlemen, once again, we hope you enjoy your lunch and your energy has been recharged, because next session we will be presented with presentation in the scope of biodiversity for agriculture and health from our invited speakers. And this second plenary session will be chaired by our moderator. Our moderator was a deputy for life sciences in Indonesian Institute of Sciences, famously known as LIPI, and she is now a senior researcher in Indonesia National Research Agency. And I believe everyone here already know who she is. Therefore, we would like to give the time and stage to our moderator, Professor Dr. Eni Sudarmonowati, to chair the second plenary session. Well, ladies and gentlemen, please give a round of applause to Professor Eni as she will chair the session. To Professor, time is yours.
Thank you, MC. It seems that I like to sing in here. No, well, it's always a very difficult time after lunch. Yeah, so to make people gather back to the seminar room, they chat, especially because this event maybe after a long time we are in silence because of COVID. So they're really enthusiastic to come to this event, and especially this is one of my strategy. Bali was chosen for the venue. So I hope you enjoyed your lunch. Yeah, But I also hope that you don't fall asleep during the session, because maybe you ate a lot. So I know the food were very delicious in here, even for us. Yeah. So without further ado, I would like to invite my colleagues, my friends here. They are all my friends. Professor Ilya Raskin, please come forward to the stage. Yeah. In here first, yeah. Take the board. It's not really the board, but it's the paper. Yeah.
I remember my name. Although I'm approaching the age where I start forgetting.
Okay, thank you, Ilya. And my second speaker for this session, Professor Ernawati Sinaga, also my close friend. So there will be like a reunion family. Yeah. Yeah. Okay, and the third one will be, this is a rising star, young generations, Dr. Masteria. Please come forward. Rising star, yeah. Okay, so while waiting for participants to join us here, we just start. I mean, because we cannot really wait for a long time because we already 40 minutes behind. Although we missed one speaker supposed to be here with us, Professor Heather. It's really sad we could not have her here because she's sick. So she just emailed us like last week, right? Only last week on Thursday, one day before she left for Indonesia. She got sick that she could not even talk, and quite serious illness. So, but she's very optimistic. Sorry, very optimistic that she could be on Zoom meeting today. But still, just now, Ilya got an email from her that she still cannot talk. Maybe still difficult to breathe also. So it's very sad that so we just pray for her for the speedy recovery. So it's only 3 speakers for this session, and I would like to read the CV of each speaker. So I would like to start with Professor Ilya Raskin. Hello, hello. Number 1 first. Yeah. Okay. Thank you. No, this is the second one. Yeah, okay. So Professor Ilya Raskin, I made this one. I made this slide, yeah, just to be different from other moderator, although I really didn't have time. But yeah, so I summarized. So he got the PhD from Michigan State University, MSU, in 1984. So we look much younger. No. And joined the faculty of Rutgers. That's the directly Department of Biology, since you are in Department of Plant Biology, the State University of New Jersey in 1989, and currently he works as a distinguished professor and president of Global Institute for Bioexploration, and plus other other engagements. And work research experiences more on pharmacology of plant natural products, and he's also actively involved in international bioexploration, conservation, as Professor Jim Simon already mentioned this morning, working with Professor Ilya Raskin, also this including in Africa, also in South America, Central Asia, and Indonesia, of course. Publication of 240 scientific publications and other popular press articles. Award, among others, there are many awards. So one of the 108 most cited researchers in plant and animal science, and he also got award from, is that Albert Scholl? Award for outstanding contribution to plant biology, and also Thomas Alva Edison Patent Award. And he owns 24 patents. His research interest since 30 years ago, experience in academic research in plant biology, phytochemistry, and pharmacology, and also research, industrial research in plant biotechnology. So that's about Professor Ilya Raskin. Now I'll give the floor to Professor Ilya Raskin. Oh, you're going to stand there, okay, yeah. So the time would be the same. If you can make it 25 minutes the most. Then we have each person 5 minutes per, and then we have at the end 15 minutes or more maybe for discussion. Thank you.
All right. If I can have my first slide, please. And first of all, of course, before I start with my slides, I really would love to thank Professor Sudarmanawati and Professor Sinaga. By now, I hopefully can call them dear friends and dear colleagues. It's always a pleasure for me to come to Indonesia, and there's so many faces whom I already know who look familiar, our trainees, our colleagues. Thank you so much for having me and my friends from Rutgers. It's just always wonderful. Despite the fact that it takes 36 hours of flying, the moment you land, you feel like at home. And that's just, to me, a wonderful feeling. So I will, may I have the first slide? So we'll talk a little bit today about botanicals and human health. I apologize if you've seen some of those slides already. Initially, I'll talk to you broadly about sort of what I believe is the future of plant-derived natural products, plant-derived therapeutics, show you one example of what we do in the lab, trying to promote this area, particularly in the area of functional nutrition, and then just briefly mention Gibex, but there will be a bigger Gibex talk or magic talk at the, thank you so much. I think on day after tomorrow. Okay, next one. As you of course know, people always equated plants with healing. It's been true for pretty much all ancient civilization. It's certainly true now for most Asian countries who still derive a lot of their health from natural medicines. Slightly less true now for where I live, which is rather unfortunate. But plants has been truly the foundation of our nutrition and of our health for 10,000 years, if not more, since really the dawn of civilization. Because, as we all know, they harbor this amazing array of biologically active compounds which work together, which are optimized for biological function, and which occupy very large chemical space. Next slide, please. And that is because plants are so profoundly different from animals. Because, as you know, animals really use their energy to protect themselves from all kinds of stresses by locomotion. Most of the energy we actually invest in our bodies is to support muscles, to support brain, and to support digestion. So we are not very good in spending energy on doing chemistry. Plants, on the other hand, they're anchored in one place. They're sessile by nature. They cannot run away. They cannot really yell. They cannot scream. They cannot fight back. So they do what they do so well. All they do is through chemistry. So in plants, the energy of sunlight and the water and the carbon dioxide go into the production of a vast array of natural products, or secondary metabolites as we call them, which are designed to protect the plants from all kinds of stresses they encounter. Next one, please. So, unlike animals, plants really do not have what we call chemical homeostasis. Because if you grind an animal, you'll come out with a pretty much similar mixture of primary metabolites. Those compounds are essential to maintain life. Unlike humans, plants actually contain a vast array of the secondary metabolites, which are used for what animals use their muscles, use their brain power, basically used to protect from diseases, protect from stresses, protect from ultraviolet radiation and so forth. And those compounds, very interestingly, in contrast to the pharmaceutical industry, which for the last 100 years has been basically making one pill containing one compound, plants learn that if you have one particular chemical with which you can try to fight bacteria or viruses, you're not going to succeed because resistance will be developed. So what they're really good at, and using the synergy between different compounds which work together. That's why it's so difficult to isolate a single very active component from plant, but often the extracts are actually more powerful and more potent than a single isolated compound. And this actually has been very well known, particularly in the Eastern civilizations, and that's been really foundation of many traditional medical systems. Next one. And therefore, biochemical evolution of plants is really driven, the biological evolution is driven by the biochemical evolution, so to speak. So plants possibly arrived about 3 billion years ago or so. They came from the ocean, and half of that time they spent in the ocean because they couldn't figure out how we can actually move to the dry land, because they couldn't figure out how to support their structures, how to support their different organs, until they learned how to synthesize lignin, which is this glue and which is this polymer which actually binds cellulose together. And that allowed plants to become vertical and to reach towards the sun. And that took 1.5 billion years. But once they learned how to make this lignin as a secondary metabolite, they could really reach upward and compete for sun and colonize so successfully the land. Next one. So there's a lot of interesting recent research about what are the genetic drivers for evolution of naturally products. How did plants manage to evolve all those pathways for making secondary metabolites? In the last 10 years, we've made dramatic progress on this, and what we have learned through the work of several laboratories, that the primary genetic driver for the evolution and for birth of secondary metabolic pathways is actually the gene duplication. The duplication of genes which are initially were involved in primary metabolism being something which makes compounds which are essential for survival. But plants had the ability to duplicate these genes, and the second copy became redundant and therefore acquired freedom to mutate and to evolve and give rise to enzymes with different function, and allowing plants to start this gigantic earth-wide biochemical experimentation with the compounds they can make with those spare genes, with modified spare genes and enzymes. And also intraspecific horizontal gene transfer, which is actually usually a retrotransposon, parasites, viruses, apparently can take large parts of the genetic material from very unrelated organisms and then move them to other organisms. For example, flies have acquired some plant enzymes which allow them to detoxify, sorry, it's a jet lag. Phenolic glucosides, for example, from milkweeds, and so forth. There are many examples of this horizontal gene transfer, probably mediated by viruses, retrotransposons, and so forth. Next one. Well, here is an old review which we've published a while ago, and other people did the same. Just a list of pharmaceuticals which are commonly used in modern medicine, which doctors will prescribe to you, which are derived from plants. And this is still true that about 30 to 40% of all medications prescribed today come originally from compounds discovered by plants. For 10,000 years, people have been experimenting in chewing on different plants, and they watched animals doing the same, and they really learned what works. And then chemists, who started really about 200 years ago, were able to isolate a lot of those compounds, and they found their way into basically pharmaceuticals of today. Alkaloids are by far the largest group. Those are the nitrogen-containing compounds. Terpenes and steroids are second largest, and then different phenolics also represent a reservoir for potential new pharmaceuticals. Next one. However, and this is, I know, something which for somebody who made their living and got their grants from this great notion that we're going to keep discovering novel pharmaceuticals from plants forever, things have changed, and different technologies have evolved, which really started to compete with plants as a sources of discovery of novel pharmaceuticals, or novel medicines. For 10,000 years, we experimented with plants. Chemical synthesis 120, 150 years ago, people learned actually how to make matter. We became experts in making compounds, so we could now do it independently of other living organisms. Combinatorial chemistry, target-specific computer design, where you can design molecules on the computer screen. And recently, I think, truly the tide of biologics, and you've seen it for COVID, that basically pharmaceutical industry now is shrinking their departments of chemistry, and certainly they got rid of most of the natural product-based drug discovery. What they're doing now, they're moving into the era of biologics, into transgenics, into vaccines, into the antibodies, into gene therapy. And definitely this is where medicine is moving. Frankly, it's very hard to get interest in the new medicinal plants. If you go to Pfizer and Merck, fortunately or unfortunately, they're all in New Jersey, they're all our neighbors. So there's competing technologies, and technology just evolves, they change. So what are plants good for? Next one. So I still believe, and this is sort of, you've heard this message, but I keep sort of driving it home because, again, we're lucky to be in the midst of a lot of large corporations in New Jersey. And the belief, I think, which is now being born between everybody, that the future of natural product research is going to be a little bit less in drug discovery. It will never disappear, but it will be less, but much more in consumer products, in foods, and particularly functional foods, dietary supplements, cosmetics, and personal care. These are the areas which are growing very rapidly. And frankly also, and I think somebody already mentioned it, plants are also good in producing recombinant proteins. So as long as we're moving into biologics, we can actually make many proteins in plants, and that's a very effective system. For example, transgenic TMV, tobacco mosaic virus, inoculate plants, and in a week, actually produce vast amount of therapeutic proteins. Actually, the first Ebola vaccine, which was made in the world, was made in tobacco plants. Next one. So, with functional foods, with dietary supplements, we're truly moving into the different area, the different market. It's not cure and prevention and disease, which is a definition of a pharmaceutical agent. But what we're really now doing with functional foods, we're moving in the area of prevention. Something which if you do now, you will benefit in 20 years. And it's very difficult for us as scientists to develop methods to study prevention. And yet, if we talk about functional foods, and this is what we are really talking about, and this is probably something you've seen, we believe that our lifespan may be about 120 years or so. This is what we're genetically programmed. We can live under the ideal conditions. Most of us never achieve this potential to make it that far. And we start the slow decline because of chronic diseases which creep in and basically disable us. So we have pretty miserable sort of last quarter of life. The idea that functional foods, dietary supplements of the future, the emphasis on prevention can actually change this curve, so we have a very happy, whatever, 100 or so years of life with a pretty good quality of it, and then just like leaves in the fall, rapidly senesce and drop. So this is an area which I think getting a lot of attention, and this is the area where we need young people like you to participate. Next one. And we are seeing it already in the food stores. We're seeing it in the food stores in the United States, although the food industry is very much regulated. But in the last maybe 10 years, we start seeing different labels on our food packaging. Of course, since the civilization, we were just growing calories because we needed to eat to survive. For 10,000 years of agriculture, we've been only focusing on calories, on just filling our bellies. After vitamins were discovered, well, nutritional labeling, vitamins, essential micronutrients, they become important. But now we see all those claims, cardiovascular health, normal sugar metabolism, and so forth, which is really taking us into this generation of functional foods. Next one. And this is where I see a lot of very interesting developments, because what I'm seeing that now there's a lot of scientists and there's a lot of companies which actually going to be studying the human genome, and we all have very different genomes, and we have many genomes in our body because we also have microbes in our gut and microbes in our skin and microbes in our noses, and they also contribute to how our body functions. But we can now, through meta-sequencing and omics, we can truly understand the genome, our microbiome, and develop history of our lifestyle and what our parents did, and actually started to understand what are the important
ingredients, active ingredients and foods we eat, which can actually prevent the disease. There are many companies which are now emerging, trying to analyze your body and then tell you what you want to eat. The biggest challenge to this, and this is what we don't understand, and everybody talks about it, and it's very exciting, but here is a challenge. If you really ask people to identify the compounds which we need to increase, we have technologies for CRISPR, somebody mentioned it, for transgenic approaches. We can now pretty much change any metabolite in the plant which we eat. But the big question which is facing all of us as scientists in this area is which compounds really we want to increase to prevent certain disease to which, for example, myself or my colleagues are going to be predisposed 20 years from now. This is a challenge for the functional foods, and this is, I think, a wonderful opportunity for research and research funding in the future. Next one. Just an example of what we've done in the lab. Antioxidants, of course, if you ask people what part of diet is good for you, in the US, they'll tell you antioxidants. Give me more antioxidants, we've heard they're good, we don't know really what they do, but fruits and vegetables have them. Red vegetables, red fruits are good for you because they have all antioxidants. We're going to eat more and we're going to prevent cardiovascular diseases, diabetes, and so forth, and memory loss. And yes, there is evidence that it is true, but it's clearly not such a simple answer because many of these antioxidants actually, they don't give you health because they just quench reactive oxygen species, but because they have very specific targets in the body and they all work differently. So it's a very big class of compounds. Phenolics, flavonoids is one of the groups. And what we decided to do is just to take a regular lettuce, because lettuce is the third most consumed vegetable in the world and not known for much of anything, and see whether we can increase the amount of antioxidants in regular lettuce without using transgenic approaches, because again, the regulatory environment for transgenic plants is very complex. So what we used a tissue culture, and Isabel Armas, who is here, actually been involved in this research, and so some of that actually was done by Isabel. But then, can we have the next one? So we've done the basically breeding on a petri dish. We took a lettuce plant rich in antioxidant, red lettuces, broke them into different cells, and somebody by the name of Natalia Pogrebnyak, who was a very capable cell biologist, did it. And then she broke them down in millions of cells and looked for the reddest cell on the petri dish. And from that red cell, because plants are totipotent, right? Each cell can give rise to a plant. Through this selection process, she was able to regenerate very darkly red plants. Next one. Which we call Tratke super lettuce. Next one. Let's move on. Can we have the next one, please? And when we measured, so after several cycles of tissue culture selection, we've identified this red lettuce, which has about 8 to 10% of antioxidants per dry weight, which is unheard amount for the lettuces. And we identified them using the analytical equipment we have in the lab, very high amounts of chlorogenic acid, quercetin, and anthocyanins. All those compounds are antioxidants. All of them have been essential for, been reported to be beneficial for human health, although not through thorough clinical trials. But the reason, of course, we look for red, because red is anthocyanin. It's an end product of the long phenylpropanoid biosynthetic pathways in plant, which makes lots of natural products. And so that was the last step. That's the color. So that's why we look for the reddest possible lettuce. Next one. So again, this is just a comparison, and those super lettuces we developed, the red ones have 7 to 8 grams of antioxidants per gram fresh weight, compared to blueberry, which is a golden standard, which has only 3. Next one. And then I think this is what Isabel has done when she was in the lab with some of the colleagues. She planted a lot of red seeds, really dark red lettuce, and identified certain green plants which appeared with a certain frequency among this population of darkly red seedlings. And when we looked at them, they look chimeric to us. So they had these little red sectors in them. And then we did the biochemical analysis, next slide, and genetic analysis, that although the plant was green, because really lettuce growers told us we don't want red lettuces. We want to have green lettuces because that's what people buy. So when we looked at those lettuces, which were generated from the red ones, we actually found very high levels of antioxidants in them as well. And when we genetically, Shanat Gurdon, who was a postdoc in the lab, when he actually did the genetic analysis, he's seen that the gene responsible for anthocyanidin synthase, which is the color-producing gene, basically was totally muted. It was blocked. There was no expression of its RNA in the plant. So we blocked the last step of polyphenol biosynthesis. Next one. And then he did the genetic analysis, which was published a couple of years ago in Plant Physiology. We observed that the large transposable element was inserted inside the gene, therefore it was basically a nonsense mutation and there's no synthesis of color. And those antioxidants, like quercetin and chlorogenic acid, they accumulated because there was a biosynthetic block on the pathway. And so we still have now green lettuce with about 7 to 8% of antioxidants. Next one. 2 more green high polyphenol mutants came as a result of AMS mutagenesis. Again, with a complete genetic analysis, we know where the mutations were. Those are produced through EMS mutagenesis. One had a very high level of camphorol, which has been documented to have anti-cancer, anti-diabetic effects. And the second one, may I have the next one? Well, just let's move on. And one which has about 4 to 5% of naringenin, which is a compound reported to improve memory and learning. Okay, so all of that was genetically characterized. We understand these genes were mutated. So we actually were able, thanks to this original work from Natasha and Isabel and Shanad, we were able to come up with 3 different lettuce varieties which were enriched, dramatically enriched, with some health-promoting antioxidants. Next one. And actually there are several companies which are now experimenting with those varieties, and in some stores you can buy them. And we hope that soon, you'll be able to see them on the supermarket shelves. Actually, this is from ShopRite. Of course, I would like somebody to do serious clinical trials with those varieties, because we don't really understand how the consumption of very high levels of those antioxidant compounds, how can it actually benefit or human health? Is there any toxicology involved? So we wish there would be more research done, which we haven't done in the lab. But this is a way to at least use some of the modern technologies to produce plants with higher levels of potentially beneficial compounds. Okay, and then last talk, of course, and this is something which is very much, as part of this, related to Indonesia. You probably already heard about GIBEX, Global Institute of Bioexploration. And day after tomorrow, 2 of my colleagues here will have a workshop on GIBEX-related technologies. This is something which was established in 2003. We have the next one. With a mission of truly give the world a novel, ethical, sustainable, and cost-effective approach to bioexploration and actual product research. Because what existed before is what is called, obviously, bioprospecting, and we have been doing quite a bit of that, and we've been actually getting significant amount of money from USNH to run some of the bioexploration, bioprospecting programs throughout the world. This is how we originally met Dr. Satorov in Tajikistan. But that method of going to the tropical country, developing countries, collecting plants, drying them, bringing them back to the US or Europe, testing them, really has a lot of issues associated with the Biodiversity Convention, with Rio Convention, with Nagoya Protocol, and ultimately got a bad reputation, almost like biopiracy. So what we said, let's just change completely the way we do this so-called bioexploration, bioprospecting. Rather than taking materials out of the country, let's develop simple, cost-effective, sustainable methods which can be used inside the country by people with very little training, very little expenses, very little money, to at least validate and identify some potentially valuable pharmacological activities from their biodiversity. Let's reverse the flow, not take anything out of the country, but give that country the tools, if they need it, or or help this country to develop their own biodiversity for potential economic benefits. Next one. So on top of just the ways to collect and novel ways to collect that, we also have developed a variety of field deployable, simple pharmacological assays, which can basically from the trunk of the car provide you some information about potential biological activities. So the two really components of GIBEX, and I think this is what day after tomorrow you will see, we've developed rapid metabolome and genome extraction and storage technology, which truly allows in a very short period of time to collect the metabolome and genome of plant species, and it's not only relates to plant species. You can do it with almost anything, with marine organisms, with mammals, with insects, which I think is the last frontier, with soil microbes, with many things. And on top of this technology of rapidly collecting the metabolome for studies and for assaying, also this formatted a number of different assays which are compatible sort of with the collection technology. Next one. Since 2003, we actually taught GIBEX workshops in, I believe, 16 or 18 different countries in the world. It's been an extremely rewarding activity for all of us. And many of our Rutgers students participated in this, actually countries in red, and where we actually ran the workshops. Other countries joined the GIBEX programs, and we shared the information with them without the workshop. Next one. And this is Sarah Skubel, who just graduated with her PhD from the lab. Her teaching actually how to use the collection, the RAMES part of this GIBEX technology in the field, Kruger National Park, where we had run some very successful GIBEX-related programs with local rangers and local scientists. A couple of good papers came out of it after that. Next one. So this is how the samples look like. What we do, we rapidly grind the plant in the field. Only takes 5 minutes. We have all the tools, and we brought them with us, and you will be able to learn some of that. And when we impregnate, we basically spot the extract, which we produce right in the field. It's spotted on the fiberglass discs, 1 centimeter in diameter, which are being rapidly dried. So now you have the disc which contains 100 microliters of plant extract. Because of a very rapid drying, which occurs naturally, or you can force it with a fan, you have the replicate, about 20 from each plant, 20 discs, which now basically contain those glass fiber discs impregnated with metabolome of that particular plant. And this is extremely effective and cost effective, and we've demonstrated that actually these compounds are very stable, and they can be, let's grab the next one, and they can be then eluted. But on top of that, I mentioned that we're also interested in genome, because I think collecting genomes is the next way of validating the botanical identity. We used to collect herbarium vouchers. That is very complicated, very laborsome, and storage is very difficult. But now, if you just punch a disc off a leaf and put it in a plastic bag with a silica gel so you rapidly dry it, you basically have a sample of plant genome for posterity. You can put it in a freezer and store it forever, next to this microfiber discs, which contain the metabolome of the same plant. And this is your voucher. This is your genetic voucher, because you can always take that disc, the dry disc, extract DNA from that 100 years from now, and truly establish whether the plant was correctly identified, or if this plant disappears from the surface of the earth, at least you have the genomic record and you have the metabolomic record of a particular plant. And on top of that, what you can do functionally, you can take those discs and you can use them in microtiter plates, a variety of different assays. This is just antibacterial assays, which I think we will teach. 24 microtiter well plate. The discs are put on the bottom. As the bacterial inoculum in the field, we use human saliva, and we use a colorimetric reagent called MTA, which is normally blue, but if cells are alive, if bacterial cells are alive, the solution becomes yellow. So you just see how beautifully it works. You can do it in the field 24 hours. Garlic extract works. And you see discs like on the bottom of each well. You just drop it and then you put a little bit of saliva there and you have your assay right there in the field. So again, we were very fortunate to get real support to develop this technology, GIBEX technology, and hopefully for the benefit of Indonesia. And Indonesia is now the first country which actually taking this methodology and using it to create the first in the world Metabolome and Genome Innovation Conservation Center, MAGIC, with the help of UNAS and BRIN and many other organizations are joining, actually using the RAMES technology to collect metabolome from the plants. And we are very happy to report that I think 500 different Indonesian plant species have been already collected and stored. And those discs are available either for functional evaluation or through, if you're interested in doing metabolome analysis, natural product analysis, they can be easily eluted and run through your analytical technique, whatever you have in the lab, to get the, identify the compounds in them. And of course, we also need to acknowledge the generous funding from US National Institutes of Health, which allowed us to establish the Center for Botanicals and Chronic Diseases, CBCD. Initially, we worked with Tajikistan for a long time with Professor Satorov, but we were very happy that in 2002, Indonesia joined the center, and we have our trainees. It's actually a research training program. I believe we have 28 trainees in both countries now. It's absolutely a delightful thing, and I know that I think 9 came to this conference from Jakarta, 1 from Tajikistan. We couldn't bring everybody, but this has been an extremely fruitful and productive collaboration for us. And we're really hoping that Indonesia, next slide please.
I'm sorry to remind you, I mean, maybe you
That's
can
the
speed
end.
up. Okay,
That's
thank you.
the last. So it's been amazingly rewarding, and Indonesia now is becoming, I think, the first country, and we're already getting into some other countries, but I think Indonesia leads, become the first country to create the MAGIC library, which preserve the metabolome and genome of rare, endangered, and endemic species for Indonesia. And soon we hope to add the genomic samples to this, so we have samples of metabolome and genome. They can be shared with any scientist, because from 2 grams of tissue, just 2 grams of plant tissue from the leaf, we get about 20 discs, and then we can punch 20 discs of leaves. It's non-destructive, you don't want to kill the plant, but yet you have a record. You have a record to biodiversity, and that record can be used for functional characterization, and that record can be used for future research on natural products and assaying the activity. So thank you very much. This is our wonderful trainees. This is, I think, last year when we've done the second workshop, second GIBEX workshop. And of course, my colleague, Dr. Dushenkov, is here also, who has been invaluable in running this program, and he already gave his talk on biodiversity. So I don't think I have any more slides. Yeah, thank you, should be the last one. So thank you very much. It's a joy to be here. Sorry for the jet lag. And I know that I should not give a talk after, flying for 36 hours and after lunch, but you always learn. You always learn on this one. Thank you very much.
Thank you, Ilya. Thank you, Professor Raskin. It's very interesting, yeah. So I hope we still have time for discussion later on. So next to the second speaker, Professor Enawati Sinaga, she will deliver a talk on Indonesia underutilized fruit as the potential source for functional food for the management of metabolic syndrome associated diseases. So this one's still related. Before Professor Raskin also talk about functional food. What's wrong with this? Okay, that's the one. So yeah, can you show the CV, short CV of Ibu Erna? Yeah, just a little bit. Sorry, Slavik. Thank you so much. Yeah, Ibu Erna is from UNAS, Universitas Nasional. She's currently the Vice Rector for Research, Community Services and Collaboration, and as Director of Center for Medicinal Plants Research at Universitas Nasional Jakarta. She did the PhD from Department of Chemistry, University of Indonesia, Universitas Indonesia, with research emphasis on molecular biology of drug delivery, conducted as joint research with Department on Pharmaceutical Chemistry, University of Kansas in the States, and obtained MSc degree from the same university from Universitas Indonesia in Jakarta. She has a patent on synthetic peptides, and her research interest focuses on research related to botanical and chronic diseases, in collaboration also one of them with Rutgers University. So without further ado, please Bu Erna, time is yours. Thank you. Careful with your bag.
Thank you very much, Bu Eni. Good afternoon, good day, ladies and gentlemen. I am extremely happy to be here to share what we are working, me and my colleague and my students did in UNAS and is going now about the functional food from fruits. So first of all, I would like to thank the committee, especially Ibu Professor Eni Sudarmono, for invite me to be invited speaker here, so my students can see me present what they are working, what they were working before this time. Ladies and gentlemen, next slide please. We know that Indonesia is the second mega biodiversity country. They said like that. And some of Indonesian scientists said that maybe Indonesia is the first mega biodiversity country if our resources in marine, like what Pak Master Ria will tell us this afternoon, also measured. But it's okay to be the second mega biodiversity country because it's already a huge number of biodiversity in Indonesia. Besides the famous orangutans and the rhinos and the tigers, the Sumatran tigers, we also have many kind of fruits in our forests. Come back please. Okay. There are more than 300 species of fruits in Indonesia, either cultivated or wild fruits. Unfortunately, most of them, about 250 species, are growing wild. Only 50 to 60 species are cultivated. Why we not domesticated or cultivated those fruits? Because we don't know yet the usefulness, the benefit of the fruits. Next slide, please. The fruits that not used optimally, we usually said underutilized fruits. That is the fruits which are neither grown commercially on a large scale nor traded widely. The underutilized fruits are those which have minor market value, not widely grown in the field, and are rarely found in the market. They have been linked with the cultural heritage of the locality, traditional crops in localized areas, and neglected. Next, please. We in Indonesia have a huge number of underutilized fruits. I just want to show you some of those fruits. For example, this Anacardiaceae, the Mangifera casturi, is the endemic mango of Kalimantan. Mangifera kemanga. I don't know whether it is endemic or not, but we have many varieties of Mangifera kemanga here in Indonesia. And we also have Mangifera pajang. Next, please. And this is the other Anacardiaceae. We are working on the Boya macrophylla, or we call it Ramania. Next, please. And Phyllantaceae family, fruits from Phyllantaceae family are many kind of them in Kalimantan and Sumatra forest. Some of them are here, the Bacorea lanceolata, and other Bacorea. Next, please. Next, please. This is also the Phyllantaceae, the Bacorea. We have many of them here. Next, please. This is the Clusiaceae. It's belong to this same family with the queen of the fruit, the mangosteen. That is Garcinia mangostana. This is the wild mangosteen, Garcinia forbacei and Garcinia, I can see, Garcinia species. Next, please. We also have Asam glugur, or we call, the name is Garcinia. I cannot see here from here. Sorry, but this is the Clusiaceae. I just want to show you that we have so many Clusiaceae here that are underutilized. Next, please. This is the Moraceae. It's the sister or brother of the jackfruit. Look at the orange one. It's very delicious. And even though it's white, but it's sweet and very delicious. They grow in the forest of Kalimantan. Next, please. And this is a very, very delicious fruit. I made these fruits when I was wandering in the forest of Kalimantan, and they call it is fruits from heaven, because its grow is so high in the top of other trees, but it's very sweet and milky. It's so good. Next, please. And this is the striking pink color Lycia garcinia or kalangkala. Next, please. These fruits are good resource atau good source for functional food, because they have good taste and brilliant color, and they also have polyphenol or phenolic compounds and flavonoids. And also they have fiber, dietary fiber. So we thought that this is a good source for functional food that Professor Ilya just told us, that if we cannot consume medicine or drugs, it's good. Just consume the food, the functional food. Because the functional food is the food which is not only beneficial for nutrition, but also have a potentially beneficial effect on health when consumed as part of a varied diet on regular basis. Shortly, functional foods are foods that provide a specific health benefit in addition to the benefit of general macro and micronutrients. Next, please. This is a more complete definition of functional food or functional beverage. We can just, there are so many definitions about the functional food and functional beverage. We can search it in the internet. Next, please. This is what I tell you that why fruits are suitable for functional beverage, because they have high polyphenols and flavonoids. They have high dietary fiber, they have good taste, excellent flavor, and also attractive color. Next, please. Now we look a moment at the metabolic syndrome. We know that metabolic syndrome is a syndrome that have 3 among 5 criteria. Next, please. No, come back, please. If we have 3 criteria from 5, hypertension, insulin resistance, high blood glucose, obesity, and hypercholesteremia, that person can have metabolic syndrome. So be careful. If you have 3 of these criteria, so you have to be very careful because you are in risk of getting the degenerative diseases like cardiovascular diseases, diabetes, neuromuscular diseases, etc. Next, please. Yeah. So in the past 5 years, we in Universitas Nasional, me and my colleague and our student, investigate or analyze some fruits, some underutilized fruits of Indonesia, like Rhodomyrtus tomentosa, Boya macrophylla, Ziziphus mauritiana, etc., and look for their antioxidant first. Because as Professor Ilya said, that one characteristic of natural products that have medicinal value is the antioxidant activity. So we look for the fruits that have high antioxidant activity and have polyphenols and flavonoid content. Next, please. We investigate at least until now 3 species of fruit juice of 3 species of fruits. That is Rhodomyrtus tomentosa. Next, please. Boya macrophylla, and the other one is Ziziphus mauritiana. Rhodomyrtus tomentosa fruit juice has a very attractive color, and it's very delicious. This is the fruits you can see here. It's very high attractive colors, and also from its color, we know that they have the anthocyanin or anthocyanidin because they have a color like this, red and blue, etc. And the fruit juice of this Rhodomyrtus tomentosa is for me, it's very attractive. And I think, so we give this fruit juice to rats fed with high fat, high cholesterol diet. We would like to see if the juice, the consumption or the supplementation of the juice can block or can inhibit the hypercholesteremia and also the atherosclerosis formation. Next, please. This is the effect of the Rhodomyrtus tomentosa fruit juice supplementation on lipid serum level. We can see here that the total triglyceride is very much depressed in the rats consumed or supplemented with the Rhodomyrtus tomentosa fruit juice. The red one is the total triglyceride of the rats fed with high fat, high cholesterol diet and give nothing. No juice and no other medicine. The green one is the rats fed with the high cholesterol and high fat diet, but give simvastatin. We know simvastatin is anti-cholesteremic drugs that can block or can inhibit the formation of cholesterol in the cell. We can see here that the effect of the Rhodomyrtus tomentosa fruit juice is almost similar. Almost similar, even it's in the high, the yellow one is the rest that is given the juice in high dose. We can see here that the high dose supplementation of Rhodomyrtus tomentosa fruit juice have the same effect as simvastatin. So it's very promising.
Bu Erna, your time is only 7 minutes left.
Thank you, Ibu. Next, please. Next. This is the effect of Rhodomyrtus tomentosa fruit juice supplementation on the lipid or serum. Next, please. I just want to show you here. This is the last, Bu Eni. This is the effect of the supplementation of the fruit juice on atherosclerosis formation. We can see here the first one at above, it's the healthy rats aortic wall, and the second one is the aortic wall of the rats fed with high fat diet. The third is the aortic wall of the simvastatin rats, and in the below is we just see the last. The last is the aortic wall of the rats that give the Rhodomyrtus tomentosa fruit juice in high dose. Next, please. Yeah, please at the end. I would like to give the conclusion. Please move forward. Next. We do the same thing with this Boya macrophylla fruit juice and give similar effect. Next, please. To the conclusion, please. In conclusion, we can state that Indonesia has a large number of underutilized fruit that should be investigated to reveal their potential as functional food for management of metabolic syndromes. I hope that we can get many friends, many colleagues to work with us to do this, because we know that Indonesia has a huge number of underutilized fruits. Thank you very much for your kind attention.
Thank you, Prof. Erna. I've been counting how many times that Prof. Erna mentioned that this fruit is very delicious. I don't believe it until I really taste it. Sorry. Okay, one day you have to bring it to me from Kalimantan. Okay, so it's very interesting presentation. So now we're going to move to the next one, the last one. Will be delivered by Dr. Masteria Yunofil Saputra. In Indonesia, Putra means a man. So if someone named Putra or Putri is women, so you can tell from the name. He will deliver a talk, drug discovery and development for Indonesian marine organism. So from terrestrial, now we're going to jump into the sea. So this is why I invited Dr. Masteria, because I know that he's been doing research on drug discovery in marine, so that our conference will be covering all different spectrum from terrestrial, marine, and plants, animal, microorganisms. So yeah, I would like to read a very brief CV of Dr. Masteria. So as I mentioned earlier, he's the rising star at BRIN also, of BRIN. Now he's the director of Research Center for Vaccine and Drug Discovery, right? Yeah, because all new names, yeah. Sorry, can you enlarge a bit because this one is too small? Okay, thanks. So from BRIN, yeah, from National Research and Innovation Agency, and he's got the PhD in 2012 from Italy, Marine Biotechnology, and also for the master degree also from Italy, is that Marcia or how to pronounce it?
Marche.
Oh, Marche, sorry, Marche Polytechnic University. And also he did postdoctoral in Florida, in Faculty of Pharmacy, Department of Medicinal Chemistry in Florida. And he's now, yeah, I mentioned earlier, he's head of Research Center for Vaccine and Drugs, a research organization for health at BRIN. And yes, I mentioned already also he did several postdoctoral program. And one of the things that I should mention of the award, he's also got Fulbright scholarship, program. So it's just like myself as a visiting professor, but Dr. Masteria as the postdoctoral fellowship. What university this one? What university?
University of Florida.
Oh, University of Florida also. Back in 2018, and his research interests marine natural products and drug discovery. The award, one of them is actually from the President of Indonesia. This is the same award also I got, Satya Lancana Wirakarya, for your achievement. So we have a lot in common, Pak Masteria. You are my younger generation.
I learned a lot from you, Prof.
Thank you. So the time is yours. Yeah.
Thank you very much from Prof. Eni for nice introductions. And I would like to thanks to all of the organizing committee and local committee from Bali that invited me for this nice meeting today. So, So today, I think in the first presentation and the second presentation, we already talked about terrestrial, and this is my last time discuss about the marine organisms. So next, can I use this for up and down? Okay. Okay, Prof. Eni already give the introduction from me. So if you have any question about my presentation or my research, maybe you can send me email or send me WhatsApp to discuss together. So I think much better from you. Okay. So I already mentioned before my presentation about drug discovery and development from Indonesian marine organism. So it's almost I think 11 years for me to research about marine natural product from Indonesia. So mostly my work is marine invertebrates, but I work also with microorganisms. So, but today I will present about the marine invertebrate, particularly about soft coral, tunicates, and sponge. You already watched the movie about sponge, I think, SpongeBob, I think, then I will present you about the sponge today. So, managerial introduction, maybe the first I will introduce you about why the ocean is very important for marine natural to drug discovery and development right now. And my second outline of my presentation about soft coral and tunicates and sponge. So, yeah, next please. So, why we call it displaced Earth? So, I think because we are more than 70% of Earth's surface is the oceans. So now marine organisms contain 2.2 million organisms. So as you know that coral reef, less than 2% of ocean floor. And coral reef are the rich biodiversity because they are home to 25% of marine species. So if you're able to diving or snorkeling, I think in 0 meter or 10 meter, until 10 meter you can see soft coral, you can see sponge, and you can see tunicates, and you can see some of others organisms. But marine natural products, not like terrestrial plants, natural product, because terrestrial plants, natural product, Prof. Ilyas already mentioned, almost 10,000 years. But marine natural product is developing is 1965 or 1916s. So only some, I think, 63 years old for research about marine natural products. Next. So why we call it Indonesian biodiversity? So as I'm a chemistry and also biologist, because my PhD and my master about marine biotechnology, so combine about biology and chemistry. So in terms of the fission of biologies, we also looking about diatom, about organisms, microalgae, fishes, soft coral, algae, gorgonian, mollusca, tunicates, and sponge. Next. But in terms of the chemicals, so the fission of the chemicals or the fission of pharmacies, they are looking inside of the marine organisms. So we are looking the compounds that potential for drug discovery or potential like a functional food that Prof. Ernawati mentioned before. So what of our point of view is looking the chemical compounds that can be developing drug discovery. Next. So as you see, this is the story of marine natural product. So from 2012 and 2017, there are increasing number of the new compounds. So from the marine natural product. So as you see here, the invertebrates 2012 is the number ones. But in 2017, is their decrease to be the microorganism is number one. So that's why, because mostly the marine organisms is living together or be symbiotic together. So for example, sponges, they are living or they eat marine organism. They eat bacteria. So for the growing. So that's why people right now looking the marine bacteria more interest than the sponge, because they are also produce the same organisms with the marine sponge or the marine soft coral or marine tunicates. Right now we have the 8th marine natural product already approved by FDA or EMA, and mostly the 8th, I think 6 compounds here is for the anti-cancer. So the first compound, for example, is the trabectedin, FDA approved, and for the anti-cancer, and brentuximab vedotin is FDA approved 2012, and eribulin mestilate FDA approved for 2011. And this also, right until now, we still use this compound for anti-cancer, and I think it's very expensive right now, these compounds, because this is the third line of the anti-cancer for patient in the hospitals. Next. So I mentioned you before why we interested in marine natural product for anti-cancer. So I see here marine compounds than others disease. So mostly when we do the isolation of the compounds from marine natural products, we tested the cancer and saw the activity for cytotoxics. I will explain to you later in my next presentation why. And you see here that from publication from Montasser and Lutz, they have mentioned about 1.8% extract from animals, marine animals, as saw anti-cancer activity than terrestrial pen. So terrestrial pen only 0.4%. So that's why most people working with marine natural product, they tested the first thing to do for anti-cancer. Next. This is why the marine natural product is very interesting for the anti-cancer. See here, they are competition, where sponge and soft coral and tunicates, they cannot walk or they cannot run. They are static, sessile organisms. So if they are growing, they have to be competition each other. So they produce the chemical defense or the chemical compound that will be toxic from organism, the side organisms. So here is this. Competition of environment server, defense attack signaling. So they do the defense themselves against predators, then prevent other animals from moving into their space. So the chemical defense they use to prevent animal or from to harmful these organisms, and find a match also and protect themselves from infection. So from the exposure, for example, as I mentioned before, so many bacteria and so many microalgae in the ocean that will be infection to the marine invertebrates. Next. So why marine natural products or marine organisms very interesting? So I see here it is the presentation from one of the company from Amar. So they are developing the first compound for the anti-cancer. So you see here's the marine organisms. So they have wide range of chemical defense. It means we can use these compounds to anti-cancer, anti-inflammatory or neuroprotection, everything. But the problem is the industrial supply. Why I said the industrial supply is the problem, because sometimes we isolated the compound around 1 milligram, 2 milligram. So it's very difficult to do the in vivo and to develop for the pharmaceutical company. And other people thinking about the microorganisms. But the microorganisms is very easy to develop because abundant industrial supplies. But the problem is more than 99% non-cultivables. So we can only 1% cultivable of marine bacteria. That's the problem. So if you are isolated actinobacteria or bacteria from marine organism in the same area, you can find the same species of bacteria. So, and the other technology right now, biosynthesis, environmental DNA or metagenomics. So directly to the genes. If you get to the genes, you can very easy to develop of these compounds. But the problem is the heterogeneous expressions. It's very difficult. We need, I think, 5 years or 10 years to get the heterogeneous expression, because very difficult, and not the people will be with experience to do that. And next, so what the industrial supplies do to do the marine natural product? So, for example, here the sample that I mentioned to you before that already for anti-cancer trabectedin. So trabectedin is the anti-cancer compound. So they do the semi-synthesis. So the skeleton of the trabectedin is almost the same with safrasin B, so produced by Pseudomonas. So they do the fermentation of the Pseudomonas, and after that they isolated the safrasin B, and the next they do the semi-synthesis with 18 steps. So more 18 step, the industry scale is very good, and the range of the price is, I think, will be good for industry. Next. So what we do for discovery platform marine natural products. So I think the first thing is expedition. Indonesian, more times, I think Indonesia is the biggest country, I think, for the oceans. So that Prof. Ramartya mentioned before, if we combine with terrestrial and marine, we are the first country for the biodiversity in the world. And I think mostly marine biodiversity in the world already in Indonesia, because we are in the coral triangle positions, the coral triangle position between Philippines, Malaysia, Papua New Guinea, and Indonesia, and almost 75% of the marine organism in this area. So the first thing that we need to do is the extraction, expedition, collection, and marine organism. The same with the terrestrial plants. So, and because the marine organisms have the symbiotic and have living together with microbiology or bacteria and fungi, I think they produce the same compounds also. So we need to also to do the isolation of the bacteria or fungi. And cell biology, if we have to interesting with anti-tumoral screening, and if we have if they are active, like for example for breast cancer, for colon cancer, and so on. So we have to understand the mechanism of actions. And after that, if the mechanism of action is new, we can see the synthesis process and analytical process of this compound. Because right now, for example, in FDA, that maybe Prof. Ilyas read little bit more now than me, FDA is right now not approvals for the cancer drugs if there are the same mechanism of action with the compound already approval by FDA. So they have two different mechanism of actions. So if we got all of things for pharmaceutical developments and in vivo analysis and human trial and data analysis. So this is the discovery platform for marine natural products. Next. And we begin with Indonesian marine biodiversity that I mentioned you before that we are the biggest archipelagic country in the world. So we have 5 biggest mainland, the biggest islands in Indonesia. So we have, I think, more than 75% marine organism in the world. Next. So the first compound that isolated from Indonesian marine organism 1988. So I think 25 years ago. So difference with the terrestrial plants. I think terrestrial plants are people are working in Indonesia since the colonialisms of Netherlands, Dutch colonialisms. So, but from the Indonesian marine organism 25 years ago. So the first compound, low limalidae. Low limalidae got the clinical trial first once, but failed for the clinical trial because of the very toxicity. This compound isolated by the group from University of California. Next. So that's what I said, very interesting from Indonesian marine organism. We always isolated, sometimes the compound that we isolated, we give the name, the area that we isolated. For example, we isolated from Manado peroxide, B, so Bitungolidae, Lembehine, Biaketidae, so Papua Namidae, Barangimidae, and Swinholidae. These all of the compounds that are already isolated in Indonesia and they have the potential for example, for anti-cancer, antibacterial, and anti-inflammatory. Next. So this is one of the review of my team. I think Mbak Anggia also here, also the author of this review. So we make the review about the Indonesian Marine Natural Product 2021. So you can see here the soft coral, sponge, and tunicate. So Acanthospora is why, and Aptos is the most abundant of marine sponge that are already collected in Indonesia. That's not because they are very abundant in Indonesia, but it's very easy to collect. So why I mentioned to you, why I said very easy to collect it, because the Aptos in Acanthospora is the sponge that we can able to collect it by snorkeling, because they are living in 3, I think 5 meters you can collect it. And this why people very easy to collect this sponge. And you can see here the most compound that already isolated from Indonesian marine organism is sponge, alkaloid. Okay, the second about terpenoid and polyketide and peptide. Okay, next. So this is our R&D marine natural products platform in BRIN. So as I mentioned you before, so we collected marine microorganisms and we collected marine microorganisms also, and we make the large collection of compounds with potentially significance for pharmaceutical benefits, and we also looking for the library of microorganisms. Next. So this is my first compound 10 years ago. So this is a steroid from the soft coral Sinularia. So we isolated 8 new steroid from this Sinularia, and this is very interesting because they have the cyclopropanol ring here, and number 7 here we call it gorgosterol. If you isolated marine soft coral, Sinularia, Lobovitum, or Sarcovitum, you can isolate it, this gorgosterol. The gorgosterol is the typical steroid compound from soft coral. Next. And we tested all of the compound with farnesoid activated receptor. Farnesoid activated receptor is the strategy for the treatment of cholestasis. Indonesia, we call it batu empedu in Indonesia. And this compound is the gorgosterol here, so they antagonize for the phaneroid activated receptor. So this is very interesting for the treatment cholestasis later. But the problem is the synthesis of this compound is very difficult because they have cyclopropanol ring here. But we're trying to do some semi-synthesis of this compound, and we thought the cyclopropanol ring is the antagonist, the FX-mediated transcriptions. Next.
Masteria, you only have 7 minutes left.
Oh, 7, okay. And this is all of the compounds from soft coral sinuria also. So we selected 70 compounds here. Next. So we tested also for anti-inflammatory, and you can see here sinularicidea and seborrheicidea, so the anti-inflammatory. Next. So we also isolated from sarcopitoxidae and sarcopine from sarcophyton, and we tested the TRPF3. TRPF3 is the ion channels that's important for neuroprotection, and NF-kappa B for anti-inflammatory. So sarcopitoxidae D is the major compound from sarcophyton. If you isolated this compound, if you working with sarcophyton almost 100%, if you do the fractionation with N-hexane, you can get the sarcopitoxidae. And the sarcopitoxidae and sarcopine is show the potency for TRPF3 and F-kappa B for anti-inflammatory. Next. So this is from Tunicat. So Polycarpa aurata is very abundant in Indonesia. So we isolated polyaurine A and polyaurine B. So we tested also from the in vitro of Schistoma mansoni. So polyaurine A, so have the potential impair X production in vitro Schistoma mansoni. Next. So this is sponge. So marine sponge Lichetta. So Lichetta is lichetamol A and lichetamol B if the major compound from sponge of Lichetta. And these compounds show the 6% of the organic extract and also have potential for antimycobacterium. But from lichetamol we tested with TRPA1 and TRPM8 channel. So this is the ion channel for itchy, cold, and pain also. Next. I think we need to do next. This is the only synthesize. Next. So we do the synthesis with lichetamol A. No. I think go. No. Up. Okay. So we tested the TRPA1. So lichetamol A and lichetamol B a potential for neuroprotections also. Next. This is from the sponge. So we selected from Escherichia, Ceribunamide, and Halorchianamide. So Ceribunamide because we collected from Seribu Island or thousands islands. So Seribu in thousands in Indonesia is Seribu. So we give the name Ceribunamide. So we collected from all of this compound from all of this organism from Seribu Islands. So we collected also from the Chelonaplicilla. So Spongidae diploid 1 and Spongidae diploid 2. We tested with human tumor cell line, but only Spongidae diploid 1 showed the moderate cytotoxic. Next. And I think this is the last one. So we isolated also some of the alkaloid compound from Chelonaplicilla. So we tested also from anti-cancer, but all of the compounds show no significant activity. Next. So I think I just jumped to the conclusion. So Indonesian marine biodiversity have potential for drug discovery and development. So sponge still the major source of the marine natural product in Indonesia, and soft coral and tunicates still lower than sponge, but we need to explore to get the active compounds. Next. Thank you. So this is all of the collaboration for this work. So we got more, I think 70% of our grant from the Kormap CTA from World Bank, and we like to thank you for Ministry KLHK for the permit for collected the sample in the Marine National Park, and also bring my institution. Thank you very much.
Thank you, Dr. Masteria. Very interesting talk. So now we come to the discussion. Today we already heard research talk or integrated talk in Kalimantan from Professor Kamita Kahara and then from Professor Erna also Ernawati. So later this afternoon it will be from Professor Erin Vogel, maybe partly also in Kalimantan. So Kalimantan is very high diversity, it has high diversity, but that's for plants, but for fauna, mostly the high endemicism. Tomorrow I'll give in my talk in Papua and in Sulawesi. So Indonesia has, different diversity, richness, endemicism. So so now you've heard already from 1 hour plus, 1 hour and a half of this session. We heard that like Professor Ilya Raskin, he and his team has RSL. He said it's Rutgers superlattice, but maybe can be also Raskin superlattice. Well, yeah. Yeah. Yes, so, and then Bu Erna with very interesting, colorful fruits also from Kalimantan, and last from Dr. Masteria. So now I open for discussion. So please raise your hand if you have questions, because we have like 25 minutes, because the next session would be only one person giving the talk, because I mentioned earlier also the day after Professor Heather contacted us that she's not well, there are two speakers also from US, a couple, Professor Philip and Dr. Paula Brantley, also contacted us that they could not make it just one day before they left also, because of the death of their family member. So we have time, about 25 or 25 minutes or so. So please have discussion. I think the idea also, the session, we can discuss more, not just listening one-way communication. Please raise your hand. Or maybe from later on I will give the chance, opportunity for the online audience. Yes, Ibu? Yeah, please give the mic to her. You can move forward. You have to stand in front, to be seen by the online audience. It's over there, yeah. Should be over there. Thank you.
Okay, good afternoon. My name is Nadira from Merck Life Science. Actually, this question I have for Professor Ilya Raskin. So the problem for developing countries is the cost for healthy foods. Sometimes nutritious vegetables are more costly than junk food. Maybe you know about this. The idea of enhanced vegetables is great solution for the future, but cost-wise, how do you think the researcher and even for the commercials can make this nutritious food more affordable for the people, and especially for the people in the developing country? Thank you.
Thank you, Bu Nadira. Can I just collect other questions first? Oh, over there too. Okay, Pak Made, Ibu. Yeah, okay, you just mention your name and then come forward. Yeah. Yes, please.
This very interesting result, from three speakers. But I also want to learn more about the underutilized fruit that Prof. Erna was mentioned. That there's many. We have many biodiversity and also the fruit that can consume, but I just looked at the fruit is really spread the country. Many places have the fruit. Sometimes the fruit is not cultivated by the people. So, did you ever think how to educate the local people how to sustain the fruit, because the fruit is very potential for the medicine. Thank you.
May I take your name, please? Name and affiliation?
I'm Astri from the Universitas Jenderal Sudirman. Thank you.
Okay, one more, Prof. Made. Oh, Ketut. Sorry, sorry. So that means you are the fourth children, right? Ketut?
Yes.
Yeah. So yeah, for in Bali, they're already taught from the past already family planning, not more than 4. I mean, not more than 4. Number 1 is named, I don't know, Wayan or something. Number 1.
Yeah, Wayan,
Wayan,
or
number
Putu.
2.
Yeah, Putu Made.
And then Made.
And then
Number
Nyoman.
4 is Ketut or Nyoman.
Yeah.
And then after you have another one after 4, back to number 1 again, the name. Yeah, so that's you can find many Ketut, many Wayan in here, many Putu in Bali. So thank you.
Yes, thank you, Prof. Eni. But Ketut will be endangered, because
because the family planning, our government now
our
not more
government
than
is
2.
successful in Bali, something like that, because there are family just get two children, something like that. So there no next future, no Ketut. Yeah. Thank you, Prof. Eni, for the time for me. My question is addressed to Dr. Masteria. Yeah, thank you. My question is about marine organism. Yeah, on my dissertation, I got many organisms around Bali Sea. Yeah, and then I find one kind of organism that is Aglaopenia. That is from Silentrata. The form of the organism is look like seaweed. Yeah, but that is an animal, of course, as you know. May I consult or know about the information? What is maybe how to make use this organism? I want to know actually, because I can't find it yet. Whether it also can be to use for cancer or not, something like that. Actually, I consult to you about that. I hope you can make me the information about that. Yeah. Thank you, Prof. Eni. Thank you.
Thank you, Prof. Ketut. Yeah, Prof. Ketut is from Udayana University, which is State University of Indonesia in Bali. Yeah. So he's showing the picture. Okay, but that's good. I'll take for this session 3 questions first, and then later on when we have more, and then I can open for the next session for question and answer. So one for Professor Ilya Raskin, one for Professor Ernawati, and one for Dr. Masteria. So for Professor Ilya, it's Nadira. Is that Nudira? Nadira. Okay. Please.
Right. So I believe there were 2 questions. The first was about sort of the junk foods which dominate the market, and I can absolutely assure you that this is not only the problem of developing countries, or it's a major issue in developed countries in the United States. And of course, the way to deal with it is through educational programs and through socially responsible behavior of the companies, of the food companies, I think, and this is an issue which obviously leads to major problems of metabolic syndrome in the United States, in developed countries, and also in developing countries. So the second question, I believe, was about the cost. Will functional foods be more expensive? And I think the answer to that is they should not be, because clearly once we have the lettuce, it's just like we have the seeds of, for example, dwarf wheat or other major agricultural advancements. Inherently, it costs the same to grow nutritionally enhanced lettuce and regular lettuce. So it's all about, boils down to social responsibility and government and policies which governments use to educate people and to create incentives for the companies to sell healthier food instead of the junk food. And that is something which should be solved through, I think, governments and education. So they don't have to be more expensive. Thank you.
Thank you, Professor Ilya. Are you satisfied? Or okay. Otherwise, you can talk later on. Yeah, did he answer your question? Yes, okay. Bu Nadira, from which university? Merck? Oh, from Merck. Okay, from private sectors. Yeah, from Merck. You know all Merck, the chemicals company. Okay, Bu Erna, please. Yeah.
Thank you, Bu Eni. Thank you for the question, Ibu Astri Zulfa. Actually, in my opinion, if you want local people to use the underutilized fruits, we have to show them the benefit of consume that fruits. If you fail to show them the benefit, they will not consume that fruit. So first of all, we have to make research to prove the medicinal value or the benefit of eating or consume that fruits. And secondly, after that, after they know the benefit of eating the fruits, we can educate them how to consume it, how to take it. Because in our research, we find that consumption of clear juice is less benefit than the cloudy juice. So the cloudy juice is better because it contains dietary fiber and other substances. So first of all is we have to make research to prove the benefit of the fruits. I know Ibu Aci is doing research about the fruits that is the primate for primate feeding, right, Bu Aci? So I think Bu Aci, I know is selected the fruits that to answer the question why the primates or the orangutans take that fruit for their diet. Maybe there are something in that fruits, because in Ibu Aci research or also Erin's research, Erin Vogel is my colleague, and Professor Ilya Raskin colleagues who studied orangutan. In their study, they find that orangutan is smarter than human. Is cleverer than human. So maybe we should take lesson from them. That's why that's what Ibu Aci doing research now. So for your question, I just can give to underline that first of all we have to prove that the consumption of those fruit is benefit. I think that's my answer. Thank you.
Thank you, Bu Erna. But I think she also asked or underlined how to make it sustainable. So how to cultivate also. Is there any effort to cultivate them? Yeah.
Yeah, thank you, Bu Eni. Yeah, the underutilized fruits is going to be an endangered species because it's not used by human. No people want to cultivate it. So first of all, we have to prove that it has beneficial advantage to use the fruits and to cultivate the fruits, the fruit tree. So I think conservation is going in the same way with the research to use to prove the benefit of the tree. I think that's the answer. Thank you.
Yeah. So maybe you don't have to do it in UNAS or partner. That maybe you can be partnering with other institutions that do the cultivations or propagation. Yeah, thank you. So now the question for Pak Masteria to answer. So please.
Thank you very much, Prof. Eni. Prof. Made, yeah, thank you for your...
Prof. Ketut,
oh,
jangan
Prof.
ikut
Ketut, Prof. Ketut. I'm sorry. Prof. Ketut, right now working with marine natural products is very easy because we have the marine database, we call it marine leads. So in the marine lead, we can search by organisms and also by molecular weight of the compounds that we isolated, that's new or non-compounds. And if you search by species, and you see me the picture of your marine organism, I think that's gorgonians. And the gorgonians mostly in the paper and in the database, they have the potential for anti-inflammatory, and some of them also potential for anti-cancers. But mostly the researcher working with the gorgonian is for anti-inflammatory. So if I can help you to check, then ask my colleague to search in the marine leads your species, but I need the exact species, your organisms, so we can check what the potential of these compounds. So when we can also know what the compound inside these organisms, and yeah, if they're interesting, then we can working with this organism, and hopefully we got a new compound. Sometimes also when we check, when we do the marine natural products, we not actually directly to do the extractions, but we do the small extractions, and we use the LCMS to get the information, the molecular weight inside of the organisms, and each of the molecular weight we check by marine lead, and we know that we inside the species have the potential non-compound or new compounds. Yeah. That's. Do we need the molecular analysis to check the link? So you mean for DNA, taxonomy? I think you can send to BRINS, the Research Center for Oceanography. So they have the people taxonomies for gorgonians, and they have the people also taxonomies for sponge, and you can send to them to check your species. I think if they are gorgonians or if they are seaweed or whatever, so they will confirm this the name of the species. Yeah. Yeah.
Pak Ketut, can someone give the mic to Pak Ketut so that the online participants can also listen? Yeah.
Thank you. Yeah, I have checked this organism to LIPI. Yeah, so the name is Aglopenia species. Silantrata, but I didn't check it using a molecular analysis yet. So as you think about that, Do we need about that?
Yeah, I think right now we just only need the taxonomies first. So sometimes because there is not much more problem with the DNA taxonomies. But the problem if you're working with like some of the sea cucumbers, or if you are working with some of the bacteria, maybe you need to check with DNA first. But for the marine species, maybe the first thing you need to using the traditional taxonomies first, and there is some of the books for the identification of gorgonians. I forget the name of the professor in the Research Center of Oceanography. They have the book that they are collected the gorgonian from Orange in Indonesia. I forget the name of the professor. Maybe I will. Then she already released the book 3 years ago, so still new. Then I think I have the book in my computer. Maybe I will check it later. Then I give it to you if you need.
Is that Professor Sudarsono?
No,
okay, that's okay.
Yeah,
Later on. Okay, thank you.
Yeah, thank
Maybe
you.
later
Thank
on
you.
you can discuss during coffee break or tomorrow. Yeah.
Sudarsono, hard coral.
All right. Okay. I think I'm going to give the chance to the online participants. Can someone help me to check whether there's question? No? No questions? Okay, at least I already offered the chance. Okay, I know you all very tired. I can see from someone sitting next to me. Yeah.
Oh, no,
yeah. Well, I was going to give the chance on one burning question actually for audience in here, but I can give it to you.
It's really fascinating because, of course, I'm going to start from the beginning. Just a simple question. Do you know actually where those natural products are made? Are they made by the sponges, or are they made by symbiotic microorganisms which live inside the spongi, which possibly can be culturable, and then you can solve some of your supply issues?
That's a very interesting question. And Tenalasin, he have I forget the name of the bacteria, the species for the bacteria. So when you isolate the bacteria from Tenalasin, you can always isolate this bacteria. And when you make the fermentation of this bacteria and do the extractions, and you do the extraction of the Tenalasin, they produce the same compounds.
So it's a bacterial product, not the sponge.
Yeah. And there is also like sponge, like the nudibranch. Nudibranch is the small species, the small one with I think they are only small like this, and with some of the color, variety of color. Sometimes they're red, and sometimes they're blue and red, something like that. And they're living in sponge. When you do the isolation, the nudibranch, so the part of the nudibranch living in sponge, they produce the same organism. So that's why I mentioned before, the marine organism of the marine species is very interesting. Sometimes we don't know who is the exact who is the producer of the compound. Sometimes of the bacteria, or sometimes of the sponge. But that's the case of the sponge. But in the case of the soft coral, have the symbiotic, the microalgae. I forget the name of the microalgae. So when some of the researchers from Taiwan do the culturable of the soft coral without the microalgae. So they isolated the same soft coral from the sea, and they do the extraction together. And the soft coral, without the microalgae, they don't produce the amount natural products than the soft coral from the sea. So I think that's really, fascinating for the marine organisms. Then that's why I still working with that. Then I think terrestrial is very interesting also, but I think the marine organism is more, for me, more interesting than terrestrial plants. Thank you.
Yeah, thank you. I couldn't agree more with you because still a lot of research to be done related to marine organism, especially because Indonesia consists of more waters than land. So yeah, that's why we need to collaborate. So here I open for collaboration also in marine research, including plant marine research. So I'm not going to summarize the talk for this session, but I just want to underline that research need a long-term funding and also program. So we all heard from Professor Ilya Raskin also the current research and also future research until you really have the product, a commercialized product. So and also collaboration is very important. So you can see from the slides at the end of the speakers, slides of the speakers, a lot of logos of the collaborators and funding agencies, and also from this morning session. So with that, so I would like to thank all of you. The three speakers here, please join me to give them an applause, round of applause. Thank you.
Ladies and gentlemen.
So, yeah, thank you very much. Yeah, so I'll give it to the Master of Ceremony back. Thank you.
Thank you, Professor Annie, for chairing the session. And of course, thank you very much to our invited speakers for incredible presentations of your research. And now we would like to present you token of appreciation. Therefore, please to our speakers and moderator to come to the front of the stage. And we would like to invite the representative of our sponsors today, PT ITS, Chail Jedang, and also PT Indolab, to present token of appreciation to our invited speakers in plenary session 2.
There are a lot of paper from natural productions. I think the next year I will go to US.
Once again, we would like to invite our sponsor, PT ITS, Chail Jedang, and PT Indolab, to present token of appreciation to our invited speakers. So all of our male speakers for this conference receive udang, the traditional Balinese headwear. And thank you very much to our sponsor who have handed the token of appreciation to our invited speakers. Thank you very much, and now we will have a photo session. together with the invited speakers and also the representative of our sponsors. 1, 2, 3. One more. 1, 2, 3. Bali. Okay, that's all. Thank you very much to our invited speakers and sponsors, and also our moderator. Please kindly return to your seat. All right, ladies and gentlemen, on the next agenda is our third plenary session, but before we move to the final plenary session for today's event, we would like to have a short coffee break for 15 minutes. Please enjoy the refreshments provided by the committee, and then in 15 minutes we will start the final session of plenary talk with our invited speakers. Thank you very much. Once again, we would like to remind all of the presenters and speakers that we will have a short break for snack time, coffee time for about 15 minutes, and afterwards we will begin our final plenary session for today. Thank you. Kami silakan kepada para peserta konferensi, para pembicara untuk menikmati jamuan yang telah disediakan oleh panitia dan hotel di luar ruangan ini. Kita akan mulai acaranya dalam 15 menit. Terima kasih.
Kami percaya bahwa setiap hasil yang dikeluarkan harus sebuah aksi terbaik dari pendidikan lain sepenuh hati. Pendidikan layanan terbaik menjawab tantangan dan kebutuhan agar Anda dapat menemukan pilihan sesuai kebutuhan Anda. Kami merupakan institut laboratorium dan farmasi yang dapat terus mengembangkan sejak tahun 1970. Fokus kami adalah untuk membangun Indonesia lebih mandiri dengan memanfaatkan teknologi berkualitas tinggi di dalam laboratorium dan farmasi. PT 8th Indonesia selalu berdedikasi untuk perjuangan dari teknologi berkelanjutan agar dapat menerapkan dengan maksimal seluruh masyarakat. Dengan pengalaman yang dimiliki oleh PT 8th Indonesia telah berkoordinasi melakukan industri hingga universitas-universitas Indonesia. Bersama dengan brand pengembangan ini, kami berkolaborasi melaksanakan yang terbaik untuk Anda. Memberikan pengetahuan baru, sangat hadir untuk membantu, tanpa mengorbankan diri, untuk terus membantu Anda dalam menyediakan produk-produk Anda. Dengan sumber daya manusia yang ahli pada bidang bidangnya, kami saling berkolaborasi membangun tim yang berintegritas dan selalu dan akan terus melayani Anda dengan sepenuh hati. Dalam hal penjualan produk, PT 8th Indonesia memiliki pelayanan penjualan dalam negeri, memberikan layanan terbaik dan membantu menemukan solusi terbaik untuk Anda. Kami adalah partner yang dapat mengandalkan untuk membangun masa depan. Kami terus menatap ke depan untuk terus berinovasi, membangun visi besar, membangun teknologi industri laboratorium dan farmasi yang lebih baik.
I don't know how they connect. Iya. Okay, I think.
Oh, she's there. Can she see us? Which camera is it back?
they connect this, that they will, but it's not all. Oh, yes. They will turn this on.
Thanks. Hello, Erin. Can you hear me? Hello. Okay, everything is fine, yeah?
Okay.
Okay.
Should I try to share my screen first?
Okay, let's try.
So she can hear. Okay. It says the host has disabled screen sharing, so I think you have to allow me to share my screen and making me a co-host, maybe?
Okay then, see you then. We will start at 3:50.
Hold on a second. Let me get my slideshow to work. How does that look to you?
That's fine.
Can you see just the slides?
It doesn't look full. Is it the what do you call it? Like the background thing to it?
The frame or things. They will adjust the template.
Oh, they're adjusting the template?
Yes.
Okay, just 1 second, Aaron.
Okay. That looks good.
We can't see, but we can't see the whole slide. The slide is longer.
Where did you go?
Can you move to the next slide, Aaron? It's showing like, I feel like it's showing, okay. Okay.
I think it's fine. I think it's okay.
Okay. Could you go to the third slide, Aaron? Just indulge me.
Yes.
Yeah, it is cutting off.
Is it? Because on my screen it looks...
Do you see the whole, like, does your presentation look like a square or a rectangle?
It looks like a rectangle. I can see, like, on the thing, like, I'm looking at the admin computer, and it looks like the ad on screen. Looks correct. What are the admins with
the Zoom looks like line, but in this screen looks like square.
Oh,
Yeah.
can, but on this screen, are people going to be able to see the whole thing?
Yeah. On the Zoom, the people look.
What about us
display,
here?
Landscape.
Oh, okay.
White screen. Sorry, white screen.
And then what about here, like for the crowd in the public? Can that be adjusted or no?
Do you want to display the crowd?
No, not the crowd, just what I'm worried is that the left part of the PowerPoint is being cut off right now. Does that make sense? Porque, so the Zoom is fine, Aaron. It's what's being shown, what's physically being displayed here. It's cutting off the left part of the PowerPoint. If it can't be adjusted, I'm sure it'll be fine. But on Zoom, it should look fine.
And Zoom looks fine because I can see the admin screen, but yeah, maybe I can adjust it there. I don't know. It was really good, right?
It's nice to see you. Oh yeah, good point. Oh, Will and Issa everybody just walked in. No, it's fine. Taking a photo. Oh, just kidding. Hi, Erin.
I can't see that.
Can you see me? Cute photo.
Oh, it looks a little bit more.
Background ya, kenal ya. Tadi saya telepon-telepon sih mau foto.
Okay, I'm gonna put this down. I'm gonna put this microphone down because it's giving me. Do you want to say hi to your advisor?
Hello, Aaron.
Hey, Will.
Hi, Erin. It's Isa. It's a whole meeting. Yeah, we'll send you a picture.
Please do. I'll see you soon.
But the information is still full, or just like the designs of the presentation that are cut?
I'm not fully certain, to be honest with you. Erin, can you do me a favor and go to the 5th slide, please? I think there's nothing left to the orangutan image, right?
Nothing left to the orangutan image, nope.
Okay, so yeah, I think it's fine. I think it's fine. Cool. No, it's fine. It's fine. Thank you so much. Okay.
Bisa balik ke itu lagi sponsor. Okay. Suara at the top. Yes, you have to. I don't, right? No, you have to. I will invite you. Oh, everybody will come.
178. Fokus kami adalah untuk membangun Indonesia lebih maju lagi dengan memanfaatkan teknologi berkualitas tinggi di bidang laboratorium dan farmasi. PT Hartek Indonesia selalu berdedikasi membukakan jalan bagi teknologi terbaik dunia agar dapat diterapkan dengan maksimal sesuai kebutuhan Anda. Dengan pengalaman dan ilmu yang kompeten, PT Hartek Indonesia telah berkontribusi memajukan industri hingga universitas-universitas di Indonesia. Bersama dengan brand terkemuka di dunia, kami berkolaborasi mempersembahkan yang terbaik untuk Anda. Memberikan pengetahuan baru, saling hadir untuk membantu, tanpa mengorbankan mutu, menjadi solusi bagi Anda dalam menyediakan produk-produk Anda. Dengan sumber daya manusia yang ahli pada bidang-bidangnya, kami saling berkolaborasi membangun tim yang berintegritas, yang selalu dan akan terus melayani Anda dengan sepenuh hati. Dalam hal penjualan produk, PT Hartek Indonesia memiliki tim layanan purna jual yang handal, memberikan layanan terbaik dan membantu menemukan solusi terbaik untuk Anda. Kami adalah partner yang dapat diandalkan untuk membangun masa depan. Kami terus menatap ke depan untuk terus berinovasi. mengembangkan visi besar, membangun teknologi industri laboratorium dan farmasi yang lebih baik.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen.
Well, ladies and gentlemen, we are about to start our third plenary session. So please make sure that if you have friends or colleagues that are still outside of this hall, please make sure to contact them and invite them to come back to this hall as we will begin the third plenary session.
Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat.
To participants who are still outside of this hall, please come back because we will start our final plenary session. Kepada seluruh peserta pembicara yang masih berada di luar ruangan ini, kami silakan untuk kembali untuk kita lanjutkan acara selanjutnya, yaitu sesi pleno ketiga.
Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani, agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia.
One final call to our participants that are still outside of this hall.
Dalam memajukan industri dan penyaluran produk benih dan bioteknologi.
Please come back to this hall as we will start our third plenary session with our invited speakers. Thank you.
Baik ekonomi, sosial, dan lingkungan.
Food doesn't come from the grocery store. It comes from the land, which means if we want this, we need to protect this and them. How can we produce enough food while also nurturing the land and the farmers who grow on it? 2 words: sustainable agriculture. That means the soil stays healthy, the environment stays healthy, and the farms, you get it. So what does that look like? At Corteva Agriscience, it looks like seeds that are adapted to different environments. So farmers not only conserve much-needed water, they increase the likelihood that seed will make it to harvest. Our biopesticides help farmers control pests and protect crops naturally. And it's not just our products.
Well, ladies and gentlemen, may I have your attention, please?
Maximizing yields,
To all
quality.
participants, guests, and also speakers, please kindly be seated as we will start our third plenary session.
Sustainability. It's a word you hear a lot. But what does it really mean? At Corteva Agriscience, sustainability is more than a word. It's who we are and what we do. We're in it for the good of farmers.
Well, distinguished guests, ladies and gentlemen, thank you very much for staying with us until the last agenda in this conference for today. After having snack time, coffee time for 15 minutes, let us continue our plenary session, which is the final plenary session for today, with the theme of biodiversity on plant, animal, and environmental conservation and improvement that will be delivered by our invited speakers. And this session will be led by our moderator. Our moderator is received her PhD degree from University of Erlangen-Nuremberg, Germany, and currently she is a researcher in Indonesian National Research and Innovation Agency. Without further ado, we would like to invite Dr. Renat Bernadetta Rina Hasti Lestari. Well, ladies and gentlemen, please give a round of applause to our Dr. Bernadetta. To Dr. Bernadette, time is yours.
Thank you, Master of Ceremony, distinguished guests, ladies and gentlemen. Last but not least, we come into the last plenary section with the topic biodiversity and plant, animal, and environmental conservation and improvement. In this session, there will be 3 speakers. The first is Philip Brandle, and the second is Paula Rode Brandle, and the third is Erin Vogel. I will introduce you. They should be here, but unfortunately there is sad news that one of the relatives from Philip has passed away and they could not come here. Yeah. I will read the CV of Philip. Philip is a professor in Pennington Medical Research Center, Louisiana State University, Baton Rouge, Louisiana, USA. You can write down the contact and the mobile number. The research interest of Philip is prevention of weight regain following intentional weight loss and the translational of surgical and non-surgical intervention for severe obesity from clinical trials to application by healthcare providers and insurers. Educational background, he has finished his PhD in clinical health physiology from the University of Georgia in Athens, Georgia, USA in 1980. Okay, he has also finished his study in clinical physiology, residence at Medical University of South Carolina in Charleston, USA in 1979. His executive experiences, he holds a John McHaley Distinguished Professorship and is chief of the Behavioral Medicine Research Laboratory at Paddington Biomedical Research Center. He also has shared the PowerPoint. So for the participant on site, you have received the copy of the presentation. Please read. If you have any question, you can raise the question later and we will write down and we can forward to him. His presentation is, next please. Oh, next is Paula. Paula is his wife. She is the associate professor in the Research Integrity Officer, Pennington Biomedical Research Center in Louisiana State University. Her research interests are research ethics, research administration, the teaching and training of ethical research practices and professional behavior, human research protection, and research regulatory issues. She has finished her studies in University of Tampa, Louisiana State University, and she has finished the PhD in Louisiana State University in 2003. She is the director of Primary Care Program, Louisiana State University School of Medicine, New Orleans, Louisiana, from 2020 until present. Okay, for the participant online, I will share the presentation of Philip and Paula later in the Zoom chat. And then the third presenter is Professor Erin Fogel. She is an associate professor at the Henry Rutgers Chair Professor, Department of Anthropology, School of Arts and Sciences, Rutgers, the State University of New Jersey. She has finished her PhD in Stony Brook University, Ecology and Evolution in 2004. Her position now, there are a lot, but we have selected as a director of the Center for Human Evolutionary Studies at Rutgers University, and she's also graduate program director. She is also Henry Rutgers Chair Professor, and she's also the Associate Research Professor at the Department of Anthropology, the George Washington University. Okay, now I will invite Erin for the talk. So Erin, okay, can you hear me?
Yes, hello.
Hello. Yeah. Okay. How are you?
I'm doing great, thank you.
Okay. Wow. We really appreciate that you can present online. I think now in USA is 3 o'clock, right? Or 4 o'clock?
4 o'clock.
Okay, okay. Thank you very much. We appreciate that. And now the screen is yours.
Thank you. Does that look okay to you?
Yes.
Excellent. Okay. All right. Well, thank you so much for staying this late in the afternoon for my presentation. If at any time the audio goes out, I'm sure I'll receive a message from my graduate students who are sitting there in the audience today. I really wish I could be with you all in Bali and thank the organizers, and in particular Ibrahim, who I was fortunate enough to spend 6 months with here at Rutgers University. So thank you for organizing this wonderful event. And today I'm really excited to present to you a summary of our long-term work examining the importance of diverse diets for wild orangutan and how these diverse diets influence health outcomes in these incredible organisms. But before I get started, I want to thank my collaborators and long-term partners on the Tawana Orangutan Research Project, Dr. Suchi Utami-Mboko and Dr. Chatan Mitra Satia from Universitas Nacional, and Dr. Marina Valorda from the University of Zurich. The data I will present to you today would not be possible without our amazing field staff and the over 200 students who have collected data at Tawana as part of this project over the last 2 decades. And you may be thinking to yourself, why study orangutans? They split from our own lineage about 14 million years ago, far longer than chimpanzees. But through my research, we have realized that we humans have a lot in common with orangutans when it comes to health and diet. For example, we know from captive research that orangutans have a greater propensity to store fat compared to other ape species, particularly when they are exposed to an energy-rich environment. An earlier research by my colleague, Dr. Cheryl Nutt, showed that during episodes of extreme food scarcity, orangutans burn their body fat, as evidenced by ketone bodies in their urine. She found that in the Alaskan forest, orangutans store fat during the high food periods and burn fat reserves during the low food periods. And over the last decade, there have been several studies that have highlighted orangutan and shown that orangutans have physiological and genetic adaptations that help them cope with these energy deficits. For example, Herman Ponser and colleagues showed that orangutans have significantly reduced total energy expenditure for a given fat-free body mass. Laser pointer here. So you can see here, hopefully you can see my laser pointer. On the y-axis is total energy expenditure, and on the x-axis is fat-free body mass. And what you can see is that orangutans, highlighted here in orange in this graph, have significantly lower total energy expenditure when compared to gorillas, chimpanzees, and our own species, humans. And if we look at the resting metabolic rate, the basal metabolic rate of orangutans compared to humans and chimpanzees, it's very clear that they're significantly lower than what we would expect based on their body mass. Thus, these energy-conserving adaptations have also often been attributed to unique ecology in Southeast Asia. The forests of Southeast Asia are characterized by their unpredictability in both the timing and the magnitude of these fruiting peaks. This is often referred to in the literature as a feast and famine ecology. And it has been hypothesized that even our own species evolved in a very similar environment, characterized by unpredictability in food availability throughout most of our evolutionary history. Thus, as the largest arboreal mammal in the world, orangutans, as preferred frugivores, have to figure out how they're going to survive during these periods of extreme fruit scarcity. So our research over the last 2 decades at Tuanan has really focused on how this unique ecology of Southeast Asia influences the dietary strategy of wild orangutans in a peatland habitat, and the types of physiological adaptations they have to cope with this unpredictability and resource availability. Furthermore, we've looked at the health consequences of this, so today I'm going to talk about all of this with you. Over the last 20 years, we have found that orangutans have incredible diversity in their diets, more so than any other primate species. For example, the Tuanan orangutans include over 230 plant species in their diet, and over 700 unique species of plant-part combinations. This means that for a given species, orangutans consume 2 or 3 or 4 parts, including flowers, fruit, bark, and leaves of the same species. If we compare their diets with chimpanzees in Uganda or the Bali chimpanzee project, we find that orangutans feed on over 2 times as many species and more than 3 times as many plant species part combinations. So if we think about their diet in terms of periods of high fruit availability compared to low fruit availability, we see that during these periods of high fruit availability, orangutans spend 75% of their time feeding on fruit, and they do spend other portions of their time feeding on leaves and insects for protein, and a small proportion of their time feeding on other food items. But it's during these episodes of low fruit availability that they greatly reduce fruit consumption, and they focus their time feeding on items such as leaves and bark, flowers, and other types of vegetation, all of which are known to be more difficult to digest and perhaps have lower energetic return. What we find is that when orangutans transition from the high to low fruit period, the Tuanan orangutans reduce caloric intake by as much as 50%, which is quite a bit. This variation in the types of food items and the high diversity of food items consumed by orangutans translates into incredible variation in the percentage of fats and carbohydrates found in their foods. However, the amount of protein in most food items remains below 25%, except for the cases of insects, as seen here in pink, and young leaves, mostly in this region here. So this is energy from protein from low to high, energy from fat from low to high, and energy from carbohydrates from low to high. And what you can see is that most of their foods have fairly high carbohydrates and fairly low fat, although there are some food items that do have some more fatty, have more fat in them. Protein stays pretty low in most of the food items and pretty consistent. So what are some of the strategies that we found that primates use to cope with this unpredictability overall? One way to think about diet is to think about the balance of nutrients consumed on a daily basis and examine how diet influences this balance of nutrients. Work by my collaborator and colleague, Dr. Jessica Rothman at Hunter College, has shown that as we move from frugivores to more omnivorous primates, including ourselves, to more folivores, we see a decrease in the ratio of non-protein energy on the y-axis, which is composed of carbohydrates and fats, to available protein. So we see a decrease in the ratio from about 14.1 in spider monkeys here to about 2.2 to 1 in more folivorous colobines. So given the variation in the orangutan diet, we were really interested in where orangutans would fall along the spectrum. So we went out to study this at the Tuanan Orangutan Research Station, located in central Kalimantan in the peatland forest along the Kapuas River, right here. The Tuanan program is located in the southwest corner of the Mawas Conservation Area, and this area It is managed by the Borneo Orangutan Survival Foundation and the Department of Forestry in KPHL in this region. Over the years, we've studied over 130 habituated individual orangutans, and the data we're going to present today are based on daily nutritional intakes on over 5,000 nest-to-nest focal follows from adult individuals, males and females. We also are able to quantify the availability of fruit, called the Fruit Availability Index, as the percentage of trees in our monthly monitored phenology plots. The percentage of trees that are fruiting, I should say, in our phenology plots. And our phenology plots are composed of about 2,500 trees that are labeled with a number and monitored every month. To obtain data on the nutrition of all the food items that the orangutans feed on, we often climb the trees where the orangutans have previously been observed eating. We collect fruit samples. We process these fruit samples in camp, and then we send them to Dr. Rose's lab as part of Leafy Brin for the nutritional fact sheets that we receive back from her on all of the different food items they consume. We can then combine these data on fat and carbohydrates and protein with feeding data to estimate daily nutritional intake. We also collect orangutan urine every day, or as often as possible, and we can use their urine to examine their health and estimate energy balance status. Collecting orangutan urine in the wild is certainly not an easy task. And as my students have found, it does take a lot of practice. This has been voted as one of the top 10 worst jobs in science. This graph here represents the fruit availability at our field site. As you can see, there's great variation in availability of fruit, and we have some periods, like after the 2015 fires, where we had 32 consecutive months of extremely low fruit availability. This dotted line right here represents the average fruit availability for the habitat, so about 5% of the trees are fruiting at any one time. So what is the nutritional strategy of orangutans? What we find is that overall orangutans follow a model that we see in most frugivorous primates. We have seen very little variation in protein intake during the high and low fruit periods. But what we do see is incredible variation in daily non-protein energy intake. So non-protein energy, again, are items that are fats and carbohydrates and including fiber. So if we look at the percentage change of protein intake between the low and high fruit period, we see only about a 4% change. But if we look at non-protein energy and the ratio of non-protein energy to protein, we see over a 24% change in non-protein energy and 36% change in the ratio. So during the high fruit periods, orangutans feed on a ratio of non-protein to protein energy of about 11.6, and during the low fruit period, about 8.5. On average, humans consume a ratio of about 7 to 1. So that puts what orangutans are doing during the low fruit period in perspective. So we can ask the question, do humans maintain stable protein intake like orangutans? And the protein leverage hypothesis was introduced by my collaborator, Dr. David Rothenheimer, this is him at Swan, and Stephen Simpson to explain the human obesity epidemic. This graph is based on a case where a subject requires a total energy intake of about 2,500 calories to maintain energy balance. On the x-axis is protein intake, and on the y-axis is fat and carbohydrates. And what you can see is that just like orangutans, humans tightly maintain protein intake. Each one of these dots represents an individual who has been exposed to a feeding experiment. And no matter how many calories are put in front of people and types of foods, humans tightly regulate protein, just like orangutans. The protein leverage hypothesis also predicts that dietary decreased with the rise in obesity. And indeed, if we look at the data from 1960 to 2010 in the United States, the percentage of protein in the average household diet has dramatically decreased over time. And if we look at total kilocalories of intake during the same time period, we see an increase in caloric intake. So a small decrease in protein intake results in a very large increase in total caloric intake. So what do we see is happening with orangutan energetics? Well, I'm going to put this in the theory of energy balance. When individuals, or when people, or orangutans, take in more energy than they expend, so when energy input is greater than energy expenditure, individuals will be in a positive energy balance state, and we would expect to see weight gain. But when energy input is less than energy expenditure, this is when we would expect to see weight loss in a negative energy balance state. So how can we measure these different states in the field? Well, we can analyze orangutan urine samples. And most of this work has been done in my laboratory at Rutgers University in collaboration with Universitas Nasional faculty and students. And we can look at, for example, ketone bodies, a byproduct of fat metabolism, and an increase in ketone bodies indicates a negative energy balance state. We can also look at C-peptides of insulin. C-peptides are produced in equal ratio to insulin, and they're released or cleaved into the urine. So low levels of C-peptides of insulin would be indicative of a negative energy balance state. We also measure urea concentration. So high levels of urea indicate excess exogenous protein, so dietary protein, and or gluconeogenesis, as ureagenesis and gluconeogenesis are two processes that occur in tandem. We've also analyzed nitrogen isotopes in the urine, delta N15, and an increase in delta N15 is indicative of the use of endogenous amino acids, so the use of body protein, so skeletal muscle, and the gluconeogenesis process. So what do we find? Well, we find that during high-fruit periods, orangutans have higher caloric intake, they're consuming more carbohydrates and lipids, and they're over-consuming their calories. During these periods, they're storing fat, and they're also building up their muscle reserves. During low-fruit periods, we see decreased caloric intake, low carbohydrate and fat intake in particular, but protein intake remains about the same. And as I mentioned before, it increases by only about 4%. We see a reliance on body fat for energy, but even more so, based on our nitrogen isotope data, we see that individuals are using body protein muscle for energy via the gluconeogenesis pathway. If we take a look at a little bit more closely at the data, on the x-axis here is total non-structural carbohydrates. On the upper graph is urea concentration, and on the lower graph is delta N15 nitrogen isotopes. So even when total non-structural or caloric intake dips below 3,800 kilocalories, we start to see a rise in urea concentration. And this is not because of a change in dietary protein. But it is, so we start to see that below 3,800 kilocalories, individuals start to use exogenous non-carbohydrate energy sources and endogenous lipid sources for energy, as indicated by urea. But as caloric intake dips below 800 calories, total unstructural carbohydrate intake, I should say, we start to see muscle wasting and a significant rise in delta N15 levels. So it's really when individuals significantly reduce their caloric intake and they're entering into a state of starvation mode that they start to consume their own body muscles. This has been supported by a recent study out of my lab by Dr. Caitlin O'Connell, a postdoctoral fellow who's been working in my laboratory. What she found is that across high and low fruit periods and across all age sex classes of individuals at trauma, we see significant differences in muscle mass between the high and low free periods as measured by the residual of creatinine and specific gravity in the urine. So even dependent offspring, we see a significant decline in muscle mass during these low free periods. So how are wild orangutans adapted to low free periods? We see adaptations for efficient energy acquisition during the low free periods. We know from previous studies that orangutans have reduced energy expenditure compared to other apes and low resting metabolism. From our research, we also know that during low free periods, they actually travel a lot less and they have shorter day lengths. They just slow everything down. We also see gluconeogenesis of amino acids during these low free periods. This is providing glucose to supplement energy metabolism, which most likely maintains insulin secretion, and hence why we don't see any relationship between urinary C-peptides and caloric intake. You may ask, why would they resort to utilizing skeletal muscle during these low free periods? Skeletal muscle is costly to maintain. So perhaps during these low free periods, they can reduce the cost of maintaining this metabolically active muscle tissue. And then during the high free periods, when they're over-consuming their calories and have increased movement, they can then build those muscle reserves back up. So are these episodes of caloric restriction harmful? That is the question. How does it affect their health? And a former student in my laboratory, an undergraduate student who's now finishing up his PhD, Daniel Nemenko, studied this for his senior thesis work. He came to Swan, collected urine samples, and then in the lab, he came up with a method to analyze orangutan urine for markers of oxidative stress. Reactive oxygen species come from both endogenous and exogenous sources, and recent methodological developments and research on oxidative stress have made it feasible to study the relationship between caloric restriction and health in the wild. Reactive oxygen species are produced by energy production in the cells, and they can damage proteins, lipid membranes, and measures of reactive oxygen species have often been linked to biological aging. Oxidative stress is the imbalance between reactive oxygen species and antioxidants. And antioxidants are produced endogenously via enzymatic and non-enzymatic mechanisms, but can also be absorbed from dietary exogenous sources. Several studies have shown that dietary sources of antioxidant-rich foods increase total antioxidant capacity. Oxidative stress is also tightly linked to inflammation, which increases oxidative damage, leading to greater inflammation in this cycle. So when individuals have greater free radicals relative to antioxidants, we see greater increased levels of oxidative damage, and this results in tissue injury and inflammation, and this then results in more free radicals. This cycle has been linked to a number of chronic diseases, including cancer, Alzheimer's disease, Parkinson's, cardiovascular disease, diabetes, and obesity. Several studies on model organisms, including mice, macaques, and mouse lemurs, have shown that decreases in caloric intake lead to increased lifespan. And also when we see, if we look at individuals that are on a control or a caloric restricted diet, those individuals on a caloric restricted diet had fewer age-related morbidity and a greater % survivorship. And even in the study on humans, a very modest decrease of caloric restriction of 25% led to reduced levels of oxidative stress, as was shown in the study on humans. And this here is a measure of oxidative stress that can be reported from blood samples. So what did we find in our outcomes? We found something very similar to these other previous studies. if you looked at the relationship between previous day caloric intake and oxidative damage as measured by 8-OHTG in the urine, we saw that when caloric intake is above the average caloric intake of this population, we start to see a rise in oxidative damage, indicating that these high caloric diets are producing greater oxidative damage and oxidative stress. We also found that total antioxidant capacity is significantly positively related to oxidative DNA damage, such that as oxidative DNA damage, as oxidative stress increases, total antioxidant capacity increases, most likely as a response. So I think what our data are capturing is an antioxidant defense response to increasing free radicals induced oxidative DNA damage. What's interesting is that if we look at our markers of inflammation, like neopterin, we also see a positive and significant relationship between neopterin and oxidative stress. So what we see is that 60% of the variation in oxidative damage is explained by our marker of inflammation. So what are the consequences or implications of this in wild orangutans? Well, work by our colleagues from UNOS and Zurich, like Maria Van Noort and colleagues, has shown that orangutans, compared to any other great species, including humans, represented by this purple line in this gray box here, orangutans have one of the highest survivorships until the age of first giving birth, about 94%. And what we end up seeing is that this high level of survivorship is very similar to the level that we see in Swiss people who have extremely good health care. Other studies, such as this meta-analysis that was done on birds, have shown that those species, so each one of these represents a species of bird, those species that have lower levels of oxidative damage live longer, so they have greater maximum lifespan. So perhaps this fluctuating levels of oxidative damage in these episodes of low caloric intake that these orangutans experience are actually beneficial to them. And maybe we need to really rethink our way of thinking that these periods of scarcity are really bad for orangutans. I would propose that as long as orangutans experience the highs in combination with the lows, this is not good for their health. But we still have to have these high peaks for them to balance their energy over time. So what can we learn about ourselves and our own dietary strategy and health from the research that we've been conducting for 2 decades at TON? Humans are currently in a global obesity epidemic, with many countries characterized by more than 60% of their populations as obese. And this obesity epidemic has severe global health consequences. For example, in many countries, led by the United States of America, we see that over 10% of healthcare costs are related to obesity and the health consequences of obesity, including hypertension, heart failure, diabetes, and kidney disease. We have recognized that the main culprit of this obesity problem is a drastic change in our diet from whole foods to foods that are processed and quite artificial, foods that are high in carbohydrates and high in saturated fats. So what can our research do to inform us about human health? Throughout most of our evolutionary history, it has been inferred from morphological and isotope data that our lineage mostly consumed a diet that was lean, made of lean proteins, and relatively higher in protein than we generally consume today. However, as time progressed and continued, we transitioned from a diet that was more energy limited, as you can see here, with more protein, to a diet that was more protein limited. Over time, even more, we started relying on diets that were extremely high in sugar due to bulk refining and greater transportation of these sugars, and unlimited variety of quantity, foods with unlimited variety and quantity of energy, like we can find in our supermarkets today. So over time, we have moved away from the whole food diets we spent most of our evolutionary history consuming. My colleagues and I have observed that during the low food periods, orangutans consume a diet that is more energy limited, but still maintains a good level of protein. But during the high food periods, they transition to a diet that is more energy rich, while maintaining their protein intake at a relatively stable state. Perhaps this is how we differ, as humans have largely decreased the amount of protein in our diets while increasing the amount of carbohydrates, as shown in this previous figure that I showed you. So are humans like orangutans? I would say yes. We're both susceptible to fat retention when placed in extremely energy-rich environments. And we're both adapted to a feast and famine ecology. And this is most likely why we are susceptible to fat retention in energy-rich environments. The only difference is that orangutans still experience this feast and famine ecology, while countries that suffer from obesity have an unlimited supply of carbohydrate and fat-rich foods available at a constant rate. Perhaps we must rethink our way of eating and stimulate our own metabolism by switching our diets while maintaining a diet of lower carbohydrates and more lean proteins. I want to thank everyone for staying here to listen to me today. Most importantly, I also want to thank all of the people that have made this research possible. Our incredible field assistants have collected about 80% of the data that we're presenting today. Our fire team has helped keep our site going when we've been faced with fires, and our staff and the community members of Plaza and Tuana. I'd also, of course, like to acknowledge all of my colleagues at University Nacional and the University of Zurich, without whom none of this research would be possible. And of course, I also want to thank all of the students and postdocs and researchers who have collected data and contributed to the large-scale project. We also acknowledge all of the funding sources and institutions that have made this research possible by providing us with funding for the research and also providing us with access to laboratories in Indonesia and permitting. So thank you, everyone, for staying here to listen to my talk via Zoom, and I'm very sorry I couldn't be there with you all enjoying the beautiful blue skies of Bali. And I would like to open the floor up to questions, if anyone has questions, if we're doing questions now.
Thank you very much, Erin. Okay, now I open the floor. Is there any question from the audience on site and also the audience from Zoom participant? Okay, then Erin cannot come, but she sent the student here. So I will invite Erin's student, Eva and friends. Can you please come forward?
I think there's one question.
Yeah.
I see one question in the Zoom chat.
Yeah, there is one question. First from, maybe I will open from Rita Andini from BRIN. Yes, time is yours.
Thank you, Mrs. Moderator. Could you please hear me?
I can.
Yeah.
Thank you, Professor Koger and Koger. It's very hard for you, 4 a.m. to be with us. I really appreciate it. Okay, very interesting about orangutan because I had no research in the past about orangutan in Aceh, in North Sumatra. Maybe you have heard from the monster. Yeah, unfortunately, I was also researching about their food. And unfortunately, the food has become very scarce, as we know, because of the rapid deforestation in North Sumatra, especially in Aceh. Based from our previous research, the orangutans were very fond of CBD, maybe you know. This is we call this bamboo ball. So may I know about your research, because I think you do in other places in Borneo, I think. What are their favorite food?
Yeah, that's it.
The orangutan has similarity regarding the food taste. Thank you very much for this mobile.
Thank you. Syzygium is definitely one of their favorite foods. They definitely feed on a bunch of different species within Syzygium. And at Tuanon, they feed, as I mentioned, they feed on a lot of different food sources. There is some overlap between the two. Definitely, when I did some research up at Katambe with some of my UNAS colleagues, we found some overlap in the families or the genuses of the species that they do consume. At Tuanon, it's very interesting. One of my doctoral students, who's actually sitting in the audience right now at the conference, has done some work and found that one of the species they spend a lot of time feeding on, which I don't think they feed on in Sumatra, but I could be wrong, is a legume species, Leucoconchylus. And they spend about sometimes up to 90% of their days just feeding on this one species of legume, where they consume all the different parts, flowers, fruits, leaves. But there's other months when they spend less time on that. But overall, there's quite a bit of overlap in the types of species that, at least in the genus and the family of species that they feed on between the sites. But it really depends on which site also, because Swamp, for example, Balumbing is more of a peat swamp forest, and so there's more similarity, more overlap of the species down there. But then again, in Tuanon, for example, we don't have durian hutan like there is in Katambe or in other sites in that region. So, but there is quite a bit of overlap. And I do think we have to be cautious about saying that these, periods of episodes of low food availability are okay for the orangutans, because as I mentioned, they also have to be interspersed with high food periods. So if you don't ever get those peaks where they can build up those calories and bulk up again, then they're going to go into a state of starvation, which is what we often see when orangutans are forced into, say, monocrops like oil palm and things like that, and they don't have a refuge of a forest to go to. Thank you.
Yes, please, Elia.
Hello, Aaron.
Hi, Elia.
Thanks. The question, of course, working with metabolic syndrome myself, what happens in humans once you get obese, you often get diabetes or hyperglycemia. Is it normal that primates, particularly on high carbohydrate diet, and when they become non-calorie restricted, is there diabetes in primates?
Yeah, so there have been some cases in zoos and in captive settings when orangutans are fed a more carbohydrate-rich diet, including really sweet fruits and fruit juices, and they get really obese sometimes in captivity, and they have things, same kind of diseases, cardiovascular diseases, diabetes, and other metabolic syndromes that we see the same in humans. And I believe also this has been observed in the cats as well, and marmosets in captivity. So there definitely we see that when non-human primates suffer from obesity, similar to humans, they have very similar diseases that are associated with obesity.
Because it will be very easy to measure sugar, glucose in urine, which you collect, right, and figure out what's happening there too.
Yeah, we could measure glucose. We do use these chemistry strips when we do the ketone analyses, and we could also measure the glucose in the urine as well. That's a great idea. We haven't done that, but we get a rough estimate from the strips.
Yeah, well, thank you.
Thank you. Oh, there are my students.
All right. Is there any more question? Okay, if not, we have Erin students here. First is
Eva Hernandez Sanur.
Isa Betancourt.
And Will Aguado.
Oke, apa kabar? Baik, baik. Okay. How long have you been in Indonesia for this research? And can you tell us your experience during your research here? Because being a scientist is not only dealing with science, but also with passion. We must feel the energy in this research, right? So we can improve better and better. I think so. Okay, can you share experience during your research?
Hi everyone. So I have been in the Tawanan Orangutan Research Center / like Indonesia for 7 months, and it's definitely a new experience for me. The peat swamp it's different than anywhere that I've ever been. And I think one of the most exciting things, aside from being able to see orangutans for the first time, is to be able to work with people who have been they know the forest very well for the last 2 decades and the orangutans very well. There's been a lot of firsts, it was my first time climbing trees, it was my first time going into orangutan nests to collect hair. So yeah, there's been a lot of new experiences, and I'm going to be talking a little bit about it tomorrow during my presentation in the afternoon. So yeah, it's been, sometimes it's rough, and it's also magical, and it's a lot of work, but it's kind of like those once-in-a-lifetime kind of opportunities, and I'm really grateful to be able to be part of it, to be able to learn from so much people, and to be able to contribute to the data and the knowledge that is being harbored in Tawanan.
Hello. So I first started off as a research assistant in Erin's lab back in 2015, working on the orangutan project. And my background, though, is in entomology. And when I was there, I was like, oh my gosh, I have to come back and do a project on bugs. And that's what I'm doing this year. I arrived in October and have been looking at the insects that are in the forest there, because there's a lot to be discovered. It's a historically understudied habitat, the peat swamp forest in Kalimantan, because it's very challenging. Sometimes I'm like, I came back, it's so hot, it's so humid, it's so hard to work here, but it's also so rewarding because there's so much life when you walk into the forest there. The water is red and glimmering and sparkling on the tree, on the underside of the leaves of trees. And there's just a lot of life there that's really beautiful and hasn't been fully documented and appreciated. And so part of my project this time around is making an insect research collection. And so far it has been really exciting, and we already found a species of dragonfly that a survey several years ago didn't find when they were here. And it's a species that's listed as endangered, and that's just one thing that we found so far, and there's still a lot of identification to do. But yeah, like Eva said, it's really rewarding to work with the locals there who have so much knowledge of the forest and the species, and it's just a very special site, and I'm grateful to be here in Indonesia working there and learning from everyone and collaborating. So.
I guess I don't have too much more to add. I think Eva and Isa both put it pretty well. It can be rough, but it's just a really great place. It's a really interesting forest that, like they said, is just dripping with life. So many species that are still undescribed. And I think one of the coolest things about being there too is just all the collaborations that are going on too. So we're all studying different things and all learning from each other. So Eva's studying isotope ecology, Isa's studying insect biodiversity. My project is on orangutans, but it involves a lot of plant sampling too. Andrew Aldercott, who's also here, is doing a project on canopy pollinators and plant phenology. So yeah, it's been a lot of fun. It's really cool to climb trees and collect plant samples and see the forest from the orangutan's point of view. And yeah, I think that's all I have to add.
I do want to excitedly say that we are added with Andrew, who isn't able to attend this afternoon. We're the largest Fulbright group to work in Tuanan, and I just wanted to say that it's been really exciting because Will just mentioned, we all work in a different kind of project, but I've learned so much about like insects and pollination and like secondary metabolites in plants, and it's just, it's a really amazing opportunity, and I just wanted to mention that. Kudos.
Thank you very much. Erin, you have very great students. Okay, is there any questions from the audience here or from the online participants? All right. If not, I really appreciate that you have found the spirit or the fun of research that really important, because with that spirit you can do more. Okay, I try to link with the previous session that is about medicinal plant. So my question, maybe Erin can answer also, or maybe the student, three of you. Okay, what if the orangutan is sick? Will orangutan also find medicinal plant to cure himself? Because maybe if human we can go to the doctor, but I think there is no doctor, orangutan doctor. There is no. So maybe is there any way of orangutan to find that? I try to brainstorming. Erin, do you have any idea, or you?
Well, I would say, can you hear me?
We can hear you.
Okay, I would say that there is somebody, I believe, in the audience, Astrid Zulfa, who is a lecturer at UNAS and doing her PhD on this very topic. So I believe Ashi is there right now. She's been working with the Tuanan Orangutan Research Project for 20 years and working with us as a partner on this project. And her dissertation, her doctoral dissertation, is looking at the use of medicinal plants by orangutans. So she's looking at plant species that are used by the local Dayak people for medicine, and then looking at overlap with those species used by orangutans in the forest. So and then looking at what is the chemical composition of these shared species that both humans and orangutans use, presumably for medicine. So Ashi would be a wonderful person to ask about this, as she's been doing collecting data on this now for a few months, or years, I should say.
Okay, then we will invite Astrid. Do you have any comments? Please come forward.
Hey, Erin. Actually, I will present tomorrow about, a little bit part of that.
Sorry, this is too early then. You can keep that answer then.
Yeah.
It
So
will not be surprised.
I hope everyone and the participant will look my presentation tomorrow. Yeah, there is a connection between the ethnobotanic and the orangutan food that can they eat when they health is not going well, like that. So I keep for tomorrow. Thank you.
Okay, so that's the secret then. Okay, so we will listen to Astrid for the answer. Okay, then is there any question? I open question for the participant. Also from the participant who are online. Okay, if not, okay then. Eva and Andrew, you work with orangutan, and okay, Isabel, you work with bugs. So, is there any relation between orangutan and bugs? Maybe are they friends?
Will can answer that one.
Well, because you're doing that. Well, I guess I can start with one answer. That Well, tomorrow I'm giving a presentation on one of the parts of my project where I'm exploring what insects might utilize the orangutan nest as a home or a foraging ground. So I'll talk more about that tomorrow. But then also, yeah, orangutans eat a lot of bugs. And Will's looking into that as part of his project. And actually, it's nice to be at the station at the same time because as Will and even Eva are collecting the food samples of the orangutans, I'm working with them to voucher specimens for identification purposes so we know exactly what species they are eating. And I don't know if you want to add anything about the sampling of the specimens or eating.
I guess the only thing to add would be that termites are actually one of the most consumed food items of orangutans at Tuanon. And it can be really impressive to watch them eat termites because what they'll do sometimes is they'll grab a log by their foot and they'll bring it up into a tree. And sometimes the dead wood can be a couple meters long. And then they'll just sit there for sometimes a very long time, just picking termites out one by one.
Yeah, it's really fun to watch.
It's like a little buffet.
Okay, thank you very much. Okay, I think the answer will be secret because they will present tomorrow, so maybe I cannot ask further question. So I will invite all of you to come to their presentation. And one more question, is there any question? Okay, yes, Ibu Eni.
Hi, Erin. Can you see me?
Hi, Ibu Eni. Nice to see you.
Yeah, thank you for being able to talk to us, to share your research with the students. Excellent presentation, and this is not really a question, but it's impressive that especially I took a note here on the oxidative stress and antioxidants that you did with the team. So I think that can be explored further because there are OS things we also measured for plants, especially my team also doing that in several plant species. So that would be good to match. And here, one of the speakers is from Kyoto University, Professor Kamita Kahara. When I discussed with him that orangutan actually eat, I mean, they are very picky in eating the food, I mean, from the plants. And because of the nutrition concern of the orangutan. So he's very interested in that research also, so that maybe we can link together with Japan, Kyoto University, and like broaden up our networking. Thank you, Erin. Yeah.
Thank you, Ibu Eni. I would love to further explore working with you. I think that looking at antioxidants in the foods that the orangutans are actually consuming would be really wonderful. I know the Jewish, there is an assay for antioxidants, but it'd be really interesting to explore what types of antioxidants are in these different plant species. So I would be, as always, very happy and honored to collaborate with you and anyone else on this project. So thank you.
Thank you. Okay, enjoy. I think it's not really enjoy. You have to go back to sleep, right? This is still 5 a.m. Okay. Okay. Thank you, Erin. Hi.
Thank you.
Okay, there is one question.
Bapak Eni. Hello, Erin. How are you doing?
Hi, Fajito. It's nice to see you. I'm doing well. Thank you.
Yes, I just want to know whether in your study area there is indication that orangutan have kind of staple food instead of the other foods. Because I mean staple food means that every day they have to find that food. Like
we are Indonesia, rice is our staple food. Do you have indication on that kind of things? Because in Sumatra we have this kind of things, figs or strangle figs is a kind of staple food in that area. Thank you.
Yes, that's a great question. And yes, I know rice is the staple food in Indonesia, and Pajita does not eat a lot of rice when he goes into the United States for 6 months.
I lose my weight, 9 kilo in 6 months. Okay.
But yeah, so in Swan, as I mentioned, the orangutans feed on, and Will Aguado, who's sitting up there, is actually writing a manuscript on this right now with our INS colleagues. But in Swan, we find that orangutans generally feed on this one species is definitely what we would call a staple fallback food, more or less. They feed on it year-round. It's Leucopapa polycarpus, and they feed on it different food items from this plant year-round. So I think that Swan, at least, this is definitely a staple food source.
Yeah, would be interesting if we investigate the compounds, the chemical compound of that staple food. Thank you.
Yes.
Okay, there is one more question.
Hi, Erin. I have a question. You talked a bit about the orangutan behavior changes during low fruit and high fruit periods and how they are less active during low fruiting periods. And I'm wondering, have you guys noticed there being more or less fighting going on during the low fruit versus high fruiting periods? That's my question.
That is a really good question, and we haven't, because rates of aggression are so low, it would be really, we haven't looked at this exactly, but it would be interesting to look at. Rates of aggression are very low. But I will say anecdotally that after the 2015 fires, we lost, about, I don't know, I think it was like 60 hectares or 80 hectares of the study area. And the individuals in the north were compressed down into the south, into the east. So what we saw is that females that normally had their home ranges in the northern area, because of the fires, were pressed downward, and from the south they were pressed upwards. The individuals' home ranges were kind of retracted, so they had to overlap, and everybody kind of shifted their home ranges. And we did see, certainly over those 4 years after, 3 years after the fire, when fruit availability was low, we saw an increase in aggression or aggressive interaction. In one case, we even observed that a female killed another female, which was most likely with a male. It was most likely over a territory. So anecdotally, I do think that it could induce more competition. We haven't sat down and actually analyzed the data yet, but that would be a really interesting project to look at.
Thank you.
Thank you.
Okay, I think it's very interesting discussion, but we are limited with the time. I will not conclude, but I will summarize a little bit that we can learn from orangutan. And yeah, so it is necessary that we can conserve the biodiversity of plants, animal, and environment. And I hope this conference will open collaboration, like Bu Eni has said, that maybe Rutgers Erin with also Kamitagara Sensei, and also Brin, also KBI, because this is one of the goal of this conference, as stated by Pak Mastor, the president of KBI, that we hope that this conference will open collaboration. Okay, that's all for today, and I will invite to give applause to Erin and the students. And I give the time to the MC. Thank you.
Thank you so much, Dr. Bernadetta, for chairing the session, and thank you to Professor Erin for your wonderful presentation and, of course, active discussion. To all the participants, also thank you very much. To Professor Erin, please kindly stay online and connected with us via Zoom, because we would like to present you token of appreciation in a symbolic manner. Therefore, we would like to invite our sponsor, the representative of Indotech, and we would like to kindly ask the students of Professor Erin to act on behalf of Professor Erin to receive the token of appreciation. Therefore, we would like to invite the students to come to the front of the stage, and then we would like to representative of PT Indotech to give the token of appreciation, and we also would like to invite Professor Annie to accompany our sponsors in handing the token.
Erin, this one only one, only one, but to be received by your 3 students, but just on behalf of you. So it's yeah, Pak Indotech, you can give it, yeah. So it's something, yeah, in the middle maybe, to be in the middle, and it's definitely a lady, because this one is a Balinese fan, yeah. So yeah, thank you. You can open it so that Erin can see. Yeah, you can. Yeah. 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 So this one, can you see it? Yeah. Yeah. So this one made of undak, we call it. If it in Java, it's batik, like I'm wearing now, but this one undak is typical Balinese. Yeah, so all different type of undak. So I hope you like it. Yeah, so for summer in in New Jersey or New Brunswick. Okay, thank you very much. Okay, bye. Thank you.
Thank you very much, and we also would like to have a photo session. Is there already is photographer already take the picture?
We need to take picture with
we need to take picture also with Professor Erin. So please, Professor Erin, please stay connected with us, and to the committee, please show Professor Erin's screen. Yeah. And then we would like to take picture in front of the LCD screen, so Professor Erin can be on the frame as well. Okay, we will say Bali in 3, Professor Erin. So 1, 2, 3, Bali. Okay, thank you very much to Professor Erin once again for your wonderful presentation, to the students of Professor Erin as well, and to also to our moderator, to Professor Annie and our sponsor. Please kindly return to your seat. Well, ladies and gentlemen, the final plenary session we just had is the last agenda for today's event. We still have our second day, which is tomorrow, and we will have more presentations from our invited speakers, and then we also have parallel sessions, which we will have participants that submit their research papers. So we hope that you prepare your presentation well and to see you in your best condition tomorrow. We will begin our second day at 7:30 a.m. for registration on the same venue, Prime Plaza Hotel Sanur, in this location, in this hall. Once again, we would like to thank all of the honorable guests, sponsors, respectable keynote and invited speakers, participants that already that are very active in discussion, that makes this event very lively. We apologize for today's inconveniences and see you again tomorrow. Om Santi 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 Om. Wassalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya.
Dari panitia ada pengumuman nggak? Nggak ada? Housekeeping announcement? No? Everybody already registered to Bali Botanic Garden on the day after tomorrow. Sudah semuanya daftar ya? Kalau yang belum daftar,